Yesterday was filled with engagement. Yes, sir. Sure, sir.
So, இங்கிருந்து கனெக்ட் பண்ணிடலாமா? Clock 9:30 செட் பண்ணுங்க. இல்ல சார். Meeting 25 minutes. Jayakumar. 25 minutes meeting. Huh?
Without any further delay, let's together welcome the session Dr. Arunachalam for the further proceedings. Over to you, sir.
Very good morning to one and all. So, in the morning session, we have a very dynamic and young faculty member, Professor Dr. Jayakumar from Pondicherry University. So, sir has expertise in green organic synthesis and development of reaction methodology and carbohydrate synthesis. So, with this brief introduction, I request Dr. Jayakumar to handle the session. Welcome, sir.
இப்போ ஜஸ்ட் செக் நீங்கள் ஸ்டார்ட் கொடுத்துட்டீங்க அப்படின்னா இங்கேருந்து டேரெக்டாக வந்துட்டு இது பண்ணிக்கலாம். பண்ணிக்கலாம். ஓகே, ஸ்டார்ட் இது வந்துட்டு லேசருங்க சார். ஓகே, ரைட். இதான் பண்ணலாம். Yeah, good morning to all of you. Thank you, Arunachalam, for the introduction. Basically, that we are working in different area of research that including carbohydrate synthesis. Today, my talk will be palladium-catalyzed Hague-type coupling reactions for the synthesis of RLC glycosides. In fact, we work initially that we worked on the N-nitrosamine chemistry, then the amide activation reactions, then sulfoxamine chemistry. Then we have also worked on the carbohydrate synthesis. In fact, out of this, all the 4 fields that our interest is to work on the carbohydrates chemistry, particularly we focused on the C, O and S glycosides. So I'm very happy to give this talk in front of the eminent professor, Professor Jayaraman, who is also a carbohydrate chemist. So we have worked on the O glycoside synthesis and S glycoside synthesis and protecting group manipulations and the C glycoside synthesis. Today my talk will be focused on the synthesis of C glycosides, very small portion I would like to explore over here in this conference. So there are 4 major biomolecules in living organisms, including nucleic acids, proteins, lipids, and carbohydrates. Out of the 4, the carbohydrates are more complex because it's a branched and having multiple hydroxyl groups with the stereochemical enrichment, and they involved in a lot of biological process as listed over here, and most of the sugars are present on the cell surface of the bacteria and humans. Okay, there are 4 type of sugars we could see in the natural sources. That one is O-glycoside. When anomeric carbon is connected to the oxygen, then it is called O-glycoside. Then when the nitrogen is connected to the anomeric carbon, then it is called N-glycoside, and similarly S-glycoside and the C-glycosides. So out of these 4, the C-glycosides are more stable because it is a ether linkage, and all others are like something like an acetal formations over here. So because of the stability, so many O-glycoside have been mimicked to can act as a surrogates for biological active compounds. Many potential therapeutic agents have been developed based on the C-glycosides. For example, there are 2 types of C-glycosides are known. That is, one is alkyl C-glycosides and aryl C-glycosides. If you see the natural product, that most of them belongs to the aryl C-glycoside, and they have various biological properties. So here is some of the examples where some of the aryl C-glycosides were used as a drugs for the treatment of diabetics type 2, SGLT2 inhibitors. Basically, these compounds help in excretion of glucose through the urine by blocking the reabsorption again back to the blood basically. So therefore, the C-glycoside, chemistry of C-glycoside became very important and also like interesting for the chemist and medicinal chemists. If you come to the aryl C-glycoside, there are 2 types of aryl C-glycoside. One is the common one where you have a hydroxyl group at the second position. It's called 2-oxy-aryl C-glycoside, or generally it's also called aryl C-glycoside. Some of the compounds like natural products contains the, sugar unit without the second hydroxyl group. Therefore, it's called 2-deoxy-aryl glycosides. Almost both of them are equally present in the nature, and also displayed various biological activities including anti-cancer, antiviral and antibiotic activities, etc. basically. So today I will try to explain the synthesis of some portion of 2-deoxy-aryl glycosides. Basically, the glycol serves as an important starting material for the synthesis of aryl glycosides, and which can be easily synthesized from any sugars. When you are having normal sugar, which you can do 2-3 steps to get the glycol. So from the glycols, we will be able to convert the glycol to like C1 functional glycols, through which you can do Suzuki type of coupling to make the arylation, or you can directly take the glycol without any functionalization. Then you can do the simple Heg type of coupling over here. And our interest is focused on this one because it's more simpler. So here is the example of C1 functional glycols where the synthesis of aryl glycosides have been prepared. For example, that you can take a nucleophilic glycol where the anomeric carbon is attached with metals like zinc, boron or tin, and you can use electrophilic aryl sources to make the coupling reactions over here. Or you can take the electrophilic glycol like iodine or phosphonate containing compound, then you can make the coupling with the nucleophilic aryl sources like boronic acid or tin compound in the presence of palladium, and you will get this C1 aryl enes, and which can be converted by the reduction. You can get the 2-deoxy-aryl glycoside, or by hydroboration reaction you will be able to get the normal glycosides basically. So these are the method, but what is the major problem of this approach is that preparation of this nucleophilic glycols as well as electrophilic glycols are difficult one way, and another way that it is not very stable even after the preparations over here. So then people have moved to alternative route, like for example take a simple free glycol which is very stable. They can do the Heg type of coupling by using various aryl donors like boronic acid, hydrazine, sulfonates, sodium sulfonates, carboxylic acid, etc. And unfortunately, this method is not going to provide you only one product. More than one product have been also seen. Like for example, 2,3-deoxy sugar or 2-deoxy sugars are ring opened product. Therefore, this method was also not much appreciated in the carbohydrate synthesis. So this is the time that I joined at IIT BHU. Then I started my work on the aryl glycoside synthesis. And we were working previously on aryl diacetyl salts, which is well known as a coupling partner with various type of cross coupling reactions. And they have strong ultraphilicity allows them to make cross coupling reactions, particularly in palladium-catalyzed cross coupling reactions at room temperature without any ligands and etc. So in this way, what we thought that we'll try to replace this boronic acid and iodine with aryl diacetyl salt, and expected one of the product supposed to form, because most of the reaction proceeds with a similar reaction mechanism. But we did not get this product. Instead, what we got exclusively alpha aryl glycosides. This is alpha aryl glycosides with 3 keto group over here. Then as usual, like an optimization provides a better yield by using palladium acetate 10 mole % acetonitrile water. Then having optimized condition developed, then we have established the substrate scope by varying the diacetyl salts and the sugar. So with respect to diacetyl salt, we have tried coupling with various aryl diacetyl salts bearing electron donating groups and electron withdrawing groups. And with the sugar, we have tried with glucol and galactol. In all the cases, we are able to get the desired products more than 50% to 70% yield we were able to obtain in this case. Then later we moved to L-ramanol and D-xylol and D-ribol. So 5-member, pyranose and furanose sugars, both we were able to kind of test under this condition, and all of them have given the desired products in good to excellent yields. And of course, the protecting groups play a very important role in the carbohydrate synthesis. Hence that we have tried various different protecting groups containing glycol were subjected to the coupling reaction with aryl diacetyl salts. And including the acid sensitive protecting groups, everything has found to be very stable under these conditions. So what is the reason for the alpha selectivity? Why we supposed to get only alpha selectivity exclusively? So here is the mechanism that we have proposed. Like you have a glycol and palladium in the presence of aryl diacetyl salts. Aryl diacetyl salt in the presence of palladium undergoes syn coupling. This is well known in the literature that it will undergo syn coupling. That either there is a possibility that both the group comes from down, or both can come from the top. The next step is supposed to be the syn elimination, where you are having a hydrogen beta hydride is over here, which can undergo syn elimination to provide your product, vinyl ether, which is hydrolysis to the ketone. Or in this case, the second one, when aryl groups are on the top of above the plane, then you will have no chance of getting this elimination because the hydrogen is below the plane over here, and therefore we are not able to get the beta compounds over here. But if you see the literature, most of the example that you can see that having the beta linkages. So this again make us to work further to develop a method where we can get into the beta sugar. What we thought that we are having glycol, if we can invert this glycol to the anti-glycol or the C3 inverted glycol, then the hydrogen will be in the opposite side, that which can facilitate the elimination to get the beta glycosides. So this is the like an approach that we have decided. For proving that our mechanism is quite correct, then we have synthesized C3 inverted glucol, starting from the glucose, and also C3 inverted L-ramanol, because both of them are important to propose the mechanism. So having synthesized these compounds, olefins, then we have subjected for aryl diacetyl salts. Then as expected that we have got the beta glycosides, and allowing us to understand the mechanism, what we proposed is quite correct. The yield of the reaction is not up to the mark as what we saw in the normal case. It's just because probably the strain of the molecule that we always get into small amount of ring opening as well, we could see here. So what so far I showed you that we can take the 3,5-syn glycol, you will get the alpha glycoside. If you are taking the 3,5 anti-glycol, we can get the beta glycoside. The problem that in this case the yields are not up to the mark, therefore we thought that we will try to develop another method. In this case, what we thought, we just oxidize this allyl group over here, allyl alcohol 1, by using hypervalent iodine compound, and we want to test what's supposed to be our product over here, the stereochemistry. We have got, when we tried with the aryl diacetyl salt, we got two products. One is the major product, which is a 1,4 addition product. So another one, a minor product, which is oxidative coupling product over here. In fact, this is the same that we got from the beginning itself. So therefore, we were not interested in one, but we were interested in this product because we have a chance of reduction of this aryl enones, C1 aryl enones, that which by stereoselectively to get one of the desired compounds. So therefore, we thought to develop a method to get this compound more importantly. So here is the approach. You take a glycol, oxidize to glycol enones, then make the aryl enone by oxidative coupling, then you reduce it. So aryl diacetyl salts were not successful at all. Multiple conditions we have developed and we have tried, and we were not able to achieve this oxidative coupling product as the major one. Then we moved to aryl boronic acid, which is less acidic when compared with aryl diacetyl salts. And as I say, that this also required a ligand. We have tried multiple ligands and different reaction conditions. This dictates us that reaction at 100 centigrade with 1,10 phenolphthalein, we got 90% yield of this oxidative addition product over here. So after having this methodology established, then we have studied the substrate scope with respect to boronic acid and the glycols. So galactol enone and glucol enone was initially tried with different boronic acids containing aryl donating groups and electron donating groups and electron withdrawing groups. In all the cases, we are able to get the desired products in kind of more than 50 to 80% yield in most of the cases over here. So not only the glucol and galactol, we are also tried with the arabinols and ramanols and L-ramanol and D-ramanol. All the cases, the coupling reaction, oxidative coupling reactions went through very nicely to deliver the desired products in good yields. Then the protecting group capability also was checked, compatibility also was checked with like from ester protecting group to the ether protecting groups and the acid level protecting groups. In all the cases, the desired products were obtained in good yields basically. So we have successfully completed the first step, that oxidative coupling. The second step, we have tried the reduction by using lithium aluminum hydride, sodium borohydride or palladium hydrogen carbon. The first two was not very useful in this case, but the third one, palladium hydrogen carbon, which is a syn reduction. So in this case, we are able to see the stereoselective reduction where only beta product we have observed in this case, where alpha was not observed. This is just simply that it is well known that if you take any glycol, then the nucleophilic approach is down bottom of the space over here, alpha phase used to get. Therefore, we are lucky to have direct approach to get the desired products in good yield. So here is the examples of the stereoselective reduction of vinyl enones. Then we have tried two different conditions, palladium hydrogen carbon in ethanol. In this case, that we are able to restore the benzoylic protecting group as well in the one, and the yield is a little bit less in this case. And any ester group has no problem with the palladium carbon hydrogen. Therefore, the desired compounds was able to get into good yield. But when we want to get the fully deprotected 2-deoxy-aryl C-glycosides, we have used methanol as a solvent, and a little bit longer time was stirred at room temperature. Then we are able to get the completely deprotected, biologically usable compounds. We are able to get into this. Then very recently we have also tried to establish a method with aryl iodide instead of aryl boronic acid, which is quite cheaper. And in this case, we did not use any ligand, and we have tried with palladium acetate and silver nitrate at DM of 100 centigrade. We are able to get the C1 aryl enones. Here are the substrate scope, and almost same like aryl boronic acids, and we are able to get the C1 aryl enones in good excellent yields. And as well as not only glucol, as well as galactol and other protecting groups, almost same whatever we got in the aryl boronic acid, we are able to get into the with aryl iodide as well. So what we thought that, okay, we'll try to demonstrate one of the anti-diabetic drug dapagliflozin. So in this case, we need two systems like a glycan port and another one is a glycan 1. So glycan 1 already we have known how to get into this one. A glycan port that we have started from the carboxylic acid, almost 7-8 steps, then we got this aryl iodide. After having aryl iodide and the standard reaction conditions, we are able to make the cross coupling reaction to get this compound. And by subjecting to the reduction by using palladium carbon hydrogen methanol, we are able to get the desired compound in a 91% yield. This is the 2-deoxy derivatives of dapagliflozin. Then we moved to the synthesis of dapagliflozin itself, that we have just reduced the ketone by Lucci reduction, then where the double bond was restored again. Then followed by the benzylation, then hydroboration. It provides this dapagliflozin analogs, protected analog, then which was subjected to palladium carbon hydrogen and methanol, and we are able to get the dapagliflozin in the 90% yield in the last step over here. Yeah, with this, very recently we have also like tried to make 1,4 addition, stereo-selective 1,4 addition with like palladium catalyzed one with aryl boronic acid in the glycol. And here is the substrate scope, and this work is in the under revision. With this, I would like to conclude my talk that I have like explored the synthesis of 2-deoxyryl glycosides from glycols. Different reaction conditions provides different products with this, complete stereo-selectivity. And this is the my research group. It was not in Pondicherry University, and previously I was in IITgn. So this was the group from IITgn, and the students who were worked on this one, Adeshkumar Singh, and who is a postdoc at University of Bristol. Then rest of the students are somewhere working in like colleges and universities. And I would like to thank funding agency IITgn for the startup. Then Max Planck Institute also I received some fund, SCRB and DST for all. Okay, the last slide is the Pondicherry is a very nice place that most of you must have explored. If someone is not explored, then it is nice that you can visit Auroville, Aravindo Ashram, Paradise Beach, Rock Beach. There are so many places that you can get into Pondicherry. And Pondicherry University also very well known for the chemistry, and I request if any students want to pursue their master degree or maybe PhD, that you are welcome. With this, I would like to thank the organizer for this opportunity and also for the Arunachalam for his introduction. Thank you very much.
Okay, thank you, sir, for your energetic presentation. Is there any questions from the audience? Yeah.
You are talking about aryl diacetyl salt synthesis or other than aryl diacetyl salts you are asking? Actually, aryl boronic acid and aryl iodides are very commonly used for the synthesis of C glycosides. Actually, we are just replacing them with aryl diacetyl salts because it is cheaper and it's more reactive, basically. Thank you.
Okay, if no more questions, then yes, yes.
Yeah.
Very nice one.
Thank you so much.
You joined in IIT.
BHU,
first?
yeah.
So that is the time you came from Shiburger School.
Yes.
Okay, you were the one conducting the
Yeah,
RAC?
I was.
Okay,
Yeah.
RAC, right?
Yes.
RSCC or RS?
Yeah. This is the recent advances in carbohydrate research. Yes.
Very good, excellent.
You
So
are
now
there.
you are in Pondicherry.
Yeah, of course. I very recently I just moved. So 1 year ago I moved.
Oh, a year ago. Okay, very good. A good piece of work. Yeah. So, did you try out anything to do with these compounds?
Yeah,
Because
actually,
obviously there is a purpose why you develop the methodology and
Yes,
then
sir.
you are able
Basically
to get the assortment of compounds.
yeah. Basically,
Did
we
you try
are
out
trying to develop this anti-diabetics
No,
compound.
I'm not asking about
Okay.
anything seriously biology which you will not be able to manage,
Yes, sir.
but something very finding out what could be the acceptance of these molecules in a biological front. How is the solubility of these molecules?
So this molecule is highly soluble in
Are
almost
you
all the.
removing all the protecting group
Yes,
at the
sir.
end
So
of it?
methanol or even like some protic solvents is highly soluble.
It is soluble.
Yes, sir. But not in water. But in methanol it's soluble.
Is it okay? Very interesting. So there is no role of any carbocation, oxocarbenium ion here.
No, sir.
There is
It's
not.
not involved in this one. Simple cross coupling reactions followed by the reduction. Yes, sir.
Okay. So how is the selectivity between galactose and
Yes, sir.
glucose?
Basically, then in some cases we found the glucol is more selective when compared with the galactol. In some
In
cases,
terms of the anomeric selectivity.
anomeric selectivity. Sir. In both the cases, we did not have any problem because the third hydroxyl group really determined the stereo selectivity. Therefore, we did not have any issues. In both the cases, we got almost the stereo selectivity is very high. Very high means we did not
what I'm
observe
asking
another isomer at all.
What I'm asking is that if you are getting one selectivity for glucose, do you get an alternate selectivity for galactose?
No, sir. Both are same because the hydroxyl group over here. Okay, I'm sorry, they corrected.
Okay.
The third hydroxyl group is the same configuration, glucol and galactol, both the cases. So
But
therefore,
it is same only if in isolation.
Yeah.
But if you compare with the C4, they are different.
yeah,
right?
they are different. But that is not involved in this stereo selectivity over there. Therefore, we did not find much difference in that.
It will be worthwhile to see that because you have shown the half chair conformation.
Yes, sir.
In a half chair conformation, galacto configuration having the axial hydroxyl
Yeah,
group
true.
will be blocking one of the phase. Whereas with the same chair conformation having the C4 hydroxyl group equatorial, then
will have.
it is not on the way of the entry of your nucleophile or electrophile. So I would normally would have anticipated that there is a stereoselectivity switching
Okay.
between the gluco and galacto configuration.
Okay, sir. We did not observe that one, but we observe the rate of the reaction, difference in the rate of the reaction as well as the yield of the reactions. For example, in some cases, glucol is given much better yield, where in some cases, galactol is given much better yield.
Very, very interesting work. Good piece of work.
Yeah,
So there is no issue about the hydrolysis here.
No, sir. We
No
didn't
issue about hydrolysis.
No, sir.
There is no hydrolysis.
No.
Except that it can ring open.
Yes, sir. There is a ring opening is the major trouble with this one. Particularly under acidic condition, it opens the ring.
It opens. It's a pyron, right?
Yes, sir. Pyron.
It's not
only, sir.
pyranose.
Yeah, no, sir.
It's a good thing to say that the C glycoside,
Yes,
but otherwise it's only a pyron.
Yes.
You go.
Okay, so please join hands to thank Dr. Jayakumar. So a small gift from the organizers. Sir, please. Okay, the second section is given by Dr. Selvakumar. So, sir is the founder CEO of DSK InnoSciences, Hosur. Sir has completed his MSc from St. Joseph College, Trichy, and PhD from IISc, Bangalore. So, he has also done 2 postdocs in North America. He has a very rich experience as a synthetic organic chemist at Dr. Reddy's laboratory, Hyderabad. And DSK InnoScience is founded by him almost about 11 years back. So over to you, sir.
Not moving. அதான் பண்றேன். Yeah. Good morning, everyone. I felt I'm slightly an odd man out because most of them have come and they shared their exciting chemistry to you. And I'm going to share something very different for all of you. But for most students, what I assure you, that you will find the presentation and you will find the contents very useful for the future. In that way, I will be confident that in the end of my presentation, you will feel that you are taking home some message. So, first of all, I should tell you that I am very privileged to give my speech today about the company that we established about 10 years back. And but before that, I should tell you that I have some long history with Madurai Kamaraj. The first time I visited here was, I think, way back, maybe 20 years back, for campus recruitment. So I used to work at Dr. Reddy's lab, and I was the first person to visit MKU for hiring people. I know people whom we hired, they are all doing very good now. Subsequently, we came couple of years, and then maybe after that I left the company and I moved on. So with that brief memory, I will now take you to the presentation. So what we are going to do, we are going to talk about the company DSK, which is established by me about 10 years back. And what I'm trying to concentrate here is a journey of a chemist trying to establish a chemistry company. So I felt that was a challenge. It's if you see, historically, people in Tamil Nadu or India, if you see, the business community, they will be doing the business. The same thing is true in this kind of business also, business people only will do. But in this case, as a chemist, I was trying to do that. So it was not easy, initial days was very, learning for me, but eventually we succeeded and now we are doing well. So today I am going to tell how we established the company from a chemist perspective. So the slide which I am showing here is one of the labs which is very modern, and we have about 50 fumutes of similar kind in our group. So you have to help me, it's not moving. Oh, sorry. More, yes. So, yeah, I think now I will speed up. So before starting the company, I felt that, we need to have a vision. What is our vision? Our vision was to establish a most reliable CRO. So the company, by saying CRO, we are a contract research company. We basically, bring research from overseas, and then we, do work on contract. So we felt that our objective must be to establish the most reliable company. That means when our clients give the work to us, they should believe that they will get the product. So that is how, with that vision, we started. I want to tell you that, in order to realize our vision, I felt these 6 are very, very important. So it will be kind of a different perspective for you. See, we feel, we are driven by innovation, and we are very different in execution. How? You will see in the following slides. And we are focused on our customers' satisfaction. It's a little bit on the business side. But what is more important, even though business, chemistry is important, it is also important that you have to give respect for an individual, and we care for the environment, because our business is chemistry, we pollute. So we need to have the care for the environment built in our business, and we are an equal opportunity employer. I think some of you, the students particularly, may not know what is the meaning of that. The meaning of the equal opportunity employer is basically to say that in a country of diverse nature like us, with the people from different languages, different castes, religions, we felt that we should not have any bias. We will take people only on merit. That means the equal opportunity employer. So with this, as our practices, to realize the goal, we went on to our business. So in our business, what we are very good at. So when we are talking to people all over the world, what we say, we are different in these things actually. That means we are very strong at providing solution for even difficult chemistry problem. So I would say today, if you ask me, what made you, what was the primary reason that you have succeeded in establishing the company, I would give the number 1 as my, the selling point. So we were able to demonstrate that we were able to solve some of the most difficult problems with our clients. And I think one slide later I am telling you. So this is basically a slide about me. Already he introduced, so I am skipping all these things. But what I wanted to tell you is like, we are, sorry for that. I am a chemist turned entrepreneur. This point I already told you. So what we have done, see, we are now, I'm telling that we are 10-year-old company. What we have done during these 10 years, we completed, over 1,050 projects. So if you look at the history of 10 years, 1,050 projects, which are ranging from 1 milligram to 1000 kilo. So our operating range is from 1 milligram to 1000 kilo. So up to a ton we deliver, and 1050 project is not a small number. So we were able to do all of them successfully, and we have several programs with the big clients in all the areas of chemistry. And this is one of the slides I'm just showing you. Hereafter the slide will be more like pictures and just to watch and less to read actually. So this is one of the facility, basically our boardroom. It captures all our team leaders. So the team leaders are all highly experienced, working in some big companies, as I have mentioned here actually. And this slide again, I'm telling you that it will give you a visual capture that this is one of our chemistry labs. And what is very important is, for doing a decent chemistry, we have all equipments. I think as a chemistry services company, we are completely well equipped and equal to top-notch facility in the world. I think here I should mention that, as you all know, when you are trying to do chemistry to the world standard, it is very important to have the analytical facility quite strong. And we are a completely equipped company in analytical. If you see the list, it starts with the 400 MHz NMR, several LCMS, several HPLCs, some of them you can see in the pictures, and we have PRIP HPLCs, which is very, very advanced, very expensive, and several jacketed reactors, photochemical reactors, polarimeter, lyophilizers. So I think, in order to do chemistry from any scale, from a milligram to ton, everything what you need, more than required, top-notch, the analytical equipment, we were able to establish it in the last 4 or 5 years actually. And yeah, this is just to give you a glimpse about some of the pictures, and I'm not going to go into those details for the sake of time. And this is again, I'm going to tell you, that you please watch the picture. These are some of the facilities and the labs what we have. Yeah, I think this picture is very important that you all must see, because working in an R&D facility, you may not know what is the beauty of the scale-up manufacturing. So if you see the picture on the left-hand side, this is one small portion of our scale-up laboratory, and what you see here is all a series of autoclaves to do very hazardous and high temperature reactions, all state of the art. I think this is again, as I was telling I will run through some pictures just to give you some exposure. This is a picture of our scale-up lab. Several reactors are placed, but very modern, very presentable, very auditable facility actually. And yeah, I think this is another lab, but here I want to stop you and then show you some of the areas what we are working. I think I see a lot of interest in carbohydrate. The previous speaker was speaking about carbohydrate, and he may be surprised if I tell, we may be the only company who have done extremely well in carbohydrates from the business point of view. So all our clients are overseas. Some of the professors whom you spoke also, I think we have worked with them. We know him very closely. You said Seeberger, right? So with the Seeberger's group also we are working, and we have done extremely well in the area of carbohydrates. I can tell you one third of the company business from the starting time is from carbohydrate. Here I must mention to you something very interesting. I remember, one of our carbohydrate customers visited when I was having 2 or 3 people in the company, employees. I got a call, and I'm just telling interestingly what actually happened. It was maybe 10 years back. I didn't have a business, and I got a call that, are you in the company? I said, yes. So we have a client is from overseas, from Europe, and they want to visit. I am always available. So the person, walked into the company. The number of employees were only 3, and he was wearing shorts, not even a pant. And then, I gave him a presentation on that particular day evening, and he was quite impressed with, our ability to do the chemistry. But what he asked is, you are, you don't seem to have carbohydrates as an experience, I'm telling you. Then I told, so it may be true, definitely I have not done carbohydrate. But what I feel is carbohydrate is also a chemical molecule. So let me see, when we have achieved so much of chemistry in other areas, why I cannot do. That is the way I started, and then I took the first project for the company from carbohydrate. Today we must have completed about 350 projects in carbohydrate. Even today, anytime you walk, not less than 10 projects in different scale, 100 kilo or 10 gram, it's all going on in the company. So we are extremely successful. Most importantly, in carbohydrate, we delivered 100%. We have a delivery record of 98% overall, but in carbohydrate, I don't know, to my surprise, we are 100%. So this specifically for my previous speaker, I told, but it gives you a glimpse, like what are the different areas we work. We are working on new chemical entities, chemical intermediates, monomers, polymers. I'm not a polymer chemist, but we have a strong program. We are looking for scientists who are chemists who have the PhD in polymer. We are trying to build polymer department now, but we already have a team and we are delivering. And we have team for peptides, metabolites, reference standard, impurity. These are all pretty much very specific to pharmaceutical actually. So this is one slide about library synthesis, but I'm not going to go here, but I'm going to tell you this machine is very, very good. I'm a hardcore synthetic chemist. I know, you give me anything, I will be able to purify in a column. That's how we were trained. But unfortunately, the current trend is everything machine. But to my surprise, yes, this machine is super. After this machine was purchased, very, very expensive, the chemists are very cool because they can load any sample inside and then they get purity more than 95% just in overnight. So it's a fantastic machine, and this is quite expensive, and after this came, our chemists are really having good time. And this slide I'm going to skip for the sake of time. And this is also I'm going to skip, but it's an important area. It's called impurities in the area of pharmaceutical. For the sake of the students, I'm telling, the impurities is an area in the pharma research. It will be formed in small scale, and we are supposed to make them for the research purposes actually. So that's very challenging because they don't have any literature, and we have really, really great record in this particular area. For the last 5 years, continuously one lab is working on impurities for a large client with great success, and that is what I'm capturing here. I'm skipping it for the sake of time. And till now I have given a run about the chemistry business, but hereafter I will run through in different side. Basically, we are very conscious about the quality, and some of you, mostly from students, you will not know what does it mean, but basically the clients who are working with us expect your company to be maintaining a minimum quality. So we are a quality management completely compliant company with recently an award given by the ISO 9001, and this I am skipping it for the sake of time. So this is about recruitment. This is very important, and what we do, this is one of the strengths of our company. Though young, what we did was we handpicked the people. What I did, I always like this word called the road less travel. That means I don't go and hire people from top-notch places. For example, VIT. I know some professors are there, but I don't go there. Rather, I send the people to universities, government colleges, and then we conduct our exam. So this is one of the exams people are writing. Today we have a campus recruitment pitch at MKU, and some of you may be writing the exam. So we are very successful in that. This model I brought from Dr. Reddy's. As I already told you, the first journey for me to MKU was for the same purpose. So what we do in this year, we are going to hire about 30-40 chemists actually. So we are going to expand. We are 100 currently, but we want to become about 150 in maybe about 4 or 5 months time after you all complete your masters. So our campus recruitment is going on all over the state. I'm giving more importance to Tamil Nadu because we are located there. And we completed already about half a dozen campuses actually. We had Professor Arunachalam is from Gandhinagar. Last week we had our campus pitch there. Today here. Last week we also had from INR Janakumar College. So what we do there, we conduct the exam at campus. 100 questions we give, most of the questions written by me, and then we select and then take the people to campus. We give them an exposure to visit a campus and take part in the interview. At the end, we give them offer letter. Usually about 150 people, usually if they write about 8 people, 10 people are given offer letter. I know when we are selecting the best people, some of them will have opportunities to go for PhD, but only 2 or 3 will join. So that is our record, but because of this, I think this is the biggest differentiator. We get some of the best talents in the country. And this I am skipping it for the sake of time. So, after having spoken a little bit about business chemistry, I am also giving you a different perspective about the company, business, life, society. What I feel when you are growing, when you are trying to do something big, it is very important to integrate yourself with the society. You should be part of the society. So if you visit our company, what we do so much social activities which I don't have place to accommodate. Just a hand-picked 4 I have accommodated in one slide. For example, if you see, the first COVID vaccine in the district was given by DSK. It was a very bad time those days, and the whole country was dying. People are reporting mass deaths actually, and the government announced the release of vaccine. The first day, our company administered the COVID dose of about 200, about 80 of them for the employees, 120 for the public. The company became super popular. They felt that this company is part of us. So I think we were very happy about that, and we invite the village people for many occasions to our company. So we are located in a village, but we don't think we are a company. We are part of the society. So we call them for every function. We make them to eat along with us, and we provide toys and books to neighboring schools. This is one of the government schools we donated, all of them from our company, and offering meals to primary health center. So this is our neighboring primary health center, where whenever there is a camp, every 3 months there is a camp for a specific cause. This is one of the camps conducted for handicapped people. More than 500 people visited with handicapped problems, and our company provided them all meals. So I think we take pride in being part of our society, and our neighborhood people never thought that this company is coming from somewhere and going back with the money. We felt that we made them to feel that we are part of them. And that is another slide about women. See, I'm trying to give you a different perspective. I always think very differently. It's not only chemistry, it is not only science, it's not only money. Definitely, money is not the only ultimate goal. We feel that we take pride more in such activities than making money. So this is the slide which is very passionate to me. We give a lot of importance to women empowerment. How? You see, these are very people from downtrodden families from our neighborhood, and some of them are COVID widows, some of them are abandoned by the husbands, but each of them are having minimum 2 children. So they are struggling for livelihood. So what we do when we have a vacancy, we handpick the needy people, and then we employ them for all hospitality. So it is a all women crew in hospitality. That means you go and have food in your canteen. It is made by all women. All hospitality by women. It's just a small number. We have more than a dozen women working in this particular segment. Now, it's not only that, we encourage women to take managerial positions. I know today a girl from Periyar University, she got selected from campus interview 3 years back. Today, she is leading a major program for a top client. She is sitting in the telecom and talking to the client in the US. So we encourage a girl with a 3-year experience taking leadership positions. That is the way we nurture the woman. But if you go to big companies, it is not the case. 10 years, you will be master student, you will be working on bench. You don't know really what is happening. But in our case, that is the beauty actually. We feel we should give opportunities to all the people at a very early stage. That is one of our specialties. So, we are very proud to say that the entire company, being a chemistry company, many people said, women, often don't suit for chemistry. I said, who said that? I will make women to do the scale up, manufacturing. We have girls working in manufacturing successfully. So we are 1/3 of the whole company is women. That we take pride in that. And where are we today? I think, we are looking for some, aspiring for some prolific growth. And we have the record of about 98% projects delivered. And we are located in a very beautiful geography. And we have adequate space for expansion. And we have a global office in UK. It has been more than a year and a half. We have an office in UK. And that shows our commitment to further growth. And we are very interested in about social responsibility activities. And here is a slide where, our company being a young company, we have never solicited for an award, but we were very surprised. Recently, we got a call from Government of India that they picked us, and then they said, you have been selected, as a first prize for Export Excellence Award. We were very surprised. Oh, we didn't know. We thought, okay, we are quiet and we are working. And the Minister for Commerce and Industry, she gave away the award to Sendil, who is part of our audience today. So I think after we received the award, we understood, okay, in our pursuit to establish a good and aspiring company, I think we made the first step forward well. So this, with great happiness, I am sharing with you. And that's, I think almost I am done, maybe some 3 more slides just to make a picture. Yeah? 2 minutes, 1 minute, 1 minute. So this is about one of the pictures. We are surrounded by 3 lakes, and I titled it as Little England. And so several sustainability measures what we are very interested in. I am skipping it for the sake of time. And yeah, I think almost the last but one slide. So it's about employee welfare. So after doing all these things, so what are we doing for employees? So if you see, we have a canteen which is supplying all the 3 meals, and we do organic farming. So this is very special, very different. All of you must watch. So what I felt, say, I am from a farming background. I am a farmer by training actually. So what we did, it's a 5-acre piece of land. 3 acres we have dedicated for the company. 2 acres we have dedicated for the farm. So we have farmer on the payroll. He creates, this is a paddy which is grown in our farm. So we create from paddy to all vegetables and fruits right at our campus, and that is supplied to our canteen. So our canteen is predominantly food prepared by us in an organic way. So that is one of the very, I thought as a farmer, this is something that I should contribute to my company. This is what we have done. And for employees, we provide all insurance, and we have taken care of employees during COVID-19 to the best. Because I am coming from a medicinal chemistry background, I ensured that all the employees live very healthy. If you ask me, the best contribution to my employees is this one actually. And we provide transport for the employees, and our employees have fantastic career. Whoever joins our company, whenever they leave, they go with 5, 4, 6 jobs actually. I am very happy about that. So yeah, with that, this is just a picture which is telling about so much of women working. They are all chemists actually. And this is our UK office address. And this is my message. For the sake of time, I am skipping it. And yeah, I think I have come to the last slide. So this is our brand new office we recently occupied. And I want to tell you that our best is yet to come. I always feel as a scientist, you don't think, you are always having a goal, but once you achieve, our expectation about the best has moved up several fold actually. So I always feel, we have to keep growing, keep expanding. I feel still our best is yet to come. And thank you organizers for giving me a few minutes more. And I am completing my lecture. Please go ahead.
Thank you, sir. Thank you for your nice presentation. So your presentation, I think your presentation have inculcated entrepreneurship among younger minds. So thank you so much. Is there any? Yeah, please. Maybe you can have.
You loud. You tell. I can understand.
The new instrument. That one purification only the waste material, or you have to work with only the chemicals or organic or inorganic solvents or compounds.
One more time, you tell me.
That instrument you are showing, that is the doing. It is enhance that column procedure. That instrument is purify that waste materials. You are using that one, or in the only compounds.
Yeah, I will explain you. So what we do, that particular machine is very, very advanced. Most companies don't have that. What we do, this particular instrument, it purifies the compound of your interest, whether it is present in 5% or 3% or 60%, it picks that compound and purifies and gives you. So
waste material also, sir.
Waste.
Yes, sir.
Waste material.
Yeah.
Whatever the material you want, you have to fix the mass. My requirement is 388. You fix 388, it will pick 388 all your compounds and give you. So usually what we do, we pick our mass and then we purify. Yes.
Sir,
it will
we are
purify
the
anything.
chemist. We are using a lot of solvents or a lot of chemicals. We are wasting.
Yeah.
We are some labs are storing that one. How many you are doing that work? You have to contact with the universities or any other industries. They have to give you the solvents or because we are a lot of wasting just like acetone. We are every time cleaning the glasswares. We have to throw directly sinks. In India, nobody store that all things. I feel like this one we are affected the environment.
Okay.
Then you have to like these things. You have to collect that all waste solvent and you have to do purifications.
No, not purifications, but what we do the waste management very effectively. That slide I skipped it fast. Basically,
sir,
what we do, all the solvents we generate, we recycle.
Time you can have the discussion during the tea break. Maybe you can interact with him. Thank you, sir.
We will
Thank you.
talk. Yeah.
Sir, a small clip from the organizers, please. Okay, it's a time to introduce the next speaker. So the next speaker is Professor N. Jayaraman from IISc Bangalore. So sir is the distinguished professor known for carbohydrate chemistry. So sir is the associate editor in Glycoconjugate Journal and Journal of Chemical Sciences. Sir is also a fellow of Indian Academy of Sciences, and sir is also a recipient of Sandhi Bhatnagar Award. And currently sir is holding a position of Secretary General, Chemical Research Society of India. Since we are running out of time, with this brief introduction, I invite sir to present his talk. Sir, welcome you, sir. Yeah, sir, second here 30 minutes. 30 minutes, sir.
Slide changer. Very good. So, very good morning to all of you. Namaskar. So, it is very pleasant to see a large number of very young students and a large number of budding scientists coming here and giving talks. I guess that Dr. Shivakumar probably started in a very innocuous way of thinking that he will just call few people and then have organized one-day meeting or something of that nature. But to see that today he has a couple of hundred people here, or more than 200 people sitting here, as well as large number of young budding scientists and lots of people from industries, he eventually turned it to become almost like in a conference as good as an international conference. In fact, you are having a speaker from the international level also. So it really speaks very well. So excellent. Well done, Dr. Shivakumar. So more so because he is conducting under the umbrella of Chemical Research Society of India. So it speaks of very well of his commitment to spread across the chemistry and chemical sciences to the younger audience here. So well done. Thanks for the invitation to come over here. And even in a very short period that I am here, I am already noticing a very high quality of science that is being discussed here. And you see, not be left out is even more important component about how the chemistry is helping in a very big way in industrial sector. So Dr. Selvakumar has given a very nice presentation on his company's growth right from scratch. So I'm sure that it is giving a feeling of, we are all chemists. So our language is chemistry. You know, language is so much a chemistry that I landed here early morning 3:45 today. 3 chemists who are making the presentation yesterday, I suspect that they are not here. So they were sitting together and they are talking morning 3:45. Man, so much chemistry here. And then I'm coming out of my room and going to the temple morning 4:30. I'm seeing them, they are walking on the road. Chemistry is still happening. So all of us are only bound by chemistry. Our language is only chemistry. So chemistry is what has brought us together to this place. So in the next 25 minutes or so, what I will do is that I will present a very brief story about a research project which is going on for last close to 20 years, maybe 15, 16, 17 years. So this is in the area of carbohydrate chemistry. So this is more to do with an observation about how nature does and why nature is unable to do. We as chemists, can we do what nature is unable to do? And when we are able to do it, what answer we can give it to the nature? Hey man, I can do something better than nature. Or nature is going to come back and tell, look, you cannot be better than me. Right? So after 20 or 25 minutes of presentation, I'm going to give you an answer, which is, yes, we can do something that nature does not do. But nature is going to have the final law, no, you can't do better than me. Okay? So this is about conformationally constrained cyclic oligosaccharides. So where does the cyclic oligosaccharide comes in and why did I choose this topic to present today? Cyclic oligosaccharides and this department is not too far off. Lot of work has gone in this department of chemistry, Madurai Kamaraj University, on cyclic oligosaccharides, particularly in the series called cyclodextrins. So Professor Pichumani and others have worked consistently making use of this cyclic oligosaccharide for its very important molecular level property. Cyclodextrins, one which is a cyclic oligosaccharide, one which is plentifully available. Today nobody is going to make chemically synthesize this cyclic oligosaccharide because you get 1 kg of the so-called cyclodextrin maybe for $5, $10. So where does it come from? From natural sources. If you look at it, we all know that starch is one of the major biomolecule. Sun energy comes, carbon dioxide is taken up, water is taken up, formaldehyde forms, and then a series of Formose reaction happens. C1 carbon of carbon dioxide has now become formaldehyde. Go to C2, go to C3, C4, C5, C6. C6 is glucose. Now multiple number of glucose come together and in the process large amount of energy is trapped. And then when we have our food in the form of food is essentially plant derived foods or starch, the energy is liberated. So we basically get the sunlight energy in the form of starch. So when we look at the starch, we don't have to go back how the starch synthesis per se happening because it's a biochemical story. I will not go into it. So take that this is the starch and what all we know about the starch is that starch is made up of 2 very major polymer. One is amylose, the other one is amylopectin. Amylose is linear, amylopectin is branched. That's all we know about constituent of starch. Taking particularly about the amylose is hydrolytically less, it can undergo hydrolysis. Yes, it does undergo hydrolysis. So when you take a large piece like this one, there are many enzymes that can act on this one. For example, isoamylase cuts off the hydrolysis at 1,6 linkage. Amylases, again hydrolysis to different pieces. This is more a case of a linear as well as branched one. So something acting on the branching, something is acting on the hydrolysis of the linear chain. And one particular degradation of the amylose is to cut into the small pieces, and the small pieces with the help of this so-called cyclodextrin glycosyl transferase, which is a transglycosylation enzyme. That enzyme basically reacts one end with the other end to form this kind of cyclic oligosaccharide, and this is the cyclic oligosaccharide. A large amount of work has happened right in this department. So if we take starch, so what we know about starch is that we have so-called alpha glycosidic linkage, and with the help of this amylase, which can be exo or endo. I have given the case of endo. Endo means it hydrolyzes in the middle. Exo means it hydrolyzes at the termini. So two pieces have come out, the so-called maltodextrin, and with the help of cyclodextrin, large number of such cyclic oligosaccharides form, and the cyclic oligosaccharides are very major economic driving, because large number of drug molecules, large number of molecules which are not normally soluble in organics, soluble in aqueous solutions, in the presence of such a kind of cyclic oligosaccharide, they will be soluble in aqueous solution. So as a result, this is a compound which probably economically very, very important, whether it is pharmaceutical sector or all the way around, including a paint sector, for example. Most emulsion polymerization happens with this one. Now you see that the pieces comes out, and the maltodextrin comes out, and the maltodextrin with the help of this cyclodextrin glycosyl transferase enzyme like this, and many of them are very, very well characterized, which is the site at which it is the linear oligomer binds and how the cyclization happens. So all these are very well worked out in the case of bioengineering and the synthesis of cyclodextrin per se. But what is interesting to notice is that this is what is very important. What nature gives is that you can get a cyclodextrin with the 6 glucose molecules, 7 glucose molecules, 8 glucose molecules by and large. But then today, because of the bioengineering possibility, one can get higher oligosaccharides, cyclic oligosaccharides, CD9, 10, 11, 12, 13, 14. But as you see that, what a very nice cyclic cavity here, as the number of glucose residues are increasing, it is becoming more and more distorted. So this much is known as of now. Lots of studies are also happening. Now the query comes, why the nature produces only with 6 glucose as a limiting case? 6, 7, 8, nature produces no particular difficulty. So this is the reason. If you want to go from 6 to 5 to 4 to 3 or 2 glucose units to cyclize, what happens is that the molecular modeling structure or energy minimization structure in the group of Frieder-Lichtanthaler. Frieder-Lichtanthaler, incidentally, is the student of Emil Fischer. So we are talking about the era of 60s, 70s, 80s, or even before that. And towards his end of his career, Frieder-Lichtanthaler, his query is that why we are unable to make small cyclic oligosaccharides that are lesser than 5 glucose or 6 glucose units. So you see that the cavity is nicely forming as we increase the number of glucose to 6, 5, 4, 3, 3, there is hardly any cavity. But what is even more important is to notice that the very stable 4C1, 4 chair 1 conformation is all the way getting distorted to something like an envelope conformation in order for that molecule to cyclize and give smaller cyclic oligosaccharide. And he told it is impossible because you cannot create a conformation from a very stable 4C1 conformation to an NNO conformation you cannot create, and as a result you cannot get any cyclodextrin lesser than 6 glucose units in the macrocycle. So this was the challenge. Nature does not produce, or at least we do not know nature produces. Nature has a reason for it. The reason is that it has to go to a very unfavorable conformation, 4C1, 4C1 to E1, E1 to 4C1, and finally you see that 6, 5, 4, 3. When it becomes 3 glucose units, this is simply a molecular energy minimization theoretical study. Coming to E1 conformation, it is not possible. Well, I think at this point in time, I should tell that To synthesize sucrose, which is a tabletop sugar, as of now we have only one or two instances in the entire literature. Back in 1967-68, the first organic synthesis of the sucrose itself came out. Now this is back in 1985. Can we synthesize chemically cyclodextrins? Yes, indeed. Tamaya Ogawa, who is an icon in the area of carbohydrate chemistry, he prepared this linear oligosaccharide of 6 sugars having a glycosyl donor, glycosyl acceptor, and cyclized it through a process called glycosylation in 21 steps. In 21 steps and overall yield of 0.3%. But at least now this is the outstanding example that we can chemically synthesize a cyclodextrin, which nature does without any difficulty. And then Kochetkov and this group came up with a different idea of how the glycosylation can happen. Instead of having linear oligomer, have a monomer of a disaccharide and allow it to cyclize. Here I am not going to take you into the details. It's an outstanding chemistry of cyanoethylidene and a trityl perchlorate, which can be removed with a trityl perchlorate acidic condition. Trityl group comes out and this comes and cyclizes to give this compound. So even in these cases, it is very clear you cannot make a cyclic oligosaccharide lesser than 6 glucose units. So in this process, long time ago, so this was a couple of my former co-workers are here, Nityanandan is here, Jayaraman is here, they know this work. Dr. Shomak Pal, he undertook this work and showed that if we remove some of the hydroxy group, then it becomes conformationally flexible, then we may be able to make smaller cyclic oligosaccharide. But unexpectedly or expectedly, nature was correct. We are unable to make anything lesser than. However, by saying that one of the very first instance of a cyclic oligosaccharide with 4 sugar units was prepared back in 2007-2008. Then it was not, see, very soon we realized that in this particular case, when we remove this hydroxy group here, then this glycosidic bond becomes extraordinarily weak, hydrolytically very weak. So the stability is not there. So we knew that it gives us some flexibility, but then not very hydrolytically very stable. I'm going to take a very long story short for reason of the time. So start from glucose and take it through steps. Now retain the hydroxy group as it is, but introduce yes, methylene group. In a normal glucose, you will see the C4 will carry the hydroxy group directly, but here it is intercepted with a methylene group. So it is hydroxymethyl glucose if you want to call. So it goes through steps, a fairly standard procedure. So I am not taking step after step here. So having this particular type of hydroxymethyl extended glucose unit, so the idea was that will we be able to create the corresponding cyclic oligosaccharide. So now you see that I have the linear disaccharide here having the glycosidic bond extended one. In a normal maltose, what you will have is that this oxygen will be directly attached to this carbon, but in this case, it is extended with one methylene. And we have here, so this is the so-called glycosyl donor, and this is glycosyl acceptor, one of the outstanding glycosyl donor, thioglycoside. If we take 10 glycosylation reactions in literature, at least 5 to 6 glycosylation will be conducted with a thioglycoside. Outstanding one. So when we do that, now we see that for the first time, in fact, we can create a cyclic oligosaccharide with only 2 glucose unit, 3 glucose unit, 4 glucose unit, 5 glucose unit. So at least we have achieved what normally not nature would do. But by saying that, nature will not carry this extra methylene group. We have the extra methylene group and created the favorable 4C1 conformation. So when we retained the 4C1 conformation, we are able to get the small cyclic oligosaccharide, but the unnaturalness comes because we have incorporated extra methylene group. So now you take it, it's still okay, we have shown it, however, it's still hydroxymethyl glucose, not a simple glucose. Now cyclic trisaccharide, when you have a cyclic trisaccharide and then allow it the thioglycoside reacting with the glycosyl acceptor hydroxy group, one of the very first instance of a macrocycle with only 3 glucose unit. So very, very interesting, and we have the crystal structure of that. The crystal, I am skipping off some of the characterization, but I can still tell about the characterization here. When you see the symmetry of the molecule, and you see the, I don't know, okay, you see that you are having 3 proton here. You have every one of them are 3 protons, and you can see in the corresponding 13 CNM are Each one of them corresponds to 3 carbons. And the whole 13 CNMR will be completely chaos if it is a linear molecule. So cyclic molecule comes up with an outstanding symmetry, and that symmetry allows, for example, this carbon, 3 carbon. Every one of them is 3 carbon. Of course, these stars correspond to the carbons, different carbons. So now you see that one can go on and on to make one with a 2, one with a 3, one with a 4, one with a 5, etc. So this is something that we were able to show that yes, indeed, it is possible. What nature is unable to do it, we are able to do it. Are we better than nature? Let us look at the answer for that. So these are all the crystal structure. I should skip off some of these things. Once you have a crystal structure, then another world opens up, because you see the molecule, you see the kind of symmetry, you see the dimensions of the molecule. So what is the upper rim, for example, what is the lower rim, what is the height, everything that one can see in this one. and also this packing diagram, so it gives a lot of information. So let us move on further, because I think I will be exhausting my time in another 5 minutes. So let's take it. Noticing this one of my students' thesis went to Yamada. He noticed, hey, Jay is doing something like this. I am somebody who is working on this for such a long time. How come I am missing this? By the way, he did not tell me, this is my inference. Sure enough, he came up with the idea. J is missing one element. That element is that if I am having the unfavorable envelope conformation right in the beginning itself, and when I try to cyclize it, I should be able to get it. Sure enough, he did it, and he got it in Science. This is 2019. This is that unfavorable conformation. Look at this unfavorable conformation, envelope conformation, that he did by cyclizing this 2 oxygens, 2 hydroxy group, not with a benzyl group, but with a benzyl group that carries the methyl group at both the end, so that intramolecularly the benzyl is clipping 2 of the hydroxy group. So normally benzyl group you put only for 1 hydroxy group, but intramolecularly he put this one. By the time it becomes bicyclic compound, then it undertakes this kind of conformation, and he got it beautifully, 1 with the 3 and 1 with the 4. So he has shown what Lishtanthaler told. It is impossible to get. This guy told, yes, it is possible if I start with an envelope conformation, which obviously we were thinking that we may be able to manage. We could not manage. We had to put hydroxymethyl. Anyway, beautiful crystal structures there. Then what we thought that we should be still able to manage it. How can we manage it? Remove some of the hydroxyl group. If you remove some of the hydroxyl group, then we may be able to introduce conformational flexibility. So we removed the hydroxy group. So I'm not going to take you through the synthesis. One can synthesize from glucose, and then all the way with 1 donor here, 1 acceptor here. There is no hydroxy group here. When we do the glycosylation with this one, the idea is that when we have the donor here and acceptor here and allow the cyclization to happen, will we get the cyclic dimer, cyclic trimer, cyclic tetramer? So this was kind of an anxiety and anticipation. The student did this work, and he came back and told, sir, it is forming, but I am not convinced that this is the structure, because his contention is that if it is a cyclic molecule with the alpha anomeric configuration, this should not be at 103. So Biswas Sarkar told that it cannot be like this. So I also left it. Okay, leave it to happen by itself. We know that looking at the symmetry of the molecule, that it is one single isomer. However, it is not alpha anomer. Then a crystal structure came in. From the crystal structure, what we understand is that we started from a 6-membered hexapyro. I mean, we have the pyranose structure, and from what the crystal structure showed is that it's actually turned out to be a furanose. So the question is that how come a pyranose transferred to a furanose? And the furanose form the macrocycle also, one with the cyclic dimer, cyclic disaccharide, cyclic trisaccharide, cyclic tetrasaccharide. Remove the protecting group, and now we have this one. Notice that I started with a pyranose, I ended up getting a furanose. Ring contraction happened. Now the nature takes care. You try to tame me, I will tame you. You will not get the 6-membered ring, because you are trying to overpower me. I will show you a lesson. You start with a 6-membered, you will end up with a 5-membered. That is the reason I am not making. Nature says. Yes, good enough. We see that a cyclic 5-membered ring is formed, furanose, a furanose forms instead of a pyranose. Now the question is that these are the crystal structures, very beautiful. See, one of the beauty of working in this macrocycle is that it just crashes down as a crystal, single crystal. You can do beautiful single crystal and unambiguously assign the structure. So we don't have to assume much because the crystal structure is already there. So now, so beautifully, the 2E conformation has come in here. So as the nature requires, we reached 2E conformation, envelope conformation. However, it is not a 6-membered pyranose, rather it is a 5-membered furanose. So where does the 5-membered furanose came in here? Where is that? Here it is. I think this is my last slide. This is my last slide. What could be the possible mechanism of this? This is absolutely something very unknown, basically not known in the literature. That in a glycosylation reaction of a pyranose, you will end up getting a furanose. If that is the case, Dr. Jayakumar will be scratching his head. I do a glycosylation of a pyranose and I end up getting a glycoside product which is a furanose. So practically unknown in the literature at large. So what happens in these cases is, what is so special about it? So you see that here is a glycoside donor, and the glycosyl donor in the presence of the activators has to generate this type of oxocarbenium ion, which is an electrophilic center. For that electrophilic center, we have the nucleophile here, which is the hydroxy group. Normally what will happen is that this hydroxy group undergoes intermolecular reaction such that the monomer become dimer, become trimer, and then cyclize. This is what was anticipated. However, in this case, what happens, what appeared to be happening, two theories, one is mine, one is the students. Okay, so I think that this is how it has to be. Student thinks no, it cannot be. So then we compromise. Okay, we put both of them because I have my own feeling of that reaction. And that feeling is that how much soever good you do it in academic research laboratory, there will always be moisture. Let us take it as a fact. The student says, no, I have done outstanding, there won't be any moisture. No, you will have some moisture because we don't have that kind of chemical research laboratory where everything can be done in an anhydrous condition. So what happens if there is no water? If there is no water, this is my students, this one. So this hydroxy group intramolecularly forms this particular anhydrous sugar, 1,4-anhydrous sugar. So what you see here is that this hydroxy group attacks here so that it forms a bicyclic system, and that bicyclic system, because it is under acidic condition, protonates this one, and that protonates followed by the electron migration leads to the formation of a 5-membered cyclic system from the 6-membered pyranose to a 5-membered furanose, and two of them come together to form this cyclic disaccharide. And I was guessing that, look, you cannot avoid how much soever. If there is a moisture, what we will be having is this open acyclic form of aldehyde and C5 hydroxy group, and then now the C4 hydroxy group is also available. It can undergo a furanose formation rather than a pyranose formation. Where the C5 will attack, it will be pyranose, whereas the C4 reacts, it will be furanose. If that is the case, this is what we will have. Under acidic condition, we will form the oxocarbenium ion, and we get this one. And very fortunately, when it went for refereeing, both the reviewers told that, look, we cannot contradict this mechanism. This is outstanding mechanism. Please don't ask any question, accept the paper. So the paper got accepted in no time. And of course, I'm going to skip off what these molecules can do. Cyclodextrin means host-guest chemistry. So a lot of host-guest chemistry happened, particularly to evaluate the thermodynamic parameter. Because of lack of time, I'm not going to go into the detail. I think I have consumed my time now. So I skip off these things. Only thing I can tell is that in terms of the binding constant, compared to commercially available beta-cyclodextrin, when we have the dimer, it is at least well over two times better than in terms of binding constant for a particular antibiotic drug molecule called amantadine, adamantane. So what I tried to tell is that I'm not going to go and read out this one. The take-home message is that nature does something. Nature conveys a message. Take it up as a challenge. Nature is unable to make any cyclodextrin with glucose molecule less than 6 molecules, or once in a while 5 glucose units. If we try to make anything lesser than that, the conformation will be distorted. It is not possible to make. Irrespective of it, Yamada has shown it is possible. That is a science paper. We, in our own way, took care of the conformational issue by the way of introducing extra methylene, exomethylene, so that we are able to maintain the 4C1 conformation, in which case we are able to make the cyclic disaccharide, trisaccharide, tetrasaccharide, pentasaccharide, etc. But if you are trying to overdo things, this is what will happen. Your 6-membered ring will become 5-membered ring. So the nature has the last laugh. If you want to make any cyclodextrin less than 3 or 4 or less than 6 units, it will break open, it will close again, such that 6-membered will become 5-membered. So at the end of it, all that I can tell is that answer is yes, answer is no. Yes, we are able to make. No, we can't make in the same way that nature does. So this is the message that we see, and I think I should stop at this point in time. I hope I did not consume extra time here. I'll be happy to answer any question. Thank you. So before that, maybe I should also mention the people who have done. Look at this. Started from, he graduated in 2007, Shomak Pal. Work is still happening. Emil Fisher told that, I'm not getting anybody working in carbohydrate chemistry back in 1901. And Dr. Jayakumar is extraordinarily lucky person. He has so many people working in carbohydrate chemistry. Look at this series, only 6 students working for last 15, 16, 17 years. Ask Kandasamy how many is ready, how many is ready to work. But there's nothing wrong in the chemistry. Nothing absolutely wrong in chemistry. If somebody says carbohydrate is very complex, the complex is in our thinking, not the molecule. Don't put the blame on the molecule. Oh, carbohydrate is very tough, very complex. Nothing is complex. It is in our thinking that it's nothing to be complex. You just, Greek language is complex, Hebrew language is complex, if we think, but there are people in a country living out of it. So there is nothing complex here. Anyways, the research goes on, and then we must thank the SCRB as well as DBT for helping us to run this research. Thank you.
We can have one quick question from.
Yeah, this very fantastic work, and we really enjoyed your talk. I had just one question, that other than glucose, can't we use mannose or galactose for making the cyclic one?
There is absolutely no difficulty. See, 1,6-cyclic oligosaccharides are known. They are called isomaltose.
Yeah.
So their cyclization products are also known. Similarly, 5-members are also known cyclic oligosaccharides. They are called unulins. INULIN. Unulins. They are also commercially very heavily used. But when your focus is cyclodextrin, it has to be only glucose. But by saying all these things, we are synthetic organic chemists. So we can incorporate the modification we want right in the monomer and allow the oligomerization to happen and cyclization to happen. We can have. Yeah. But the point is that when you make it, what do you use it for? So that must strike us. Okay.
Yeah, thank you.
So I got a special permission for one more question.
Please go ahead.
So I have gone through the fantastic, lecture. I was quite impressed with your ability to make that disaccharide, trisaccharide cyclic by just incorporating 1 carbon more, which gives you the ability to make that.
Yes.
The C13 NMR of the trimer giving you only a monomer kind of a, so it's really, scientifically so exciting. But at the end of the day, what I'm thinking, because coming from a industry background, these are fantastic molecules, new molecules. Is there a way that we can elicit some biological activity or some use? And I think if we do that, I think it opens a new area, because this is an unexplored area.
Sure.
It is not a sugar. It is a 1 carbon extended kind of moieties. I'm sure we can work on that.
True.
I'm sure you would have thought, but I just want to know what is your, journey.
See, we have incorporated extra methylene. So that way, hexose has become heptose. Heptose is quite natural. except that that extra methylene is not at the C4, it will be at the C6, at the primary hydroxy positions. Irrespective of it, there is no particular cytotoxicity difficulty with these molecules, so they are very safe to be used. So that is always available to us. So so these are new chemical entities, no doubt. Can we take it up for a particular biological study? It can be taken it up, no doubt. Methods published. It is available. Go ahead. We are not protecting anything. You please send your person. I will give you everything even more. Please go ahead and do it. It is all available. Totally available. They are non-toxic. No difficulty. Do whatever you want. Very recently, a group from Hungary and Brazil joined together have shown that they are cardiac drugs. They are ameliorating, they are overcoming, they are acting as a to arrest any cardiac problems. They have shown cyclic oligosaccharide does that. So new properties are being brought out. So we don't know what is in store for them from a biological perspective because we are not biologists. We can do only some biophysical studies, some biochemical study. But if we have to do some animal studies, other things, it has to be done by experts.
As you rightly said, sugars, because of their nature, the availability from nature, I think the safety-wise, I think they are mostly very, very good molecules.
No issue. Safety-wise, absolutely no difficulty.
Any biological activity,
True. It will undergo sugar metabolism. When it undergoes sugar metabolism, you gain energy. What else you want? You become more active. You work in sugars, you will become even more active because it gives energy. Yeah. Ganesh. Yeah.
So regarding your C2-deoxy
Yes.
being less, more hydrolytically labile.
Sure,
So what is the reason why it should be labile?
Well, I think, the conjugate base activity of that glycosidic oxygen is probably even more compared to the one that carries the C2-hydroxy. The C2-hydroxy probably has some kind of hydrogen bonding possibility with the anomeric oxygen. And such a kind of possibility will not exist if that hydroxy group at C2 does not exist. So it is basically a conjugate base acid reaction. So that is probably what provokes to the formation of an oxocarbenium ion by the way of protonation. I cannot go back because I know that the time is up, but if I can quickly go into that one, I will tell you. So his question is that why it is hydrolytically unstable, and I'm sure that Ganesh was very much there when Somakpal was there. So probably they talked about this problem right on the corridor. So it is coming up today. So no issues, but it is a very good question. So you see that here, hydrolytic stability here. So when the hydroxy group is not there at C2, most probably, unlike in the case where hydroxy group is available for some kind of hydrogen bonding interaction, there is no such hydrogen bonding interaction. Now you see that the protonation of this one is probably more facile, more fast, or the conjugate basicity of this oxygen is better than the conjugate basicity of this oxygen. So as a result, once it is protonated, the lone pair of electrons on this one will come out, and then the carbon-oxygen bond breakage will happen to form the oxocarbenium ion. So this is probably one of the reasons why C2-deoxy sugars by nature are hydrolytically unstable. And Dr. Kandasamy is very clever man because he removed the oxygen and then put carbon. Now there is no question of hydrolytic stability, but then he has some other problem.
Just a quick one. The C6, the furanose to pyranose conversion.
Yes.
So is it because of the faster ring flipping that the OH group becomes
So
axial?
what we guess is that because we are only fortunate that both the reviewers went gagas accept it because they could not find out an alternative for it. So if the switching is faster, it will be easier. It's taking time.
Maybe we'll discuss later. So it's
No,
fine.
I will finish it because I will be leaving in another 5-10 minutes. Enappa, it's not moving. Okay. Can you please operate it? Next one. Next. Here it is. So you see that you form this one, oxocarbenium ion. You have an intramolecular hydroxy group here. When it undergoes an intramolecular reaction, you form this 1,4-anhydrous sugar. This 1,4-anhydrous sugar. So this is 1, this is 4. The oxygen here is this oxygen here. 1,4-anhydrous sugar. Now you see that it's a bicyclic ring. It will be very strained ring. That strain in the presence of the proton immediately cleaves that to give you this particular, when it opens, it does not open the 1,4, rather it opens this 1,5 ring, such that this 5-membered ring is retained, whereas the C1, C5, C1, that bond is broken such that now this is originally your oxygen here. That oxygen has come out here. This endocyclic oxygen has become exocyclic, and the exocyclic C4 oxygen has become endocyclic. Right? It makes sense. Or alternatively, once you form this one, in the presence of moisture, you have a hemiacetal. Hemiacetal is nothing but acyclic aldehyde and hydroxy group. And because we have two separate C5 hydroxy, C4 hydroxy, the propensity to form a 5-membered ring is faster than coming back to the 6-membered ring. So that you get the furanose, and that furanose undergoes further reaction with the under acidic condition, generates this oxocarbenium, and two of them coming together, it forms the 5-membered ring. So it makes sense. So I am not very sure if anything can be better than this. I'm sure there will be something better than this, but one has to think about it. Okay.
Sir, only one question. Sir. So usually cyclodextrins are well known for its solubility.
Absolutely.
So when 2 glucose molecule, 3 glucose molecule, such a, what is the solubility of that molecule?
Solubility per se is,
Aqueous solubility.
Okay. I can separately give one lecture on the solubility behavior. When what is soluble at room temperature becomes insoluble at higher temperature. That is the beauty of cyclodextrin. Normally, when you want to solubilize any compound, you heat it, right? If it is not soluble in room temperature, you heat it, it should be soluble. However, when it comes to the cyclodextrin, it is exactly reverse. So it is a lot to do with the hydration of the hydroxy group. If it happens, it will be solubilizing. So when we talk about micromolar or millimolar concentration, no particular issue. They are sufficiently hydroxy group enough where it will be soluble. However, when you heat it, it can precipitate out. This is so-called dehydration in water. You dehydrate in water, so it falls down. That basically tells about the hydrophobicity. Very beautiful article. No, the next person sitting next to you, Dr. Nityanandan. He worked with Professor Ramamurthy, one of the most outstanding man in the capsules, in the macrocyclic molecules. They come up with a theory. It is nothing to do with the hydrophobicity per se, Dr. Nityanandan. It's not the hydrophobicity alone. It is the ability of getting the two molecules in the right orientation such that the reactivity is enhanced 3 times, 4 times, 5 times better, because the cyclodextrin type of molecule are cavities, macrocyclic cavities. So it has a lot to do with the solubility, hydrations, dehydrations, all kinds of things. A very beautiful molecule. Lots of curiosity can arise for this molecule, and once we start to work, you see the joy. And when you see the single crystal, that's it. You are there.
Thank you, sir. Thank you for enriching us with the carbohydrate chemistry. Your passion for research is clearly evident from the presentation, sir. Thank you, sir. It's a small gift from the organizers.
Really sorry that I could not stay over here to being with all of you yesterday. Today I am here, but I'm just moving. Tomorrow also I'm unable to be here. My apologies. Usually when I go to conferences, I don't go like this. But having committed to this, I decided I will not skip off. So I'm happy that I'm able to come here, but apologies that I'm unable to stay here till the all the sessions of this one, particularly for a lot of my young budding faculty members. I certainly would like to hear them. listen from them. But this time it is not happening. Apologies, because there is a big function in my department tomorrow and day after tomorrow in the institute. All those things has put a lot of stress on me relating to the time. So I could not find time. So sorry for this one.
Okay, it's the time to introduce the next speaker, Dr. Chandrakumar from IIT Gandhinagar. So, sir is the associate professor now, and he has obtained his PhD from IISc Bangalore. His research interest is asymmetric organocatalysis and asymmetric synthesis of bioactive natural products. So, with this brief introduction, I invite Dr. Chandrakumar.
Thank you for a kind introduction. Good morning, all. I'm very happy to be here first time to MKU. I really thank the organizers, Dr. Jayakumar and Suresh, for a kind invitation and excellent hospitality and very vibrant students and discussions. Today I'm going to talk about our journey of this application of this organocatalysis in making okay, sorry about that. I will take some time. It's not going back. Okay, up and down, left, right. Okay, sorry about that. So we are actually interested in utilizing organocatalysis. Apart from our interest in making several methodologies, we have to develop a few methodologies and then apply for the synthesis of bioactive natural products, particularly kind of gamma-butyrollactones got very much attention while we were looking at synthesis of application of this molecule for the synthesis of bioactive molecules. There is no need of a big introduction about organocatalysis. Yesterday, Professor Shekhar have given a very nice introduction about this field. I just want to use only this slide to show the younger audience one more time that proline is very much utilized, considered as the simplest enzyme in the world, and utilized very much for the synthesis of many molecules in a very simple methods like forming iminium, enamine chemistry. So why did I choose this particular slide to tell you about organocatalysis and introduction is because this got Nobel Prize for Benjamin List, who worked at that time with Barbas and Lerner. He was a postdoc that time, and he was a star author of the paper, and he got an extremely great credit on this particular invention. So the beauty of this method, using such a simplest organic molecule to catalyze, very useful reaction, goes with a lot of advantages. First, it's not toxic to the environment. Second, you can actually, I'm playing with that, I know. So you can actually utilize water in the reaction because water is actually happening, either forming or, just deforming again in the system. So it's very convenient, you don't need to close the environment, the reaction vessel with the argon, all this environment away system. You don't need to do that for this particular catalysis. In fact, sometimes we add water to make the reaction go better. You can do in open flask, right? So we have seen a lot of advantages of this. People talked about environment, very important. That is this era is going to start our synthetic route to conserve our nature rather than destroying. Anyway, already we destroyed a lot. Just we want to conserve the nature. Most probably, we are the chemist who has to take care of it. I believe this field is going to survive a lot and going to be utilized by industry a lot in the near future. So far, even though it was awarded with the Nobel Prize, still it is not gone to industry a lot. And there are if and but reasons so we have to solve and take it to the industry. So with this small information, just we want to show what we were interested beyond the work I'm going to present today. Just want to give a brief idea about what we have done in recent past. We were interested in functionalizing a molecule that having a dienamine system. Obviously, it can go reaction over here or reaction over here. Can you selectively do reaction in one position? That was a challenge we took while we were initially starting our research. And in fact, we were able to do that. Yesterday, there was a discussion about trifluoroethanol that could do wonderful work, right? Magic solvent. So in our case also, it happened that we are able to selectively functionalize at this position when you have trifluoroethanol as one of the solvents. Otherwise, no, it will give mixture of products. And we actually showed different electrophile that can react selectively at the gamma position and make it possible. Apart from that, we also got interested in using a proline as a catalyst, and we are able to even functionalize the delta position of alpha, beta, gamma, delta unsaturated aldehyde as a single diastereomer and with excellent yield and energy selectivity. The system we later understood, it is going via inverse electron demand Diels-Alder reaction. That was very fantastic. Because of that reason, we are able to get a single diastereomer, and we applied this for a synthesis of few bioactive molecules, including the potent cannabinoids. We also got interested in functionalizing simplest ketone, acetone, and make it a very highly functionalized cyclohexanone. And very recently, we started showing our attention that we can make this catalyst reusable, attaching to the polymer, and this particular core, first time we have developed it with a very few steps, 3 steps from commercially available source, we could actually make it. And we could reuse this catalyst multiple times, and we are not going to pollute the environment, I believe. Even though this catalyst is organic molecule, not going to be complicated to the environment, still we want to reuse it so that industry may be interested in looking at such a system. Okay, with this brief idea about what we have been doing, I just want to take you to the journey of we got interested in making gamma-butyroactones, chiral molecules, and apply them to synthesis of bioactive molecules. Gamma-butyroactones are almost 60% of the biological natural molecule has this core. And we just initially started to focus on making this R-paraconyl alcohol as our key intermediate. There will be any method by which you can make it in a shorter and efficient manner? There was a question we asked. This is actually R-paraconyl alcohol, has one chiral center at the beta position, right? So we want to actually find a method, what are all known in the literature. Before that, what are all the use of this molecule if you look? If you make this particular core, we could access many bioactive natural products in a very simplest way that if you just functionalize at this position of this particular chiral intermediate, you could make this variety of natural products, which are actually used by Streptomyces species to make antibiotics in natural system. So we wanted to actually access this chiral intermediate in a very simplest method. Before that, what are all known in the literature to access the intermediate is actually one of the methods known is that enzymatic resolution is known. If you just look at this system, they actually access with the 7 steps with the 83% enantiomer selectivity and 9% overall yield. There are other methods, only this method, these 2 methods are very much studied. I don't think anything better than this method exists while we were starting our research. And this particular method is a chiral auxiliary approach. You start with the chiral starting material, attach to the source of reactant, and then convert them into product, and then cleave the chiral auxiliary, you get this compound. This also goes by 7 steps from commercially available starting material with the 8% overall yield. In this case, you don't need to worry about enantiomer selectivity because you are starting from chiral compound. In between, you are making diastereomers. You could separate them by column chromatography. So we were thinking that is there any way that you can actually improve this method, or you can make a new method that can actually be better than this existing methods for the synthesis of this chiral starting material or intermediate or paraconyl alcohol. So our approach actually goes by this method that if you have this particular aldehyde with the ester group at the 4th position, we could actually functionalize with the simplest 1-carbon electrophile. Professor Jayaraman, our teacher, was telling about the foremost reaction. Actually, it's all start from formaldehyde. From formaldehyde, if you make carbohydrates, that is called 4-MOS reaction. It is very much known. So we want to utilize such a smallest electrophile to functionalize this. It's not that easy. Many aldol reaction known, but all with the different aldehydes. There are very, very rare methods known to access formaldehyde in aldol reaction because the problem is dehydration. Quickly it falls off. Once it is in this stage, very hard to control. So we took this challenge and we wanted to actually make this. So to do that, we actually screened various conditions. Before going to the condition, I just want to tell you that we are using formaldehyde in water for this reaction. And we are able to functionalize at this position aldol reaction that is not stable so that we could oxidize. When you oxidize with the Pinnick condition, it could actually, the aldehyde will become carboxylic acid. You could isolate. The problem is during this synthesis, this alcohol attacks here and lactone started forming. Some mixture of lactone and a free alcohol ester was there. So we went on to cyclize fully. Just we added a 6 normal HCl and we are able to cyclize this fully. You could actually isolate at this stage, that is carboxylic acid. So like paraconic acid at this position you can get. You also can reduce using BH3-DMS so that you could actually characterize comfortably. Handling carboxylic acid in many methods are not that easy, right? Especially you cannot do HPLC with those compound, especially chiral HPLC. So we wanted to actually reduce to alcohol. So we actually reached this paraconic anyway, that is our target to make those natural products. So if you look at these stages, all the stages we have done without column purification. At the end, we just do only one single column purification. It works. So this is our final compound to make a screening of the conditions. In other words, each and every time, we have to do all 4 steps to screen. The well-known catalyst for aldol reaction is proline. In our case, it is giving very poor yield after 4 steps. Please remember, it's every single stage you have to move and then finally you have to isolate this compound. So we actually screened some catalyst, solvent conditions, some additives, temperature also. So I don't want to go in every particular details, but I want to take your attention that this particular catalyst, which is nothing but diaryl prolinol, which is derivative of proline, was able to actually catalyze this reaction in an effective way, maybe in here, effective way, that even though it takes 6 days, and it is able to give 95% energy selectivity of the product with 35% overall yield after 4 steps of the reaction. I would consider that as a better yield because of considering the number of steps, even though that days take more. If you really want to do quicker, you have to use this catalyst, which actually can be done in half a day, right? 12 hours, you can complete this reaction. But problem is that it will give 78% E only. So you want to take E or time, that's a call. So we took E as the deal that we took this particular condition as our better optimized condition. So once you have paraconyl alcohol, what you have to do is just you have to functionalize this position to get the natural product. First, we thought that we could actually do acylation at this position and then do reduction. And then after that, if you remove the TBS group, you are going to get the natural product. But in this case, we were not able to get the better selectivity. And we thought that if you are not giving selectivity in this case, it's okay. We could actually go for direct alkylation, like aldol reaction, right? You should be able to get this, but here also the selectivity is just 2 to 1. However, we are able to purify this compound by Kalam, and then we just remove the TBS group to achieve this natural product in a very shorter step. If you remember that 7 steps to make this intermediate by the existing method, we are actually reaching the final target with 7 step itself. That is advantage. We believe that this could be very useful. That we patented this work, the synthesis of this particular paraconyl alcohol by our method. Also, if you just do other alkylation chemistry, you should be able to get other bioactive natural product, and acylation directly if you do, you should be able to get this natural product A factor. So in this method, we applied this chiral starting material for the synthesis of 3 bioactive natural products. So that's the summary of this particular small part, that we did this reaction in 4 steps. We did single column purification. We are able to reach up to very high energy selectivity and overall yield for this chiral intermediate. So we are actually got interested in other particular paraconic acid, which already exists in the literature. But problem is that did I move so much? That this, you have to consider one more substitution at the gamma position. If you are able to substitute at the gamma position with alkyl group, you could make a variety of natural products. But key here is that this natural product with respect to this, if you look at these two chiral centers, they are diastereomer in relationship, irrespective of alkyl group. In this case, both are towards us. In this case, one is behind. They are diastereomers. If you consider these two natural product correlation, they are actually enantiomer in relationship with respect to these two chiral centers. They are opposite to each other. So if you are able to develop a method for the synthesis of both enantiomers and diastereomers, in other words, all four possible isomers you are able to access, you should be able to access all these natural products. Even though the fourth isomer is not utilized in this case, but you should be able to access all four. So with this idea, we just went back to our plan, earlier plan, that we used formaldehyde, right? Instead of formaldehyde, if you use alkyl aldehyde, you should be able to do this reaction with the gamma position where you have LDCH2 here. Now you can get alkyl group. Problem is that how do you do two aldehyde reaction and tell one to act as an electrophile, other one is a nucleophile, which is difficult, right? So we actually selectively functionalized this position with the chloro. That is also done by proline catalyzed alpha chlorination first. Now you can discriminate those two. This is reactive aldehyde. This is actually kind of bulky aldehyde. It might not undergo enamine formation. That's the point. We actually were able to make it. There are diastereomeric mixtures, and purification problem, and that are all we somehow managed. What we couldn't manage is that removing this chloro at the end. Irrespective of doing many different methods, this chloro removal supposed to be removed because that we use as a chiral, bulky, not chiral, but bulky reagent at this position to do aldol reaction, right? So we are not able to remove that, so we thought we have to change our methodology. The methodology actually was considered as follows, that instead of aldehyde, can you consider there as ester, right? So that you can handle with the aldehyde and then ester, you could actually discriminate the particular chemo selectivity. Very easily we can do that. However, this reaction we found in the literature is known that you could do photocatalysis with this particular photocatalyst. We should be able to make this compound. We made it using the literature procedure. The problem is that reduction. We did several reduction. We did Baker-East. There are CBS reduction. All are known for this, but none of them works effectively. Then we were forced to take this commercially available ruthenium catalyst to do asymmetric transfer hydrogenation. The problem here is that it went well, but diastereomeric selectivity is exactly 1:1. We don't want that, right? Because we want to make all diastereomers and enantiomers. We want to have a selectivity. So what we understood that this ester carbonyl, both interacts with the ruthenium during the catalysis, it is actually making this 1:1. But if you actually selectively access with one carbonyl interacting during the hydrogenation reaction, you should be able to get this diastereomeric selectivity excellent. So how do you do that? We actually thought that we could actually make this particular intermediate where we locked this system. This particular carbonyl can interact with this carbonyl during reduction, but not this, because it has to go via bicyclic system, right? Which is very difficult to form, probably 2-to-1 bicyclic system. So we thought that this is possible, but surprisingly, this particular intermediate is not known in the literature. So we thought we can actually devise this method using NHC catalysis. In fact, this light mediated reaction we tried, but didn't work. So we went on to make a NHC catalysis for the synthesis of this compound. At least we have organocatalysis over there to make intermediate. So to do that, we actually screened various conditions with the NHC catalyst. Also, few of them we have chosen, studied. This did not work, but this started working. We got completely this product. We actually isolated these two diastereomers and characterized what is the product we got. Then we understood it is the product of this intermediate reacting further with this starting material to make this. It's like a Michael addition. Again happened to the system, and then we end up getting this product. We thought we could do retro Michael. If you do retro Michael, mean reverse Michael, you should be able to consume, revert back to this compound. And whatever exists here can be further taken. It is actually you have to develop a method by doing that. So we thought increasing the base would help. DIP went 10 mole percent to 30 mole percent. We started seeing small amount of this product in the reaction. Again, we started heating it up with the high reflex temperature. We are able to get only this product. In other words, this reacts to give this intermediate, and that further reacts to the starting material to give this product. No matter how many equivalent you are using, still you are getting this product. If you want to revert back to here and the remaining whatever comes out should be consumed again by the starting material, is the best method by which you can make this 85% yield of this particular intermediate. Of course, once you make a method, you just have to apply for a substrates, and very few substrate, even though we want only this 2 substrate we applied for few other substrate to show the versatility of this methodology. After that, we went on to do the asymmetric transfer hydrogenation optimization. I'm going to go in detail about what we have done with the different solvent, catalyst conditions, even temperature. I just want to stick to this particular condition by which DMF and catalyst 3 found to be useful for this. All these catalysts are commercially available. We bought it. Just we found that it is giving excellent yield and good diastereoselectivity and very high enantiomeric excess. So once we achieved the transfer hydrogenation reaction effectively, what we need to do is that for making that particular lactone, we have to cyclize them, right? For that, we just tried as such to cyclize. It didn't go well. Did I go in the right place? Yeah. After this reduction, we want to cyclize this. Please note down, here actually these 2 chiral centers are towards you, that when we cyclize this reaction with a simple KOH, acidic mediated condition, whatever condition we tried, all the cases, we got this as a major isomer. In other words, this particular center got epimerized. 2-to-1 to 4-to-1 selectivity we got, but never ever we got the actual isomer as such during cyclization. We consider that, okay, we got other diastereomer, right? So what we need to do now, we need to make a same diastereomer of this without epimerizing this center. We found in the literature, when you have a NBN, it might not, where I am actually, it might not go good. What we have done is we actually hydrolyzed this using a very milder condition. 5% aqueous KOH at 0 degree, we are able to just hydrolyze this particular bond. When you have a NH over here, there is another method known in the literature that you can actually do sodium nitrite with acetic anhydride. You should be able to make in situ ester, and that cyclizes very easily. It's like a mildest method of making, amide hydrolysis is very difficult. But if you have ester hydrolysis, it's going to be very easy. Lactone can easily open up, closed very easily. So we should be able to do that, and this reaction condition worked very well for us, even though we struggled a few different methods for some time, but we are able to get this to be done. Now you see that this center as such preserved. So in this case, we made both the diastereomers of this product with a very high enantioselectivity and very good yield. So now what you have to do, we have to make other enantiomers of these diastereomers. For that, we just choose an other enantiomer of this sorry about jumping here and there. The other enantiomer of this catalyst, we should be able to make SS to RR, and same condition you can apply that you should be able to get both the diastereomer of the enantiomer. So in this case, we made all enantiomers and diastereomers of this paragonic acid with the very shorter steps, an excellent overall yield. So once we have this, what you have to do is that we have to just do methylation of this system. If you methylinate this particular compound, RS, chiral center, you should be able to get this natural product. If you methylinate the enantiomer of this particular lactone, that is nothing but SR, you should be able to get these natural products. Just you have to do methylation instead of methylation. I'm slow, I think. Okay, I'm sorry about that. So if you actually do methylation of this process, this particular intermediate, you should be able to get all natural product. In other words, in this methodology, we made 7 natural products with a very, very short methodology. I don't think I'm going to explain the other one, but I'm just trying to run through very fast if possible. So we used this particular starting material. In a very fewer steps, we are able to access this compound in 47% overall yield. This can be done in gram scale without any problem, because such a very simplest method we are handling to access this compound, all possible 4 diastereomer enantiomer being synthesized and applied to the synthesis of biologically useful natural product. Okay, the other project I may not go very slow and steady. I will run a little faster. We actually thought that in this case, the earlier case, we did 3 isomers of 4 isomers. Is there any, all 4 isomers are naturally known for the chiral center of 2. So this particular pyrrolizidine alkalides, we found even though different source is being isolated, all 4 possible isomers of 2 chiral centers already known in the literature. And its corresponding ester derivatives are very much useful to different biological properties. So we thought we can actually use our method, or different method we can develop, that all 4 possible isomers we can synthesize. For that, we have chosen the simplest self-managed reaction. I will not go into much detail about it. If you take aldehyde of this, which we have used earlier, almost all this project we were using this aldehyde, right? Just use PMB amine. You could actually make imine, and then other one acts as a nucleophile. You can do managed reaction. If just only you need two starting materials, you can make this 3-component kind of reaction, and you could actually make this compound in a very simplest way. And the advantage we have here is that if you use such a bulky prolinol catalyst, which already Professor Sekhar yesterday was mentioning, you should be able to do the reaction at the backside of the iminium, right? Because the catalyst, this aldehyde, iminium, this particular group will block the front side. The reaction can happen at the backside. And we should be able to get this particular isomer, which could be in quicker time. We should be able to make this a compound. And once we have this method, kind of we struggle a little bit to establish. Once we establish that, we should be able to make this plus isoretronecanol with the commercially available proline-derived catalyst. If you take enantiomer of this catalyst, you should be able to do exactly the same, but a minus isomer can be synthesized. All conditions are the same. And if you want to make other diastereomer, we thought that instead of using this chiral bulk, use proline, which is actually hydrogen bond donor. We should be able to make quicker optimization we have done with self-managed reaction, and we were able to make, using the same method but different catalyst, we are able to make other diastereomer, and corresponding enantiomer can be made commercially available D-proline, right? So we are able to do that. So all 4 possible isomers of this pyrrolizidine alkalides we are able to accomplish. With this, I just conclude by saying that this many, did I go very fast? Just last 30 seconds. It's not moving. All this natural product being achieved using this 3 simplest method, starting from asymmetric organocatalysis. So with this, I thank all my students, particularly Abhijit, whose research I have presented here, and funding agencies. And thank you all. Please visit us wherever you have a chance for IIT Gandhinagar. Thank you.
For the tea break. Any questions?
Please.
Yes, really fascinating. Have you ever thought about using chiral NHCs?
Chiral?
Chiral NHC.
NSC, that problem is that center is highly epimerized. It's very difficult to...
I was also thinking that, yeah.
Yeah, if you do anything here, that particular chiral center would go. In fact, that is the advantage we are making dynamic kinetic resolution.
Yes,
That's what I didn't mention.
DKR you are doing.
I stick to asymmetric hydrogenation.
The yield I saw is more than 90, so you
Yeah.
must have, yeah. That's very good. Yeah, thank you.
Thank you.
Yeah, Chandrakumar, fantastic work. So a couple of small questions. So the one you had this DIPA, where you had
Yeah.
this control on the
Yeah.
retro Michael addition. So
Yeah.
how does the DIPA facilitates the retro?
Actually, it picks the proton. You know, when you have large excess, okay, now it got stuck. Somebody is moving for me? Yeah. So, when you actually pick this proton,
Oh, right. Okay.
this can actually go reverse.
Fine.
But you need a little more. And if you heat it, obviously it can happen. What we want to, once you consume all of them, this is not further reacting. It is all about you have to do retro, Michael addition, and then consume this with this aldehyde. We will get only this product.
Okay. Yeah, fine. But doesn't again go forward?
If you do not have this, it will not go forward, right? It is a bottleneck is this compound should not exist in the reaction, right? If you have enough aldehyde, let it go here, but do retro Michael, whatever comes, this also comes and this also comes back, right? Consume. Once all this compound consumed in the reaction, only this product exists. That is why we did reflex. We are not getting the other product.
Yeah. The aldehyde is the excess.
Yeah, we always use 1.5 equivalent.
Okay.
Yeah.
The other small question I had is the diastereomer selectivity you had in the aldol reaction.
Yes.
So that one, like, seems like the major isomer you get is the thermodynamically stable product.
Actually, here kinetics plays a major role because we are doing catalysis with proline. We are talking about probably here, right?
Slides 12
Here.
or 16 somewhere.
Yeah,
No,
here.
12. No, well in advance in the beginning. In the beginning, like.
Formaldehyde aldol reaction.
Yeah.
Yes, but the more stable compound thermodynamically is the dehydrated product. I hope,
We'll discuss later.
yeah, we can discuss.
Okay, please join me in thanking Dr. Chandrakumar for his impressive presentation on natural products.
We shall have a short break for tea. And as usual, speakers use the right door and participants do exit through your left door. Thank you. Be quick. Let us welcome Dr. N. Selvapalam from Kalasalingam University for the next part of our proceedings. Please join us in extending a warm welcome. Over to you, sir.
Check, check. Fine. Okay. Presenter.
Welcome you all back this after our morning session. Now I would like to introduce Professor S. Manivannan. He has been associated with BHU and he has done his undergraduate from University of Madras, and then subsequently he has done his PG and PhD from Madurai Kamaraj University under the Professor Ramraj. And now he will, he also spent quite a number of years in abroad, especially in Korea. And then he moved back to the BHU as assistant professor. Now he'll give a talk on the electrochemistry and nanomaterial and their applications. Thank you very much. Now I'll give the session to
Yes, sir.
Manivannan.
Thank you very much for the nice introduction. So, since morning, we got covered with a lot of organic chemistry. So now I would like to bring some material chemist aspect. Okay. So for example, if you look at my title, there is a virus, M13 virus. So recently we have come across this COVID pandemic. So everyone might have afraid that what this fellow is going to talk with a virus. So don't worry, not all the viruses are harmful. The one which I am going to introduce is human friendly. So very easy to work. So nothing afraid to discuss about this M13 virus. Okay. So for example, I'm going to introduce a biosensor probe. What is biosensor probe? For example, if you are constructing a sensor device for signal transducing, if you are using a biomolecule, we can call it as a biosensor probe. Since I'm going to use this M13 virus as a biomolecule, so I am going to call it as a biosensor probe. And the next keyword in my title is aggregations. So I'm going to use a nanomaterial as one of the component for my biosensor probe. So whenever we come across nanomaterial, irrespective of what shape, dimension, morphology of it is, when it get into the aggregation, there is a problematic. So irrespective of any shape, if you have come across aggregation problems. So I'm going to talk about that aggregation, how I can avoid aggregation when I use my biosensor probe for sensor studies. Okay. So what kind of element I'm going to detect? There's a mercury, well-known pollutant. So I will be touching biosensor probe, how I can avoid aggregation. So what analyte I'm going to use, what I have achieved something. So that's what I'm going to discuss in brief. To start with, so I would like to highlight some of the key aspects of this mercury pollutions. So for example, mercury, you can see it, it's a more beautiful element, okay, very flexible one. You can see it, but you can't touch it, it's a harmful one. So what is the source of this mercury pollutions? Through the natural resources, for example, vulcanized geological springs and glacier slits, mines process, and as well as the anthropogenic things, for example, coal combustion, industrial effluents. Through various sources, mercury gets added into ocean or water into it. Okay, once it reaches water, and slowly in the biological cycle, it reaches to fish, and we are human, we are consuming that fish also. So thereby it reaches to our body. Fine. In related to this, I would like to highlight one of the incident associated with mercury pollution, which one is the Minamata disease. So which happened long back 1956 in Japan. There was one small village where the childs as well as elders got affected by a suspicious disease, like their fingers got the numbness. So they don't know what happening in that there. Later it got revealed that there was a nearby industry which is keep on dissoluting the mercury into the ocean, and that mercury converted into methyl mercury that reached to fish. Since it was a farmer village like fishing, they consumed that fish. Through that mercury entered into their body, they got suffered with this, the Minamata disease. And one more thing, if you are come across this mercury, there is one famous disease, that acrodynamia. For example, it used to affect mainly children's, where your fingers used to be like more pink of color. Okay, for example, in ancient days, people used mercury to separating metals from the ore. For that, without any safety measures, they have handled such like that. So those people have suffered this kind of acrodynamia disease. Okay. And I said that mercury reaching this biological cycle through fish, it's entering. So after we consume fish, let us say mercury incorporated into fish, how it enters, how it causes all these diseases for us. So for example, so this point I would like to highlight. 1 minute. So for example, let us say we are consuming this methyl mercury from the fish. So when it enters into our intestinal tract, for example, it used to associate with one of the amino acid, essential amino acid present in our body, like a cysteine. It used to form a methyl mercury cysteine complex. Okay, this methyl mercury cysteine complex is almost identical to the another amino acid, which is methionine. So now our body has the role to transport these amino acids to various parts of our body. For example, let us say essential amino acids to be transported to our brain fluid. Now the amino acid transporters do not have the distinguishments between this methionine as well as this methyl mercury cysteines. So thereby the amino acid transporters allows this methyl mercury cysteine to our brain or various parts. Thereby mercury enters into the human body and causing a lot of troubles. Okay. So now, after introducing all those things, I don't want to put you an embarrassing question that should you avoid fish because of mercury? Okay. So now we learned that mercury 2+ is one of the highly stable species, and in general inorganic versions of mercury pollutions too. Okay. So there are several approaches available in the literature for detecting this mercury 2+. For example, the electrostatic interaction, some complex formation with the biomolecules, and inorganic complex for detecting mercury, and fluorescent stacks, as well as some biomolecules things. So all these things are already available in the literature, but still what lacks? So what one has to develop to detect this mercury more efficiently. For example, the US Environmental Protection Association and other World Health Organization, as per their standards, we have to detect the mercury in the drinkable water less than 10 nanometer. If it comes for wastewater, it should be 0.15 milligram per liter. In the case of atmospheric air, it must be less than 75 picomolar. Whereas in the case of soil, it should be 260 milligram per kilogram. So in food, very importantly, it should be less than 50 nanomolar in concentration. So what does this parameter indicates? Whatever the methodologies you develop, it could able to detect the mercury present in various elements up to this levels. So one has to develop a sensor probe which could capable of detecting this range of mercury 2+. That is the lesson. So now in this regard, there are various things are available. For example, if the mercury, if you want to detect at the laboratory scale or the industrial scale, you can easily do that with the help of the mass spectrophotometer and atomic absorption, so on. But
it is not all the time mercury detection is needed in the laboratory things only. Because, for example, if you want to detect the mercury in the remote areas, for example, you are reaching some mountain where you are finding a pool of water. You want to detect the mercury concentration in there. You can't carry all those sophisticated instruments with you over there. Okay. In that case, you need a very simple, economic, and very smart, easily accessible sensor probe where you can easily detect the mercury in the remote area also. So even though a modern equipment available for the detection of mercury at the trace level, since a easy to transport economic one devices are much needed. And one more thing, I have highlighted the aggregations because I'm going to construct the biosensor probe with the help of nanomaterials. I have highlighted the importance of aggregations also. For example, what is meant by aggregation? For example, whenever if we are using the nanomaterial, mainly we are going to harvest its surface plasmonic properties. Okay, so surface plasmonic properties will retain unless otherwise it's not getting agglomerated. Okay, so once it gets agglomerated, the unique property of surface plasmon resonance goes away. So for example, the above picture is the example of a simple nanomaterial present in the solution where there is no aggregation. You can easily harvest their surface plasmon properties. But when you use this nanomaterial for detecting, let us say example, this kind of agglomeration happens. Once your nanomaterial get agglomerated like this, then the unique property of surface plasmon resonance gone. It is no longer useful in detecting the mercury or any other analyte. So whenever you are using a nanomaterial, you must look into the aspect that how to avoid this kind of aggregation. That is the one of the very important concern. So that's what I'm going to introduce, what I have done in this regard. So one more thing, the mechanism which I'm going to follow for detect the mercury is the amalgamation process. As we know, when we mix any metal with the mercury, it used to form amalgam. The same strategy I'm going to use also. Okay, so now coming to the virus, because I have constructed my biosensor probe with the help of virus. So there are 200 kinds of virus available, which are majorly categorized into 4 different types. So the first one is this tubercular mosaic virus, and second one is adenovirus, third one is influenza virus, and the fourth one is this bacteriophage. So I'm going to work with this kind of bacteriophage virus, okay? So in this category, so I have chosen this M13 virus, okay? What is the speciality about this M13 virus? If you look at the structure carefully, which is fibrillar in nature and which is having 880 nanometer in length and 6.6 nanometer in the diameter. The beauty of this virus is because of its fibrillar nature and length and this width and the length, which offers the high aspect ratio. So one can make use of high surface area out of this M13 virus. And very important thing about this M13 virus is the protein code present over this M13 virus, which are available for the functionalization, surface functionalization, okay? So whatever the protein code available over there, with the suitable strategies, you can properly functionalize the virus surface, which is referred as a phage engineering technique, okay? So through that phage engineering technique, you can attach or functionalize any molecule of your choice. So already researchers did various attachment. For example, they have found attaching different molecules for the drug delivery, biosensor, batteries, tissue regeneration, photosensitive material, imaging detection, antimicrobial agents, and so on. And we are working on this biosensor category. So this virus surface is available for you to do any kind of modification, which is referred as a phage engineering. In that, we were working on. Okay, so how we are doing this, the phage engineering, the virus amplification. I would like to briefly introduce in that aspect also. So we all know that virus cannot involve in any metabolism. So it has to attack the corresponding bacteria and it has to amplify. Okay, for this purpose, we started with the bacteria, which is E. coli. Initially, this M13 phage, the source one, used to bind with the E. coli bacteria. This process is referred to binding. After that, it will inject its the genomic moieties, which is referred as an injection of nucleic acid. Then there will be a starting of reprogramming. The next step is production of phage particles inside this E. coli. Next, there will be assembly. They are ready to explode and get coming out and go for further amplification, which is referred as a lysine and lysis process. Okay, so in this case, you can do any kind of modification. But what kind of modification we did and what was the purpose for that? For example, we have chosen 3 different types of viruses. One is, I am going to call it as a wild type and glutamic moiety attached. This Y3 virus is capable of attracting specifically to gold ions. That means if you introduce any metal precursor along with gold, this particular Y3 virus will show affinity only towards the gold precursor. So there is a selectivity brought into picture. The third one is the glutamic acid functionalized virus. The beauty of this virus is it's cation specific. Means what? Like if you introduce anions and cations together, it will show selectively affinity towards the cations, so that you can make a selective purpose and you can make a suit for further things. Okay, for this purpose, how to identify a particular functional station is suitable for a particular affinity, it should be performed through this phage display technique. This technique is actually won the 2018 Nobel Prize in Chemistry for Smith and the Nilmer. Okay, so in this regard, there are already several good reports available. For example, the Belcher et al. Publishes several good papers for utilizing this kind of phage engineering moieties for like energy harvesting and storage applications. By getting inspired with that, we have also done some of the works. So through these phage molecules, we could able to produce some interesting structure. The advantage of all these structures are the different size and shape. Through this various different size and shape, we could harvest highly electrochemically active surface area, and we have utilized those for several electrochemical applications. And not only it's restricted to the energy storage applications. There are a number of reports available for biosensor things also. Professor Tsangukli, he is one of our collaborators in University of California. He is developing such kind of phage involved biosensor probes. So if someone is interested, you can go through this article. Okay. Okay, so with this introduction, what we have done to avoid the aggregation for detecting the mercury sensor. For example, we started with synthesizing a first project like a AU nanovirus. For example, we started with the M13 Y3 functionalized virus, as I have highlighted, which shows affinity only towards the gold precursors. I have introduced the gold precursors. The gold precursors get attached over the protein coat of this virus molecule, and by following a certain procedures, we could able to convert these precursors into nanoform. So here what we have done, by using this fibrillar M13 virus surface as a solid support or a mold, we could able to clearly wrap the gold nanostructures. The idea was to obtain the like a rod-like or wire type of structures. So if you attain so, we could harvest a higher surface area. That was the idea. Let's see. So for that, so we have performed an experimental analysis without including the M13 virus. So this gold nanoparticle TEM image is obtained in the absence of M13 virus. In the absence of M13 virus, by following the same protocol, we could able to end up with only a spherical particles. But in the case of M13 virus introduced protocol, what we have obtained is see here wire type molecular structures. So it is very clear, we couldn't see not even a single sphere particle inside in the same image. Further, we have went for the TEM analysis where we can clearly see a nano wire like structure. Okay. So the elemental mapping analysis also confirms the presence of gold and a trace amount of silver in here. And the beauty of this protocol is previously some of the reports are available for this kind of molding purpose, but all they could end up with like only coating gold or other metals with the single M13 bacteria phage. But through our protocol, we could able to attach a subsequent M13 virus by end-to-end type. That we claim that we have followed a oriented attachment between the two edges, so that as long as you want, you can go a continuous growth of nanowires. Okay. And this one we have introduced for the mercury detection. We have conducted the UV titration for performing this mercury detection. And this figure C, which shows that when we introduce mercury to the gold, so gold used to exhibit a surface Boltzmann resonance band around 550. So we have added a mercury and got the titration. You see clearly when you are adding a mercury, there is a redshift and it's going down as quick as possible. So it indicates that a spherical particle quickly get aggregates. Hence, the surface Boltzmann behavior used to go down. So it is a very clear indication that there is a clear aggregation when you are using a nanospheres. On contrary, if you are using a M13 attached nanowires, there was no such redshift occurred in here, and only a decrement is in the same with respect to the same wavelength is happening. And this indicates that there is a sensing as well as no such aggregation also. So what is the beauty of this kind of experimental results? For example, like if you add like within 0 to 50 micromolar of mercury, within that window, your spherical nanoparticle become incapable. It's lose all its property. For example, this nanowire-like structure, even 0 to 140 micromolar of mercury concentration, it do not loses its property. That means within this window, as low as concentration of mercury, we could able to construct a biosensor probe. So that is the beauty of this result. And further, this studies also we have done. And we have conducted a what is happening inside at the molecular level. We have conducted some mechanistic investigation. For that, we have performed a TEM analysis after the experiment. And it is very clear that if you are using a spherical particle, a clear aggregation is evident from the HRTM analysis, whereas in the case of nanowires, there was no such aggregation. The nanowire structures are got retained. And we have conducted the calorimetric sensor things also. The results are summarized in here. So this is one work which we have published in the biosensor and bioelectronics. Not moving. Okay. And in the second project, so we got inspired with some of the one of the researcher, like they have published a paper in a small, where they could assemble a gold nanocubes over the M13 virus surface through a proper functionalization. So we got inspired with this work, and we thought of how about assembling a sphere particle over this M13 virus. We did some studies. I would like to summarize in here. For example, if you are thinking of assembling a spherical particle in a linear way, there is a problem. What kind of problem it is? Like whenever you are trying to assemble a spherical particle, it always prefer to gather in this orientation only. This is the nature preferred orientation. It is very difficult to assemble in a linear manner like this. Okay. So if you are trying to assemble in that linear manner, what are the advantages? For example, if you try to assemble this spherical particle in this kind of linear manner, we can beautifully harvest a interparticle surface Boltzmann coupling. Okay. Yeah. So this kind of, in order to harvest this kind of surface Boltzmann coupling, so we thought of arranging a spherical particle in this linear way. And we have conducted, spherical particles, and we have characterized the gold silver core shell nanoparticle, which is core shell clearly evident. And what we have done, we have introduced this M13 4E virus to assemble this spherical particle. You see here, when there is no M13 virus, there was the scattered assembly. When you introduce your M13 virus, there is a clear assembly of one-dimensional array in here. This what we are claiming that we could be able to achieve this one-dimensional array. And this one-dimensional array we have used for the detection of mercury. This is my last slide, I will conclude. So in order to conclude that there is a one-dimensional array. We have done some controlled experiments also. All together, we came to know that for preparing the gold nanoparticle, we have used a cyclotexin functionalization. The cyclotexin functionalization facilitates the hydrogen bonding interaction between the two nanoparticles, as well as there is an inclusion complex formation between the 4E moiety with the cyclotexin, which facilitated this kind of linear array. So similar to the previous work, we have conducted the mercury titration. In the absence of virus, there was a clear redshift, which indicates the aggregation, whereas in the presence of virus, there was no such aggregation. So we claim that there is an aggregation-free biosensor probe detector for mercury ions. Yeah, that's all. Finally, I would like to highlight the funding support I received, and as well as I would like to express my thanks for my postdoc mentor and this girl, Joan Jing, who is contributing for assisting these works. Thank you for your listening.
And now the session is open for questions.
Yep. Thank you.
Heavy metals are not the only enemies we have anymore.
Yep.
So now we are daily coming across several reports saying that they are finding microplastics in everything.
Yes.
In Italy, they found it in mother's milk and they found it in water. So do you think biosensor probes can help in regarding that?
Certainly, it can be extended to those things also, possible.
Yeah. So what kind of research should we?
Depending upon the nature of the target analyte, we can bring the functionalization over the M13 virus surface. Thereby, a biosensor probe can be easily constructed.
Thank you, sir. Yeah, that's
Any more? Quickly, one more question.
Yes, ma'am.
Also for detecting any other virus also, no?
Yes.
Sir,
Okay. Yeah.
sir, what do you mean by bibular nature, sir? Any unique properties of bibular nature?
With the help of that fibrillar nature, for example, I have highlighted that because of the fibrillar nature, there is a higher aspect ratio. So that high aspect ratio similar to the AU nanorod. For example, if you compare a spherical gold nanoparticle and a gold nanorod, with the nanorod, you can have the high aspect ratio. Because of that, you could harvest a high surface area. The same structure with the help of M13 virus, we could achieve.
Sir, this virus only censored வந்து I mean mercuryக்கு மட்டும் தான் யூஸ் பண்ண முடியுமா? வேற ஏதாவது heavy metals எதுவும் sensing பண்ண முடியுமா?
Surely we can extend to other things also. Here I have demonstrated just one example. So by having the same strategy, it can be extended to any other analyte.
Okay, sir. Radioactive elementsக்கும் இது
Certainly possible, because with respect to the phage engineering, with respect to the type of target analyte, you can perform the phage engineering. Thereby, you can easily construct a biosensor probe.
Thank you, sir.
Yeah, thank you, and please join with me to thank the speaker, Professor Manivannan.
Thank you. Thank you. Thank you, sir.
So let me introduce the next speaker, Professor P. Shanmugam. He is the Chief Scientist and Professor of Chemistry at ACSAR New Delhi. And basically, he is a Chief Scientist in CLRI. He has done plenty of works on the synthetic organic chemistry. And he has obtained his BSc from Tamil Nadu as a government arts college. Subsequently, he did the PG in University of Madras, and then he did his doctorate in synthetic organic chemistry from the University of Madras. And then he moved to USA for a postdoctoral fellowship. Subsequently, he also did a postdoctoral fellowship in Japan. Then he came back to CSER and joined as a scientist. And he spent quite a number of years in CLRI, and he's a really great scientist with us today. He will talk about his achievements in these years. Thank you very much, sir.
Thank you.
It is good afternoon. At the outset, I would like to thank the organizer, particularly Jayakumar and Suresh, for giving me this opportunity to give my research, in particular Bailey-Simon reaction, which I am going to deliberate. And I hope it will be difficult for me to give a lecture just before lunch. And also even just after lunch is also difficult. So for first 10 minutes, I would like to give my journey with the small synthetic methodology chemistry, what I have carried out both in NIST Trivandrum and in CLRI Chennai. And next 20 minutes, I will brief about the topic which I am covering today. So today's topic is the chemistry of arynes, very difficult to handle. Very few people are working due to its synthesis or the stability and the generation is very tough. For past 4 or 5 years, we were able to handle this reactive intermediate also for our synthetic transformation. I will brief about it for the synthesis of heterocyclic compound. As I told you, I just want to give you a brief introduction how I am inspired. The name reaction, which you are all aware of it, that the Morita-Bailey-Simon reaction, inspired by the Morita and Baylis. Later by Professor Basavia's manuscript, I was inspired this reaction, which is not coming under my postdoc, not my PhD, not my second postdoc, nothing. I just saw that this is a fascinating reaction with a full atom economic, and it has a lot of opportunity to make several synthetic methodology for the preparation of many heterocyclic compounds. So for the benefit of students sitting here back, I wish to introduce this reaction, which is well known in the literature, very well available in the Google, but still I would like to give. If you take any electrophile reacting with activated alkene in presence of a amine catalyst, tertiary amine catalyst, you will end up with a product with all inclusive, no byproduct, 100% atom economic. If you convert this electrophile instead of oxygen putting nitrogen, you will get the same product with aza version. There are lots of electrophiles are known, activated alkenes are known, amines are also known, non-amine catalysts are known. Any of these combinations for a new electrophile can also be utilized for a new reaction. In the review article written by Professor Basavaya, he concluded that this reaction has lot of avenues. Many electrophiles are unexplored. Many activated alkenes are still need to be explored. So I thought of I should use whatever the leftover or whatever the unexplored electrophiles and activated alkenes should be utilized for the synthesis of new small entities, which is actually starting synthons for many big synthetic protocols. So what I have used as unexplored electrophiles are, in my research journey, we have used pyridines, isatine, mostly, ferrocin, bipyridine, and also biphenyl. There are still some more are coming up. And we have, in my research, I have utilized all these electrophiles. And if you can see the activated alkenes also, there are standard activated alkenes. These two are the very famous activated alkenes known in the literature. But we have used several HDDA. These are all propargyl acrylate, where we have identified unusual reaction pattern, which I actually called, while reaction of this propargyl group goes away, and we call it as the labile activated alkene, and several activated alkenes were also used by my group. So when I joined Trivandrum, the research facility, like all young researcher, first generation researcher, it is very difficult to get chemicals, very difficult to get project, money availability. So that made me to start first. I want to demonstrate as a chemist first, then we will slowly improve. So my first starting material for my research independent is benzaldehyde, and borrowed methyl acrylate from the polymer chemistry department. Of course, for this purpose, I used my PhD work, radical cyclization, one of the last tool. So what I did was, this is Raja Singh, my first student, who is going to head a session here. What we did was we took benzaldehyde and treated with methyl acrylate, and we got this adduct. And we isomerized or propargylated with homopropargyl alcohol and also propargyl alcohol with lot of manipulations. This particularly with the terminal alkyne, which undergo readily radical cyclization. By introducing the chain length, you can think of constructing a 5-membered heterocycle, 6-member or 7-membered. This is the topic which we first demonstrated using a simplest Wallis-Inman adduct for the construction of oxygen heterocycle. So first student got over and simple molecule was over. Then I thought while I was studying a manuscript published from tetrahedral letters from a Brazil author, he was using isatin as an electrophile. And he has demonstrated that reaction has been used. I will come into the picture. But my journey, this from started, right now I am using this molecule for 2 decades now. You will see how we are exploiting this particular molecule for the synthesis of a lot of natural products. And this is the isatin. Why isatin? So if you look at the molecule isatin, you have a lot of avenues to functionalize this molecule. Starting material freely available in all rich, very cheap molecule. You can think of utilizing NH functionalization and C3 functionalization, as well as this aromatic position is available for any budding organic chemists who want to explore small molecule, cheap molecule, but for preparing a large number of heterocycles, you can think of using this particular molecule. This is the reason. As I already told you, this particular isatin molecule has been already used as an electrophile. And those authors, they used only, there are 2 activated alkene. They reported this as a electrophile, and Wallis-Inman adduct of isatin is well known. And this was published in 2002. I think I read this manuscript during 2003. After my return from Princeton postdoc, I read this paper. I got interested particularly this paper because if you see in the single molecule, NH, you can think utilizing this position, and you have OH, you have a double bond, you have a electron withdrawing group, and you can also manipulate. See, for an organic chemist, single molecule with multifunctional, you have a lot of avenues to synthetically modify this compound. And when I looked at the literature, I saw this is highly functionalized, diverse reactivity pattern is there. This molecule, after reporting in 2002 till 2005, nobody was touched up. They have, like me, I think all other authors, they read this is acinyl electrophile. One of the Wallis-Inman adduct reported from the isatin, that's all. So I thought, why don't I take this as a starting material for the synthetic transformation, which I am going to show. There are, I think there are 6 PhD students came out of starting this particular Wallis-Inman adduct, isatin. I will summarize those molecules. I will not take a lot of time. Now I will go a little bit faster. So why isatin? If you see the 3 position of this isatin has found in many natural products, which actually inspire with a lot of diverse biological activity, and a lot of biomolecules are known. And we thought we should use Wallis-Inman reaction or Wallis-Inman adduct of this isatin so as to construct this portion also. So utilizing, inspiring by these reactions, we have prepared the simply known molecule MBH adduct of isatin reported, and we initially converted this particular compound to its isomerized form of O-methoxide. You can easily do it by taking heat it with the trimethoxy orthoformate, you will get this molecule. And similarly, if you take this molecule, treat it with aqueous HBr, you will convert it to this bromo molecule. Using these 2 starting material, we have prepared an array of spiroxindole molecule. Actually, there are 3 PhD students who got PhD starting from these 2 molecules. I think 2 of my students are sitting here, those who have done this work, Vaidyanathan, Baby, and Chandrakumar. They were working initially in this project. So later, I moved to Trivandrum to Chennai. I continued my research on this topic. Solai Selvi is the student from Madurai Kamaraj University. She came to Chennai to do her PhD. And she was able to use the same molecule, Wallis-Inman adduct of isatin, but she was using the activated alkene is different, and she could make large number of isatin-based molecule entities, and she got very good publications, and I think she is right now working in Madurai, some colleges. So past 2-3 days, you might have heard a lot of cyclopropane chemistry. And we have also utilized this cystatin molecule to derivatize its cyclopropane. As you can see, we are able to prepare this cyclopropane, vinyl aziridine, vinyl cyclopropane, and we have developed synthetic methodology starting from Bayesian adduct of cystatin only, and we were able to prepare all these entities starting from Bayesian adduct of cystatin. I am just summarizing. I have no detail. If all the work is published, you can see. So you can see these are all the, again, the summary of the compound which we have prepared after reaching the Chennai. So we were not only interested preparing starting metal using Bayesian adduct of cystatin. Once we are exploring a lot of chemistry of Bayesian adduct, so we were interested to prepare cystatin as the starting material, and whether we can easily prepare not through Bayesian reaction but cystatin as a starting material. We were able to prepare large number of fluorescent molecules which have already been published. We were also achieved synthesizing large number of fluorescent molecules. The structures of those compounds were shown, and we were able to prepare the 4-member acetidine and myrcene and di-analog molecule. A large number of molecules were prepared by Ka2 coupling reactions. So later we were also continuing our work with 9-fluorinone, adopting the similar methodology what previously we were using. We were also able to prepare a large number of fluorescent molecules based on the fluorinone. In collaboration with the Madras University, Professor Harichandran, we were able to prepare some compounds, multi-component reaction, which are fluorescent in nature by this kind of 4H chromones. These compounds are coming under the other topic area. We were able to prepare fluorescence material. This is other work from the same group. We have collaborated preparing this complex molecule. Actually, our interest utilizing new electrophiles were very interested. If you can see the bipyridine as the problem in inorganic chemistry because of the solubility, we thought while introducing a functional group here to make bipyridine as a more soluble material. So we thought we will buy the dimethyl compound, convert to oxidizing corresponding monoaldehyde and dialdehyde. We were able to get its Bayesian adduct simply bipyridine alkyl. First time we reported. And after derivatizing this molecule has become very easily soluble. And you can also think of cyclizing here to make this macrocycle molecule having the bipyridine. So the next one is we were interested to use unusual activated alkenes. These are all the activated alkenes known. You can buy it from Alrich, but no one was used in the Bayesian adduct preparation. Between these two combinations, aldehyde pyridine 2, 3, 4, as well as this unusual activated alkenes, we were able to prepare a large number of new entity of new MBH adducts which are not known in the literature. So Malini also used, actually she was my MSc classmate, later she joined, but she could be a good worker. And she used the unusual activated alkene preparing Bayesian adduct from the cystatin. This is that compound. While we are using this isomethanol 3+2 cyclohydrogen reaction, she was able to get this macrocycle molecule in three steps. And while reduction, we were able to prepare this acid core, which has been found in the natural product. Actually, for any budding scientists who wanted to use a simple chemical available for like not in the institutes, bigger institute, but universities and small college, they don't have ability to buy many compounds, many costly chemicals. This is the boy who came from VIT to work with me for 1 year, and he wanted to do PhD. This boy has utilized anthralenamide, very commonly available chemical. So using this as a starting material, this boy able to prepare by synthetic methodology biphenyl and 7-member ring, and also this bipyrrole rings and this pyro compound. He got his PhD, and I showed him that he is working now in a big company in a big way. But this boy learned how even a small molecule available in the laboratory can be utilized for the construction of the bigger or diverse molecular entities. So this is the current work collaboration with VIT Vellore, Dr. Kannadasan. This work is going to deliver in the afternoon in detail. but I am collaborating with him using starting from isartan is well known, indole is also well known, but if you see another nitrogen in the air ring, which is azaisartan, very difficult to handle, not like indole, solubility is the big problem, derivatization is problem, handling is problem, but its bioactivity is very high. It is well known in a lot of azaisartan molecule are well known for in the cancer therapy studies. So we wanted to do some work on this azaisartan. The details will be given in the afternoon, but this is currently going on. Very good publications are coming up. So another area of work which I am doing in CLRA is the propargyl algal system. If you take a propargyl algal attached to fluorinone, because we have already 9 fluorine we had, this can be prepared. If you use Lewis acid, this may generate a propargyl cation, which is in equilibrium with an allynyl cation. So these two cations, any nucleophile, if you properly attack, you will get a diverse functionalized product. So what we did, we prepared this propargyl algal, we generated Lewis acid BF3 ethylate, and depending upon these two reactivity, either it may be allynyl cation or propargyl cation, we were able to prepare this anthralenamide, which we already had, and nitrile oxide, substituted anthralenamide, nitrile oxide, as well as isatinamine, because these all nucleophiles are available with us. For one project or other project, these materials were available with us. So we were able to prepare a large number of 9 fluorinone appended heterocyclic compound in one-step manner only. So coming another topic where we were working on reagent chemistry, a candidate who did this work is sitting here. So we were able to prepare, if you take DMSO HBr, which is known as a reagent for bromination. Accidentally, when we did this experiment with a secondary amine, aryl secondary amine, instead of brominating agent, we have found that it will be an allylating as well as brominating reagent. So we have developed a new reagent pathway that allyl bromide and DMSO is a alkylating reagent. So this has been demonstrated, synthetically transformed, lot of things were there. This is the chemistry of benzene, a part of lecture coming up later. And we have also utilized this propargylamine using this pyridine hydrazine, and as well as the azaisartan as the nucleophile, we were able to derivatize these two functionalized molecule. So here I am summarized my work, what we have done so far, not fully, and the major work we have summarized. And coming to the today's work, so in when we are listening in a conference like this, I was surprised to see why benzenes were less used when Professor Biju was giving a lecture at Trivandrum. So he was giving a lecture on arynes, benzene. So I thought we have Bayes-Inman chemistry. When we go through the literature, no chemistry was reported using benzene. Then I thought, why not I can utilize those benzene to our Bayes-Inman chemistry so as to get a very deep synthesis of very difficult structurally different molecules. So forget about this is the background about the how benzenes were evaluated, and this due to shortage of time I am skipping this. But if you see why people were not working a lot or were not utilizing arynes is the question. When I was MSc student, my teacher taught generating benzene mechanism, aromatic nucleophilic substitution, very difficult. You need a lot of heavy butyl lithium and all reagent. You should go for very low temperature because of the high reactivity. These are all the difficulties made people afraid of utilizing benzene. But there are lots of methods now available for generation of benzene. Particularly this method using cesium fluoride, and this is the material now available. You can generate this benzene at room temperature in 5 minutes, 2-3 minutes. Yes, 1 minute. The reaction will also complete in 5 minutes or so. So that is the actually advantage of now the people are ready to use. It's a little costly, but you can use it very little amount for converting, using it as a reagent. what you need is 100 milligram or 150 milligram benzene generation, you can generate your benzene and you can convert for any reaction. The moment when you are generating benzene, but you want to know the basic chemistry, you can utilize this compound for the 4+2 cycloaddition, 2+2 cycloaddition, dipolar cycloaddition. You can use a nucleophile or electrophile, accordingly it will add, and it can be used as an insertion reaction across, and you can also use metal carbons for transition catalyzed mechanism. And this shows that the potential application of benzene in various synthetic organic chemistry transformations. So this I have already told. So having that in mind, benzenes are known and a lot of other chemistry are known. Now we wanted to use the benzene. It's my time, sir. So we generated benzene, and Bayes-Inman adduct of isatin is already known, very well known from our group. And we thought, why I cannot use this double bond using this alkyne double bond as 2+2 cycloaddition, because creation of 4-membered ring across this pyroxenol position is difficult. So actually initially that was the idea, and also we thought shift the double bond inside by isomerization, again use this so that the product what you are getting may be different. These were the original idea. The construction of 4-membered pyro position at 3 position of isatin was our idea. Yes, we started the reaction. So we prepared Bayes-Inman adduct with H, and when we added this benzene, it undergo retro reaction. We got isatin back and no reaction was occurred. We prepared Bayes-Inman adduct, generated benzene, added, instead of getting any product, we got isatin back. The adduct was cleaved. So we thought, since free OH is there, the adduct got cleaved, and we methylated this OH group. Then when we are doing this reaction, benzene did not add to this product. We isolate the starting material. Without with OH, we got isatin. Protected OH, we got starting metal back, no reaction. Our first attempt with benzene, buying all the costly chemical, doing first time and all was failed. Then we were, why it is not adding was the big question. Then what we did was, okay, let us isomerize into internally and see whether this 2+2 cycloaddition, this product is forming, and we explored the reaction. This time, reaction occurred, but we did not get what the product we want, but instead we got a interesting this pyro compound as well as C insertion, aryl C insertion compound, which we found later. Quickly I will. So those compounds were actually, this is my first slide. I will rest 5 minutes. So these are all the actual the benzene cycloaddition product, insertion product, and also dealkylated product. See, we have optimized, we have devised a mechanism for why we are getting the, instead of 2+2 product, why we are getting 3+2 product, as well as the insertion product were assigned. And a lot of examples were, and we have synthetically transformed the product what we have achieved. And I just want to summarize each work what I am planning to give in one slide. So the next objective was, if you take benzene, we have anthralamide with us. We can also position with anthralamide ortho, meta, para. And if you add the benzene, what will happen? Since the product you are getting maybe a triphenyl amine product. So we got this diphenyl substituted as well as triphenyl substituted triphenylamine. The interesting point is you can position this amide position meta or para. This will alter the property of this molecule. So we have made actually a large number of anthralamide or amino amides and a large number of alkynes, and a diverse product were obtained. We have a lot of synthetic transformation also we have made. So we have studied a lot of photophysical properties also. And one more interesting is azomethaneamines. So azomethaneamine, since we had isatin with us, we can prepare the azomethaneamine dipole. And what will happen if I have dipole here and add benzene generated here, so that the product may be highly interesting. So we have carried out this reaction. So with this diphenyl azomethylamine and this particular alkyne, we will be able to prepare this C insertion product. And once we are substituted here, instead of this product, we observed two other products here. This pyro product with stereogenic center, two different stereoisomer, with we assigned the structure based on the crystal. So mechanism, example, synthetic transformation. And the last work was, still we are working on isatin. Isatin is our lifeline in our laboratory. So we thought, what will happen if I take isatin-based sulfonamide, and when we added this particular molecule, since both the substrates are very highly reactive, instead of getting only one product or two products using alkyne, here in this case we have sent three products. So you can see this. One is the NS bond cleaved product, other one is N-arylated product, other one is elimination of this, and you will get N2 product. N2 react with again benzene. So we were able to get these three products. All these three are, we were trying to control or optimize this reaction, whether we can selectively prepare one molecule which failed. But all these three compounds were able to isolate the compound. Yeah, we got crystal. And I think I have explored a lot of arraign generated from a corresponding precursor, used our isatin starting material for the synthesis of a large number of heterocyclic compound. I think with a very short span of time, I was unable to give a detailed lecture, but I believe I have given the glimpses of the work what we have carried out in isatin as well as in the other allied area. Thank you very much.
Yes, it is. So let us please join with me to thank the speaker, Professor P. Shanmugam, for his wonderful lecture. He's a really legend in the field of organic synthesis. I can spend more than given hours, but time is restricted. I'm sorry, sir.
No problem.
Yeah, please accept our.
No problem.
Thank you, sir. A token of love. Yes, sir. Yes, there is. Yes. So, just a few minutes. Yes, the next speaker is Professor Ramesh Rasappan. He is Associate Professor from IIScr. Basically, he has done plenty of postdoctoral work abroad. Especially, he worked with Professor Correa, and it's very difficult to work with Professor Correa and his lab. And so I think his experience is really a great thing for us. So he will deliver the lecture on mostly on the ligands like bis oxysolines and their applications. I hope the interesting talk is waiting for you. Thank you.
Yeah, I hope I'm audible. So without wasting time, so I will just go ahead with my presentation. Before I begin, I definitely thank Shiva and Suresh for giving me this opportunity. I'm extremely happy to be here and to interact with you all. So our group in IISc Trivandrum, we work in the area of cross-coupling reactions. If you look at in this slide, so the majority of our reactions, they fall under the category of organosilane synthesis and its application in cross-coupling reaction. That's the major area that we focus, organosilanes. So during the course of this synthesis and cross-coupling reactions, we also introduced a silyl reagents. You can see that we have introduced our own silyl zinc reagent. We also work in different areas, for example, dual catalysis, in which we combine photocatalyst and nickel catalysis. So that's the second area. We also work on cross electrophile coupling reactions, which I will talk about a bit later. When you talk about organosilanes, how do you synthesize them? So you can introduce silicon moiety in general, 3 different ways. One is the hydrosilylation, which you can see that's the hydrosilylation, and also you can take some electrophilic silylating reagent like halosilanes, as well as you can also take an silyl nucleophile, for example, silyl magnesium or silyl lithium or silyl zinc reagents. So those are the 3 common ways that you can introduce a silicon moiety into an organic molecule. Recent years, people also using the silaboranes, which is called Zuginam's reagent, which has been routinely used in the cross-coupling reactions. So we had interested to look into the use of silaborane reagents in cross-coupling reactions. But then if you look at the synthesis of the silaborane, the modern reagent, so which actually starts with the silyl lithium. So you take a silyl lithium and you treat with the isopropoxybipene and you get the silaborane. Again, the synthesis is limited because you have to have something like a phenyl group on the silicon atom. So if you do not have such a phenyl or arene moiety on the silicon, then you have to follow a complex procedure. Then again, it's this alkyl silanes, this kind of silaborane reagents are really sensitive, and it's said to be explored in cross-coupling reactions. Now, these are about the Zuginam's reagents. So our goal was simple. So if you look at these reagents, you have to use them in the reaction. In situ, it gets into silyl metallating species. So you start with this one, you get into silyl copper or silyl nickel or any other silyl metallic reagent. So in situ, it gets transformed in the reaction. So our goal was to start with something like a silyl halide or silyl lithium. In situ, you convert into less reactive silyl copper or silyl magnesium or silyl zinc, so that we can bypass the requirement of such reagent, but then yet you will have an active silylating reagent. So that was our goal. So we have done preliminary studies, and then we have achieved the synthesis of silyl magnesium reagent in 2 different ways. One is the typical way of treating a silyl lithium, you exchange with the magnesium. The other way is to directly insert the magnesium into silyl iodide bond. So that works very well. And then we have used them in the cross-coupling reactions. You can see that this is the CO bond cleavage, and via CO bond cleavage, you introduce the silicon moiety. And the other way, we also did the NNCO-specific cross-coupling reactions. So again, this is a CO bond cleavage, but then you start with NNCO pure, and then you end up in a NNCO pure product. So that's the NNCO-specific reaction. Fine. So we have also carried out some mechanistic studies. So if you look at carefully the last reaction that I was talking about, this is an specific reaction in which we add substoichiometric amount of silver fluoride. So the role of silver fluoride is kind of crucial in this reaction. Some of the substrate, they do not require any silver fluoride, and some substrate they do need substoichiometric amount of silver fluoride. So what is the role of this one? So we were puzzled with all the possible questions. It doesn't do any of the typical jobs. So the typical role of silver fluoride is to free up the coordination site, or you form the silver precipitate so that you can drive the reaction further. It doesn't do any of those typical job. But what we found is that if you do not have a silver fluoride in the medium, so you generate a nickel 0 species in the medium. So you start with the nickel 2 complex in the course of the reaction. So via reductive elimination, you generate a low valent nickel 0 species. So this nickel 0 species becomes unstable. It turns out to be nickel black if you do not have a silver fluoride in the medium. So we propose that there is a high hetero-bimetallic complex formation in the medium. That's why it stabilizes your low valent nickel species. Thereby the reaction proceeds further. So this is the continuation of our work. So we want to synthesize acylsilane for the purpose of utilizing them in various cross-coupling reactions. So the acylsilanes are excellent, starting material for doing variety of reactions. You can see them. You can do the Brook rearrangement followed by cross-coupling reaction. There are quite number of reactions which you can do with the acylsilanes. But if you look at the synthesis of acylsilane, it's not like a simple ketone or acyl derivatives that you can synthesize them. So it's quite hard. It's not straightforward method. If you look at the the recent publications, how do they make this acylsilane? You have to start from the Umpolang chemistry. So you take an aldehyde and then you just do the diethane protection and then do the Umpolang reaction to synthesize such a simple acylsilane. You cannot do typical acid chloride reaction. You cannot take acid chloride and treat with the silyl zinc. That's not going to work. You get the poor yield. Okay, so the reason we we thought that it could be the dissolved lithium salt comes from the silane lithium. So that may be the reason that you are getting a poor yield. So we want to synthesize the silane zinc reagent, which is devoid of any of this dissolved lithium salts. So we managed, based on our previous experience, we managed to directly insert the zinc into this silicon iodide bond, and then we could generate all these different type of silane zinc reagents, which is fortunately solid in nature. So we can get the crystal structure, and then now this silane zinc reagents are equally efficient as the silane zinc reagents which is prepared from silane lithium. So after having these ligands, we prepared the acyl silane starting from the corresponding acid chloride. So we convert the carboxylic acid into acid chloride in situ with oxalyl chloride, and then you do the subsequent reaction to generate the acyl silane. So that's the one method. And then we also explored in a various transformations. We taken our reagents, and then we have subjected literature known reactions. For example, the allylation. So you can see the allylic alcohol, allylic chloride. You employ our own reagent, and then it gives the excellent product. I mean, decent product. So all our conditions, these all are unoptimized. So we have just taken the literature condition and we applied our own reagent, and then we got a moderate yield in all these transformation. You can see that the simple cross-coupling reaction or the conjugate addition. So we can in principle use our own reagent rather than the complex setup which is shown here. Now, so taking the reagent further, we have also carried out a dedicated study. So you can see that we have done the silylation on aryl halides as well as alkyl halides. So we can silylate the arenes as well as alkyl halides. So the selected substrate scopes are shown here. So we have also shown some of the applications. We have taken the product aryl silanes, and then we have shown some of the applications, which is again, these are the known reaction conditions. We just showed that it can be useful starting material. Now again, going to another topic of silicon chemistry. So if you look at the vinyl silanes, they are quite interesting. When you have a highly substituted vinyl silanes that can be used in various reactions or organic transformations, it's more of most of them comes by the alpha beta effect. We all know that alpha anions are stabilized as well as the beta carbocations. So based on that principle, so we can do a lot of chemistry with the vinyl silanes. But then when you talk about the synthesis of vinyl silane, then again you have a limitation. So if you talk about hydrosylations again, so hydrosylation you can do for the simple, you can take a simple terminal alkyne and then you can do the hydrosylation, but then you cannot use this strategy for making a highly substituted vinyl silanes. Especially you cannot introduce a trimethylsilane because trimethylsilane is a gas, so you cannot use them. And then there is another strategy which involves the silyl metallation. So the silyl metallations are highly important because it's highly atom economic, so you don't lose anything. So you can do the silyl metallation, but again there is an issue is the selectivity. So in this case of hydrosylation, it's the syn selective. So you just add on the same phase. So the silyl metallation also occurs through the syn fashion. So you get both the cases, you get the syn selective product, but then again the highly substituted products are a little bit challenging here. So apart from that, if you want to get an anti-selective vinyl silane, so the one way to form an anti-selective vinyl silane is to do the radical reactions. But then if you talk about the radical silane reactions, which are quite limited because you have to have something like a kind of stable silane radical, and that can give you anti-selective products. So these are the literature known protocols. And then what we found is that using our own reagents, so we can take an alkyne, and it's a multi-component reaction. You can take an alkyne, alkyl halide, and then our own reagent. Now you can get an exclusively anti-selective product. So it's a 3-component reaction. So I'm skipping off the scope. Now, how does the reaction happens? So again, you can see that in the typical way, you start with the nickel 2 and you will end up with a low valent nickel 0. That's the typical reaction. So it goes through the transmetallation and reductive elimination. So what is happening in our case, this is not the typical pathway. So what happens is that you have the nickel 2, but then it undergoes the pi insertion and then the reductive elimination to give this byproducts. So that's the pathway to make the active nickel 1 species. And you have the conformation to get the nickel 1 species. Right. So the other study that we have done is the crossover experiment. We were thinking that it may be the alkyne, that alkyne forms a kind of terminal silane, such an intermediate alkynyl silane, that could act as an intermediate in the reaction. So to disprove that one, we have done the crossover experiment that clearly says that we do not have such an intermediate in the reaction. So this is the mechanism that we have proposed. So you have the active nickel 1 species. The first step is the pi insertion. You can see that it adds across the alkyne. So you can see that there obviously there will be two different geometrical, the regioisomers are possible. So out of these two, we all know that when you have a arene group, so usually you prefer the formation of this alpha branched nickel species because you have this eta 3 complex with your arene. But then what we also found is that even if you do not have an aryl group, if you have an alkyl group where you do not form this eta 3 complex, but still it prefers to form such a regioisomer. This is because of the the CH on the alkyne, on the alkyl group as well as the nickel. There is an agnostic interaction, so which favors the formation of this regioisomer over the other one. Good. So now we have this intermediate here, but then this is a syn adduct. Okay, what we get, our product is the anti-adduct. So how do we get the anti-adduct starting from the syn adduct? So there is we have done a calculation of this entire mechanistic proposal with the help of Professor Lam. So there is an equilibrium exists between the syn adduct and then anti-adduct. The energy between the energy difference between these two adducts are very, very small. It's a negligible amount. So that means they are in equilibrium. So the subsequent step is the 1 electron reduction from the alkyl bromide and the nickel 1 complex. Now this step, so the reduction of alkyl bromide by the nickel 1 can happen either with your trans adduct or syn adduct. Either way it is possible. But what is happening is that, so that anti-adduct here, so the anti-adduct and reduction of alkyl bromide by the anti-adduct, so energetically more favorable than the syn adduct. So the syn adduct, if it reduces, it requires a little bit more energy. So this is the step that determines your selectivity. So the stereo selectivity is determined at this step. So now you have the radical recombination to form such an intermediate, and then the reactive elimination will give you the final products. So we have done many other studies, for example, the radical clock experiments to prove that this is a radical, you are forming an alkyl radical in the medium. So that's, yeah, so now it's almost accepted, it's accepted in anatomy. Now I will move on to the cross electrophile coupling reaction. So the cross electrophile coupling reactions are, very simple in concept. So typical reaction involves you take an aryl halide, for example, and then you have an organometallic reagent like a silyl magnesium or organomagnesium or organozinc or even boron reagent. So you have an electrophile and a nucleophile. That's the typical cross coupling reaction. So in case of cross electrophile, what you do is you take two different electrophiles and then you couple them together. So that's the cross electrophile coupling, and mostly it is developed by Weiss from USA. So what we were aiming is that we want to synthesize a ketone and starting from a carboxylic acid and then the primary amine, which is quite challenging because both of them obviously doesn't react. So our goal was to convert the amine into pyridinium salt. At the same time, you convert the acid into either acid chloride or anhydride. Now we can do a cross electrophile coupling reaction. Okay, so this is what we have done. So I will skip off the scope. So you can see that either you take a acid chloride and then convert the primary amine into in situ pyridinium salt, and then you can do the coupling reaction. So we have even taken a free carboxylic acid. So in situ you convert into anhydride, but then in that case we get a poor yield, but then you can see that the functional group compatibility is very high. You can see that N-Bock, even MOM groups all tolerated, but then the yield is poor in this case, but the rest of them are very high yielding. Right, the mechanism of this reaction is again puzzling us. So the mechanism usually starts with the nickel 2. So you have the nickel 2 in the medium, so you have a stoichiometric reductant, and that one reduces your nickel 2 low valence species, either it is a nickel 0 or 1. So when you have low valence species, what the first step could be either your alkyl pyridinium salt gets reduced or your oxidative addition happens with your acid chloride. So it can go in two different ways. So if this happens, then what you generate is the alkyl radical species, and then you have a reduced nickel species, and which will undergo oxidative addition to generate the acyl intermediate. And now the radical recombination will lead to such a nickel 3 adduct. Now that will undergo reductive elimination, and then will give the product as well as nickel 1 species, which will be reduced by the reductant present in the medium to regenerate the active spaces. So that's the radical chain process. The other process is the sequential reduction. Okay, so I will skip it off, to explain every details due to the time limitation. Now you see this slide, what you see is that this is what we originally proposed, that there are two different pathways possible, but what we have identified in the recent work, so we have done a dedicated mechanism study to identify out of these two, which one is actually active. So what we found is that the radical chain process, this is what active in the medium. So I will not go into the detail of every step that how we identified this is what happening in our reaction. But what I can say here is that, so typically when you start with the nickel 2 species, when you have a reductant like a manganese or zinc, so based on the redox potential, so if you talk about the manganese, which is having higher oxidation potential, and then people expect that the nickel 2 will become a nickel 0. But what we found experimentally is that you generate a nickel 1 species rather than the nickel 0, right? So that's what we found. I will skip it off this portion. So now again I will, okay, so what we found is then again, so we have applied our chemistry in the synthesis of various vinyl silanes. For example, you can see that in the literature, for example, if you want to make a not alkyl silane, this is alkoxysilane. So it's difficult to make such a simple molecule. So what we found is that we can take a vinyl halide and then you can do the cross coupling to get the vinyl silane. Okay, so that's what we have. Yeah. Right, I pressed the wrong button because of the hurriness. Can you please switch over? No, yeah, okay, good. Right, so what we have done, okay, so this is the reaction we have done. So you have vinyl bromide and then the alkyl halide. We can do the cross electrophile coupling to generate this expected product. So the mechanism is again, some of our findings are really interesting. So what happens is that you have a vinyl bromide in the medium. So in the reaction, so the vinyl bromide gets converted into vinyl iodide and that reacts further to give the product. So that's what, okay, that's one finding that we made. The second interesting finding is that, so you have this alkyl radical generation and then there are two mechanistic pathways. So we have to prove that this is a radical chain mechanism is the operative. So if you look at this chemistry here, the radical chain mechanism, you can have the ring closed product and ring opened product. So what we do is we find out the ratio of the ring opened and ring cyclized product. So if you find out the ratio, we can find out whether the mechanism follows either the cage bound or the escape rebound mechanism. So it can operate in a two different ways. So if you can plot the ratio, you can see that here. So upon increasing the loading of your nickel catalyst, you are gradually increasing the ratio of uncycled products. So that clearly says that you have a escape rebound mechanism is operative. So that indirectly supports the radical chain mechanism over the sequel reduction. So that sorted out which mechanism operative. I will be skipping off all this one. So I will quickly talk about a couple of slides. So it's 28. So I will be finishing it off quickly. Right. So the other way that the other project what we are working on is that, for example, we have talked about pyridine salt, how we used them to cross couple carboxylic acid. So at the same time, we want to avoid the use of stoichiometric manganese reductant. So we replace the stoichiometric reductant with the photocatalyst, and then we can perform such a reaction. For example, in this case, instead of carboxylic acid, we have taken aldehyde. You generate a ketone by coupling an aldehyde with the primary amine. So you can see that we could generate such a ketone in a very efficient way, combining the photocatalyst and nickel catalyst. So I will not go through again the mechanism. It is again beyond the interest of time. So the one last slide I want to talk about is this is our recent finding. So you have this aldehyde and then the alkyne. So which again we can couple using this concept dual catalysis. You generate an acyl radical and add to the alkyne. So that's again our recent finding using this new concept. And this one is a very interesting concept because we have a 3 catalytic cycle present in there. So one catalytic, this is a photocatalytic cycle and this is a nickel catalytic cycle, and you have isomerization happening here. So we generate in situ, we generate the catalytic HCl, and that catalyzes the isomerization of phenol. So Yeah, so that again I have to skip it because of the time. This is again the last slide. Okay, so this is about the sp3 coupling. So when you talk about sp3 coupling, it's quite challenging because you will have a lot of byproducts in the medium. For example, if you want to take an alkyl halide and organometallic reagents, so you will have beta hydrogen elimination, reduction, dimerization. So sp3 is always challenging. So the recent report, you can notably you can look at the Macmillan's work. So that where they take this furan derivative, pyran derivatives, you have heteroatoms next to the CH2, and then you can do such a cross coupling reaction. So what we have done is that without having any heteroatoms, so if you simply take a cyclohexane or cyclopentane or N-hexane or N-alkyl chains, which you can in principle activate via HEAT process, and you can generate the alkyl radical, and then we can cross couple. So this one has not been achieved, and the first time we could able to couple such a fragment. So you can see here the cyclohexane, and then you have alkyl bromide. So most of the cases the benzyl bromide or primary alkyl halide, but we can successfully do this kind of sp3 coupling from the unactivated system. So yeah, we have done that sp3 coupling. We have also extended to the formation of E-alkenes. So this is my, obviously, the work all done by the students. Obviously, I'm just representative of their work. So I have currently 4 students in my group. So we are a very small group. There are 5 students already completed in my group. And definitely I thank the funding agency, SCRB, CSIR, as well as IISc. And thank you for kind attention.
Thank you very much, Professor. Professor Asapa did a really wonderful presentation. Because we are running out of time, I'm sorry for stopping the question time. So, sorry, sir.
No problem.
Please accept our thanks by accepting this small gift.
I thank Professor Selvaparam for his wonderful chair and is maintaining the time also. Students, because of you, 1 hour we are delay. Okay, so the lunch time we are cut half an hour, only half an hour for lunch. So 2 o'clock you should be here. Okay, so otherwise other Horiba presentation is there. Other very interesting. Otherwise it will go 8 o'clock. Okay, so maintain the time. Tea time only 10 to 15 minutes. 10 minutes you maintain. Immediately you should assemble. You should not expect somebody will come and tell you. You should come before immediately. Okay. So just half an hour you assemble. 1:55 you should be here. Okay. Okay. Okay.
ఇది ఓ బ్యాగ్ లో వచ్చింది.
Sir, start பண்ணிடலாமா சார்? போயிடலாம்ல? Okay, good.
We are waiting.
Yeah. Okay, after lunch, there's a very, we are going to listen very fantastic talk. My friend P.C. Ravikumar is going to deliver, he's going to give a magical, compounds, impress you. And you have a lot of credits. The most important one is, he's a student of Professor Sri Krishna, IISc Bangalore. So that is a biggest thing. And moreover, he's he is from National Institute of Science, Education and Research, NISER, Bhubaneswar. PhD from Professor Sri Krishna, IISc Bangalore. And recently, he was recognized as a pioneering investigator by RSC journal, ChemCom. The Chemical Research Society of India, CRSI, awarded bronze medal in 2023. Very currently, he received last year. And then he ranked as a top 3% high cited ACS author from India. The year is 2019 to 20. And also he received Excellence Award as well as Foundation Awards for Institute Service while serving as IIT Mohali. He is a member editorial advisory board, the Journal of Organic Chemistry. Member editorial board, advisory board, ACS Catalysis. See, the co-convener of a Chemical Research Society of India and a local chapter Bhubaneswar. With these few words, I invite Professor Ravikumar to deliver his talk. Yeah, thank you, please.
Thank you, Sivakumar, for the kind introduction. I also thank you for the kind invitation and the generous introduction. I will quickly go through the work that we have been doing on CH bond functionalizations using cobalt catalyst. This is the schematic general representation of a CH bond activation reaction where the metal first coordinates with the directing group and activates a proximal CH bond, which then reacts with different functional groups to get the functionalized product. CH bond functionalization process generally goes by two steps. First is a CH bond activation process and then CH bond functionalization process. In this reaction, the key thing is this directing group. We need to have this directing group to activate this proximal CH bond. And in general, directing groups can be classified into two categories. One is strongly coordinating directing group and the other one is weakly coordinating directing group. Usually the strongly coordinating directing groups are nitrogen containing directing groups and the weakly coordinating directing groups are oxygen containing directing groups. In the next few slides, I will basically compare the differences between the two and what are the advantages and disadvantages of using either of these directing groups. And these are the strongly coordinating directing groups. As you can see, they are large groups and they have to be connected through CC or CN bonds. These are very strong bonds. So firstly, we have to install this group on the substrate. You can see here, this is a substrate with a CH bond. This has been installed on the substrate, which requires additional step. Then it reacts with metal and activates the CH bond and it does the functionalization. So the problem with CH activation using strongly coordinating directing group is that we have to first install the directing group. And another problem is that after the functionalization process is done, this large group stays intact with the substrate, which we don't want oftentimes, unless if our target molecule also requires this pyridine to be present, this is not a useful group. And so we can say that the very application, this largely restricts the very application of CH bond. On the one hand, we say that we directly functionalize a CH bond. On the other hand, we are left with this large group, which we don't want. So this particular drawback actually limits the broad applicability of CH activation by using strongly coordinating directing groups. Now I'll talk about CH activation by using weakly coordinating directing groups. As I said before, Oxygen containing groups like acid, ester, ketone, aldehydes, these are considered as weakly coordinating directing groups. And these groups are present in many organic molecules. These are very common functional groups in many organic reactions. And they are often present in many commercially available substrates. We can just buy them and directly use them. You don't need to add one step. Another advantage is after the functionalization process is done, we can actually use this functionality as a handle for further functionalization. So in fact, what we can say is that CH activation using weakly coordinating directing group has broader application as compared to strongly coordinating directing groups. So the bottom line that I would like to say is that weakly coordinating directing groups are much better in terms of sustainability and broader application. So another important point that I would like to discuss here is the cost factor and the toxicity of the metals that is being used in CH activation. Invariably, we have to use transition metals for activating a CH bond. The majority of the CH activations are done by using heavy metals like palladium, rhodium, ruthenium, iridium. These heavy metals are highly toxic and they are often very costly. So it's very difficult to offer them. If we can replace these heavy metals by using a first row transition metal such as cobalt, which is very cheap, you can see in this graph, this is the cost of cobalt, and you can see the other metals are very, very expensive. It's not only cheap, it is less toxic, and also it is very abundant in the earth crust. So if we can do CH activation by using a first row metal such as cobalt, it is more sustainable. So depending on the type of directing groups that we use and the type of metal that we use, we can classify CH bond activation into 4 different categories. The least sustainable category is this one, where we use a very strongly coordinating directing group and 4D or 5D metals, heavy metals. This is the least sustainable CH bond activation. Slightly better CH activation would be to use weakly coordinating directing group, and we still use heavy metals. Even better would be to use 3D metal because it's less expensive and less toxic. We are allowed to use a little bit stronger coordinating group. The best CH activation would be to use a 3D metal which is less expensive and less toxic and a weakly coordinating directing groups. So this is a concept which we have been working on for quite some time now, and recently we have covered a review on this particular topic in chemical communication. If you are interested, you can go through this. And now I'll come to, this is the background of my work. Now I'll come to the work that we have been doing. Indole is a heterocycle which is very prevalent in many natural products and drug molecules. Functionalization of indole is considered very important both from medicinal chemistry point of view as well as in synthesis point of view. Generally, C2 and C3 functionalization of indole is very well explored by using classical chemistry. Whereas the C4 and C7, C6 bond functionalizations have not been much explored, which we can address by using CH bond activation strategy. In 2019, we have published a paper using this concept, first row transition metal and a weakly coordinating directing group. We have done the functionalization at the C4 bond. So we thought we will extend this work, and instead of using acrylates, we will use alkynes to get the olefinated product here. But to our surprise, instead of olefination product, we got this acylene product. So we thought this is also useful, so we wanted to see how this works. Initially we got less yield, so we started optimizing this condition, and we got a good condition by which we could get this acylene product. I'll explain in detail when I talk about the mechanism, where do we get this oxygen from. So we basically optimize the condition, and we varied different substrates, different end protection and different substituents on the aromatic ring. In all cases the yields were good. So then we thought that instead of using diaryl alkyne, we will use alkyl aryl alkyne, and in that case also the reaction is working well. So normally when we actually try to develop a methodology, we also wanted to study the mechanism, and since this reaction is unusual, in this case, of course, I forgot to say that instead of acyloin, the hydroxy gets eliminated with this alkyl aryl alkyne, and we get this enone product. Generally, if we discover any new reaction, we are supposed to give the mechanism for it. So we do a lot of extensive mechanistic studies. So one of the things that we do is that we carefully analyze the side products formed in the reaction, because oftentimes we get the clue how this reaction pathway is going by looking at the side products. So if this is the TLC of this reaction, this is the starting material, and this is the alkyne, other starting material. So you can clearly see there are two products. This is the acylene product, and we isolated this product and we found that this is the monoketone. If you compare this monoketone with this coupling partner, you can clearly see that a water molecule gets added to this alkyne, which becomes enol, and that's the reason why we get this ketone. So that means there is some water in the reaction. So initially we thought maybe there is some moisture in the solvent. So we dried several times. Every time we do it, we get consistently this product. So that means the water is coming through some other way. I'll explain that in a little while. So then we thought that, okay, we will deliberately add water, O18 labeled water, and see what happens. So when we deliberately added 5 equivalents of O18 labeled water, we could see that O18 gets incorporated in the product as well as in this monoketone. This clearly proves that water is also one of the reactant in this reaction, but we don't know how it forms. So I'll explain how it forms in the next few slides. So this is the HRMS data, and from this we clearly confirm that O18 is going into the product. So we took the standard condition and we started looking at the GCMS spectrum, and we found that there is a peak which corresponds to this diethyl ether. So this can come from this trifluoroethanol solvent. So we think that 2 molecules of this alcohol reacts in the presence of AgBF4, gives this ether, so which indirectly proves that the water is being generated in the solution. But this is not a good enough proof, because GCMS will detect even smaller, very small quantities of ether. So we cannot clearly confirm. It may be an impurity from the solvent, so the referees are not going to accept it. So we thought we'll have to quantify it. Because the product has 2 equivalents of water in it. So for quantification, we resorted to quantitative F19 NMR study, because generally F19 NMR study is not done for quantitative purposes. So we took the reference from this paper and we adopted that procedure by using NMR experts. So we quantified, we took this fluorine tagged substrate and then did this reaction, and we got this fluorine tagged product, and we also detected this ether formation. And since this has fluorine, by comparing the relative ratio between them, we can say that this many equivalents of water is formed. So that's what we did. So this is the spectra. You can see here, this is the pure ether which we purchased commercially, and we checked the NMR. This is the peak, and this is the trifluoroethanol that we used for the reaction. So you can see there is no ether peak here. And then we did the reaction with this fluorine tagged substrate, and we got the product. This is the product peak fluorine corresponding to this, and this is the ether peak. We integrated that, and we found that nearly 4 to 5 equivalents of ether is being formed, but whereas we need 2 equivalents only for the product. So this clearly proves quantitatively that water is generated in situ. Up to this, we have proved that water is being generated in the solution, but we still don't know what is the actual coupling partner in the reaction. It may be this alkyne or this monoketone, or we also found the traces of this diketone. Initially, it made sense to us to believe that this diketone may be the actual product, actual coupling partner in the reaction. So we took the commercial benzene and added, instead of adding alkyne, we treated this with commercial benzene, and we could not find this product. That is also very surprising. We thought that it makes sense to us to believe that CH activation happens here and that adds here to get this one, but it didn't work. So what it means is that there is something else going on, so we really don't know what is going on at that time. Then we thought that we will do some application study. We were thinking that generally when you develop a methodology, you will have to do some applications. So we thought we will do some applications. We will remove this hydroxy and put a halogen there. So we were doing some application. So interestingly, every time when we treat this molecule, we were getting this dehydroxylated product. So we thought that maybe this reaction is going in a stepwise manner. So maybe this is an intermediate in the reaction. So we took this dehydroxylated product and then this is the dehydroxylated product, and then subjected to the standard condition. So when we subjected this to standard condition, we got the product, so which means that this is an intermediate in the reaction. So we have proved that this is an active intermediate in the reaction mixture. So then we removed the catalyst, we retained the other conditions, and in this case also this is working. So this means this is simple an oxidation of the CH to this hydroxide. So all this information is helping us to arrive at the mechanism. We then wanted to see whether it's a radical mechanism or anionic mechanisms. Generally we add tempo and BHT to see whether it arrests the reaction, because these are radical quenchers. So if it's a radical reaction, it gets quenched. But in this case we got good yield not a good yield, but we got product. That means it's not a radical process. And we also detected this intermediate by using high resolution mass spectrometry. We varied these groups. In every case we got good accuracy of the intermediate, which proves that this reaction is indeed going through this intermediate. So we have proved the intermediate, we have also detected the water, and we have also found the intermediate product. So then we thought that the review, we submitted this paper, and then the reviewer asked how you have to prove why it is going to this side and not to the C side. There are two CHs, why it is going to this side. So we did a computational calculations and found that this intermediate is more stabilized than this intermediate. And this is what we proposed based on all the data that we have got. Initially it forms an active cobalt catalyst, which then gives a C4 activated species. This intermediate we have proved by using high resolution mass spec. This coordinates with alkyne. The alkyne then adds to the CO bond. Then water attacks at this cobalt and forms this intermediate E. This undergoes reductive elimination to give this enol, which undergoes tautomerization to get this ketone. And this intermediate we have isolated during our application study, and we have shown that in the presence of copper acetate it undergoes oxidation. So that happens here, and that if you have alkyl group on the this side, then it undergoes elimination. So this is the mechanism. So in summary, we have basically utilized phosphotransition metal, and we have used chelation assisted CH bond activation, and it's undergoing regioselective C4 activation. We use trifluoroethanol as a sole water source, and there is a wide substrate scope and good functional group tolerance. Before I conclude, I have another work on Catellani reaction. I will quickly go through this. Catellani reaction is one of the earliest examples of CH activation reaction. This reaction involves difunctionalization of halobenzenes. In this reaction, mechanism goes through this way. First, the palladium adds to the CX bond, then adds to the norbornene. It forms this intermediate. This does the CH activation to give this intermediate. And in this intermediate, there are two beta hydrogens. Normally, we would expect the beta hydride elimination, but it doesn't happen here in this way because it is actually not syn periplanar, it is anti-periplanar. And this hydrogen also doesn't undergo beta hydride elimination because of anti-Brett rule. So there is no option for this intermediate. The only option is it has to react with this electrophile and functionalize this position, which then undergoes a reaction with nucleophile to give this one to difunctionalized product. This is a Catellani reaction. It's known since 1997. The interesting aspect of this reaction is all the Catellani reactions that have been reported are all with palladium and norbornene. One will not work without the other. If you change palladium to copper or any other metal, it will not work. If you change norbornene to any other olefin, it will not work. So they form a perfect pair to do this Catellani type of reaction. If this is the data on Catellani reaction, so 99% of the reactions are done with palladium catalyst, only 1% of the reactions were done with rhodium catalyst, but there is no report using cobalt catalyst. So Catellani reaction has never been done with cobalt catalyst. Why we have to use cobalt? Because as I said before, cobalt is cost effective and it's also earth abundant and it is having lower toxicity, which goes one step ahead to greener synthesis. Another aspect about Catalani reaction that I would like to talk about is the norbornenes, the bicyclic olefins. So the first Catalani reaction was discovered by using norbornene. After that, many generations of norbornene types have been developed, but all of them retain the basic core, the bicyclic olefin system. And these bicyclic systems, this is okay, commercially available, but all these others are not commercially available, so they need to be synthesized, additional steps. So they are synthetically less accessible, they are not cost effective and not commercial. So we thought that if we can actually replace these olefins with monocyclic olefin, then it will add another step towards a greener synthesis, because it is commercially available, cost effective and synthetically accessible. So we wanted to replace palladium and we wanted to replace the norbornene. So long back, one report has been done by Glorious, wherein they have done the orthoamidation by using rhodium catalyst and this norbornene, fancy norbornene, and this has to be synthesized by these harsh conditions. So we thought, why don't we do this reaction by using cobalt catalyst and olefin to get this orthoamidated product. We thought that this will go through this intermediate where this is anti-periplanar and there is no beta hydrogen on the other side. So this was our idea. And I am not going to go into the details of how we optimized it, but we optimized the condition and we found that this condition works, and variety of substrates are able to, we are able to do the Catalani type ortho-CH olefination with this substrate. And this is the mechanistic study. We have studied the mechanism. Just as I said before, that if you have to check the mechanism, we'll check using radical quenchers. In both cases, it is working, so it's not a radical mechanism. And we wanted to see which substrate is going. Electron-rich substrates are faster. Then we did the crossover experiment to see whether this donation, amidation, orthoamidation is actually intramolecular or intermolecular. So we have different substituents on either of them and different amides on both of them. In both cases, we found that it is going intramolecular amidation. And we also detected this cobalt-5 intermediate. Based on this data, we have proposed this catalytic cycle. It does the ortho-CH functionalization, and then melamide adds to it. Then it undergoes CO insertion to form cobalt-5 intermediate, which we detected in the HRMS. Then it undergoes electrophilic addition and extrusion to give this metala cycle, and a molecule of acetic acid does the proto demethylation to give this product. So this is the story, that two stories that I would like to share with you all. And yeah, this is another side product that we got if you use the electron withdrawing groups. With this, I'll conclude my talk. Before that, I would like to give an application. So even if you use a very complex molecule, this reaction works. You can see orthoamidation works. And we also did some application. We could able to get this molecule by treating with iodobenzene. Apart from the synthesis, we also wanted to show the application of our product. If you see, the product is hydroxyaniline, So we thought we can use that as a ligand. This is a known reaction by using amino acid, monoprotected amino acids. We thought, can we use our products as a ligand? And we screened a lot of our products, and we found that this L9 ligand is giving a better yield. So not only that we have synthesized these amino acid, these orthoanilides, hydroxyanilides, but we also showed application that it can be used as a ligand for this transformation. We took this ligand and we did a number of transformations, though the yields are not so great, but it works. So we have to do a lot of improvement on this. And just before we published, there is a group, they have also published a monocyclic olefin, and so they published in a better journal, but still we are happy that we were able to publish our work in still a good journal, ACS Catalysis. So basically, we have developed a protocol and we have used that products as a ligand and showed its application. This is the work. And apart from CH activation, we are also working on CC bond activation. So these are the works that we have done. We break a CC bond and form interesting molecules, melamides, acrylates, and then enines and diketones. We have recently written a review about CC bond activation in ACS catalysis, where we have showed how we can use small ring molecules as building blocks to make complex organic molecules like this. This involves our work and also some other groups' work. This is our summary of our CH bond activation projects. We have done indole functionalization, pyridone functionalization, and benzamide annulations, and these are CC bond activation. So all these works are not possible without the effort of sincere students, and the work that I presented today is done by these two students, Shyam and Tanmay. And finally, I would like to thank the funding agencies for their generous support, and finally thank you all for your kind attention. And if you have any questions, I'll be happy to answer. Yeah.
Thank you, Professor Ravikumar. Wonderful talk. And second, thanks. You mentioned the time also before you finish your talk.
Yeah.
Any one or two questions, quickly. Students may be prepared.
Yeah, so regarding your cobalt 5,
yeah.
Is there any precedence for cobalt 5 or this is the first time you are observing?
No, precedence is there. Cobalt 5 precedence.
What sort of ligands do they use for this? Like
what sort of ligands?
Ligands, yeah. Is it
no,
required?
cobalt 5 intermediates have been, we have shown some references. It is there,
okay.
But I am not able to remember what sort of ligands have been used on that.
Are they highly oxidizing? Any idea? Like
what's that?
Are they highly oxidizing in the sense like, are they stable?
Yeah, I don't know that ligand what you're talking about. Which literature evidences are there for cobalt 5, but I'm not able to recollect on top of my mind what is the ligand that has been used for that cobalt 5 intermediate.
Okay, then I will thank our Professor Ravikumar for this wonderful lecture.
Thank you.
Yeah, just Ravi.
Oh, okay. Sorry.
Let's move to the another session. It's different, it's industrial person who's going to interact with you. Doctors, so Sudeep. Sudiksha. Dr. Sudiksha is a very, he's completed his PhD for just 2020. He joined in Horiba, India. He's an application scientist. He's going to talk about, first talk will be Raman spectroscopy, and second will be fluorescence spectroscopy. It's a different talk. You can, you can enjoy the talks, both the talks continuously. Thank you. Dr. Sudeep, you can continue.
Yeah. Thank you, sir. Thank you, Dr. Jayakumar, for this invitation. So since morning, you all have been listening to mostly organic chemistry. So this will be a digression from that. I'll not be talking anything about organic chemistry, though I like organic chemistry very much. I ended up doing my PhD in physical chemistry, but organic chemistry was something I liked very much. So thank you to all the speakers, just brushed up my knowledge. So coming to the talk, so I'll be talking about two techniques. One is Raman spectroscopy, which you all know probably, and then also fluorescence spectroscopy, and what are the recent developments that have been made in Horiba. Okay, so I'll talk about Horiba Raman microscopes for the characterization of 2D materials. I guess a few of you at least will be working in 2D materials, which is actually the material which will come up in future in a very big way in the semiconductor industry, in electronics, in many different fields. And what do you do for characterization of these kind of materials? How do you characterize these materials using Raman spectroscopy? Okay. As is the norm with any company presentations, I'll just briefly run through the corporate overview of our company, that is Horiba. So Horiba is a Japanese company based in Kyoto, Japan. Atsushi Horiba is our group chairman and CEO. It was established way back in 1945 and it was incorporated in 1953. We're a worldwide company having more than 8,000 employees. So that was about the company overview, and we have three different segments in the company which caters to all these different applications. So bio and healthcare is one of the segments, material semiconductor, and then energy and environment. So there are plethora of instruments that Horiba makes. It's not just a one instrument company, but because you have different applications it caters to. This is a group of 49 companies spread across in 29 countries. In India, we have our presence in different cities like Nagpur, Pune, Bangalore, and Delhi. And along the way, since its inception, Horiba has acquired a lot of different companies. A few of these companies you will probably know yourself. Specs is one of the companies which makes high-end spectrometers. Dillor is another company. Mira is another spectrometer company. Jobin-Yuan, I think you have seen this. You have a fluorescence spectrometer. So you must have seen the CCDs or the detectors that come from Jobin-Yuan. Also the gratings are manufactured from Jobin-Yuan. And then PTI is basically one of the companies which produces time-resolved spectroscopy instruments. Okay, coming to the topic itself. When we talk about Raman spectroscopy, there are a range of instruments that Horiba makes. Starting from a benchtop and affordable model called MacroRam. It's called MacroRam for a reason that you can do mostly bulk analysis in this. This is basically a dispersive spectrometer where you do a lot of bulk analysis. You cannot do any kind of microscopic analysis. When you come to microscopic analysis, there are a range of other instruments. I believe you have this instrument in the department here, which was earlier called the LabRam 800. It has now been called the LabRam Odyssey. This is the lower end model among the microscopes, which is called the Explorer Plus, and this is an intermediate model which is highly automated in nature. It's called the LabRam Soline. All these are confocal Raman microscopes. Okay. All right, coming to the topic again. When we talk about 2D materials, one material or when you talk of materials in general, one material that comes into your mind immediately is graphene. Okay, graphene is one material which has been extensively used in many different devices now, in display materials as well. Graphene was, single layer graphene. When I say graphene, there was always graphitic material, but single layer graphene was discovered in 2001, 2002, when Andre Geim did this famous experiment of pulling out single layers of graphene using Scotch tapes, for which he was later awarded a Nobel Prize. So, and then it was found that graphene has many different properties, ranging from different mechanical properties to electronic properties. Since graphene had been discovered and all its properties, most of its properties were uncovered and understood, and it was understood that as you go down to atomistically thin layers, when I say atomistically thin layers, it means a single layer. Okay, this is just a one atom layer basically of carbon. It was found that these kind of materials have a lot of different properties, which is different from bulk material. Okay, and now you see in this family of 2D materials or materials in general, graphene is not alone. There have been many materials that have been discovered, and right now all the rage is about transition metal dichalcogenides, where you have the chalcogen element, chalcogen group, which is combined with transition metals, and you have different kind of materials like molybdenum disulfide, tungsten diselenide, tungsten disulfide, all these kind of materials. And these have different properties, okay, which is different from bulk material. Now, question comes, how do you, characterize these kind of materials? You have these materials, you synthesize these materials, how do you characterize? Because a lot of the device performance or the material performance as a very good electron conducting material or anything that has a very good mechanical property depends on the kind of arrangement of atoms it has, or depends on what kind of stress or strain that is present in the particular material. So you need to characterize all these different materials. Okay, why Raman spectroscopy for 2D materials? You have different properties which you want to understand, as I was explaining. Say for example, you have some kind of a dopant added into it just to increase or make this property of, let's say, electron conducting nature of that material better. So you dope it with something and you want to understand whether the doping has been uniform or what kind of doping is present. All right. So for that kind of studies, you can use Raman spectroscopy. There could be defects in any of these materials. To understand these defects, you need a characterization tool. So Raman spectroscopy can be a tool for that. Then to understand interlayer coupling, you have single layer graphene, but if you keep adding layers to it, you'll have double layer or tri-layer graphene. So if you want to understand what kind of coupling is going on, and if you have certain materials where you on the top layer you have one kind of a material, then at the bottom layer you have another kind of material in a different arrangement, in a different geometry. So you want to understand what kind of coupling is there. So Raman spectroscopy can give you an idea about that also. Band structure can be understood. When we talk of semiconductors, band structure is inevitable. You talk about wide band gap or low band gap semiconductors. So you can understand this by Raman spectroscopy and photoluminescence studies. Then number of layers. Okay, you can understand what are the different number of layers. So for all these kind of applications and characterizing these kind of materials, Raman spectroscopy is useful. Let us see how it is useful. Okay, before I get into it, I know I'm talking to a knowledgeable audience who know, probably you all know what Raman spectroscopy is, but just in the interest of few who do not know, I will say what I'll just explain it in one slide, what Raman spectroscopy is. We all know that we have atoms and then chemical bonds connecting these atoms to form a molecule, okay? And all these are never at rest, they're always showing some kind of a motion, okay? All these have some kind of vibration. It may be a symmetric stretch or an asymmetric stretch, or kind of an in-plane rocking or in-plane scissoring, out-of-plane bending. All these different motions are possible, okay? Now, all these different motions have their own characteristic vibrational energy, okay? Fine. So, what about these energies? We need to probe these energies to understand what are the vibrational frequencies and what are the different motions, fine? Consider you have a collection of molecules, an ensemble of molecules like this, and you have incident photons. There is a light matter interaction basically. So what happens once you have a light matter interaction? There are different processes that can happen. One thing definitely is absorption. As you know, you do a lot of UV visible spectroscopy, I'm sure. So there is absorption, then there is fluorescence, then there can be reflection, there can be diffraction, there can be scattering, okay? Now, these are the different things that can happen. Let us only concentrate on scattering because Raman spectroscopy is basically a scattering technique. It's Raman scattering that we're talking about. So there can be, once the photons are incident on all these collection of molecules, there will be scattering. Now, these scattered photons, the energy of these scattered photons will be almost same as, not almost same, exactly same as this particular incident photons. This is called Rayleigh scattering. There is no change in energy at all. So these kind of scattered photons are not useful to us because they do not provide any kind of information, okay? But there will be very small fraction of photons which will show some kind of change in energy. It may be higher than the incident photon energy or lower than the incident photon energy, okay? A very small fraction. And when I say a very small fraction, okay, when I say a very small fraction, what is this fraction? To give you an idea statistically how weak a phenomenon Raman spectroscopy is, if 1 million photons are incident on any material, 1 photon that you get out of it is a Raman scattered photon, okay? It's that weak, okay? Statistically, that is the probability of getting a Raman photon, fine? So all these photons, you can see there is a very small change in energy which is given by this formula, h nu naught plus minus nu t. Now everything is about this nu t. What exactly is this nu t? This nu t is nothing but the vibrational frequency. So we have seen this formula earlier. Nu t is directly proportional to root over k by m. K being the chemical bond strength and reduced mass, M being the reduced mass. So you can see this is very specific to each kind of chemical bond and the atoms that are present which are involved in forming that chemical bond, okay? So Raman spectrum usually looks like this. You will have a central frequency which is basically nothing but your excitation frequency, and then on the either side you will get Raman scattered photons, fine? So this is another diagram which you have already probably seen. This is basically an energy ladder diagram showing what Stokes scattering and anti-Stokes scattering is. If you just look at the Stokes scattering, you start from V is equal to 0, which is the lowest vibrational energy level. Once the molecule interacts with the incoming incident photons, it is taken to a virtual level, virtual energy level, and then it comes back to V is equal to 1. So in the process, what is happening is there is a transition from V is equal to 0 to 1. So there is a net jump of 1 in the vibrational ladder. So that is the transition that you're looking at, and that is the vibrational frequencies it will pick up. And we all know that this was discovered by Sir C.V. Raman in 1928, for which he was later awarded the Nobel Prize in 1930. So this is in a nutshell what Raman spectroscopy is, and what you can do from this technique. You can get different kind of information. Band position gives you information about what kind of chemical species you have. By the way, you can use this for organic molecules also. You know, if you have composites or mixtures, if you have polymer composites, you can use this for different kind of materials. So band position tells you about the chemical species, what kind of crystal phases you have. If you have an alloy, what is the alloy composition? If you are subjecting the material to some kind of strain or temperature, then there will be frequency shifts. So you can predict what kind of strain or temperature effect is, what is the effect of strain and temperature in the material by looking at the frequency shifts. You can look at the line width and predict about structural disorder. Intensity gives you the relative concentration of the materials. So Raman spectrum is basically like a fingerprint. Okay, it is specific to each kind of molecule. So here I've taken the example of ethanol and methanol, and you can see just by changing one functional group, that is a methyl group, there are differences in the spectrum, right? So it is unique for a given material, and it's like a fingerprint basically. Okay, so how do you explore your sample with Raman imaging? Now we have spoken about Raman spectroscopy, that was a single spectrum, how to record these spectra. What do? You can go one step beyond. If you have a microscope and you can do a Raman imaging, you can generate chemical images. So you can generate images of this sort, okay, wherein, you know, this is a graphene that has been put on a silicon substrate, which has all different layers of graphene, monolayer, bilayer, trilayer graphene, fine. And you can see based on the vibrational frequency differences of the monolayer, bilayer and trilayer graphene, based on the spectral differences, these can be ideally identified. How you can beautifully identify these. Similarly, for other 2D materials, you can do Raman and PL imaging. This is for the tungsten disulfide. You can also do a 3D imaging using, sorry, second talk. Okay. Right, yeah. Okay, so this is the imaging that you can do, right? So this is how you do a Raman imaging based on the differences in the different spectra. Phase identification can be done, stress measurements. I'm not going to the specifics of this because in the interest of time. But you can do this kind of mapping. Then in graphene materials, you can identify what kind of thickness it is or what are the number of layers based on the G band, D band and 2D band analysis. Okay, coming to I'll just finish this talk in another 2 minutes. Now most of the things that you saw was using a microscope, wherein, you know, the spatial resolution of a microscope is limited by, is diffraction limited. So the best you can do is the laser that is focusing onto the sample, the spot size will be somewhere around something like, you know, 1 micron or just less than 1 micron. But if you have some material wherein you want to characterize something at the nanometer scale, today morning there was a talk where M31 virus was shown, which has, you know, a fibrillar kind of arrangement, which had a length of around 800 nanometers. Now, if you want to chemically identify what is present in these fibrillar structures, so you'll need a technique wherein you need to go down to that kind of resolution. So to go down to that resolution, what you do is you can couple your Raman microscope with an AFM. So you do an AFM and a Raman measurement together, which is called tip-enhanced Raman spectroscopy, and that gives you further information at the nanometer level. Okay. So all these different microscopes can be coupled to an AFM to get the AFM Raman microscope. And this is one of the reports that was published for molybdenum disulfide. This is one of the first reports actually on molybdenum disulfide. This was published in the lab of Marlon Scully at University of Texas at Austin. This is for a carbon nanotube. You can see you can easily identify where the defects are using an AFM Raman or a tip-enhanced Raman measurement. This is for a vertical heterostructure. Okay, so with that I'll finish this particular talk. This is the end of the first talk. So we saw what Raman spectroscopy can do when it comes to characterization of all these different materials. I've shown only 2D materials, but does not mean it is limited to 2D materials. You can do it for polymer composites and different kind of organic molecules also. Okay, thank you very much. So can we have the second talk, please? Can we have the second talk, please? All right. So the second talk I will talk to you about is fluorescence spectroscopy, how time-resolved fluorescence solutions by Horiba can be used to study biological processes. So let's come to this problem. This is the different time scale, okay? And you have different processes that happen at different time scales. Now, if you start from, something like a simple chemical reaction, that will happen in a time scale of seconds to probably, a few minutes, okay? So let us say polymerization or an electrochemical process. Protein folding is something that always is happening, and in vivo or in vitro, that usually happens in a time scale of about, 1 millisecond to 1 microsecond. But there are other different processes that happen at a very fast time scale. And these time scales are ranging from nanoseconds to femtoseconds. Okay? To give you an idea, nanosecond is a millionth of a millisecond, and femtosecond is a millionth of a millisecond. Okay? So you need techniques which will probe reactions at this particular time. Okay? And any technique that you use for doing this kind of study is called a time-resolved technique. It can either be an absorption technique or a fluorescence technique or an IR technique. But there are different processes like ultrafast electron transfer or a cis-trans isomerization or a photo-induced pericyclic reaction that can happen at time scales of femtoseconds or few hundreds of femtoseconds or picoseconds. Then proton transfer, like excited state intramolecular proton transfer, can happen at time scales of hundreds of picoseconds. Then there are time scales where diffusion process and bimolecular hydrogen atom abstraction. We saw this kind of an abstraction reactions today. You can study those kind of reactions using these fast spectroscopy techniques, which are called the time-resolved technique, right? Okay, there are different excited state processes. There are two processes that are important. One is fluorescence, the other one is phosphorescence. I'm sure you all know what Jablonski diagram is, but I'll just explain it in terms of the time scales that we just spoke about in the last slide. So you have some molecule in the ground state, it is excited to the S1 state, let's say, the first excited singlet state. And then what happens here? Basically, in any kind of time-resolved spectroscopy, what you're trying to do is you're trying to understand the excited state process about how it is decaying once it is in the excited state, how it comes back to the ground state. That is the question that you're trying to answer any time you do any kind of time-resolved spectroscopy. In the process, what happens, you understand what are the different chemical or biochemical or photochemical processes that are going on. Okay. So once it is in the excited state, it has to invariably come back to the ground state, but it doesn't simply come back to the ground state because there are various decay channels available for the molecule. So first thing what happens is there can be vibrational relaxation among the vibrational energy ladder in the excited state, and the time scale for these kind of reaction is picoseconds. Okay. So if you want to probe anything in picoseconds, you need something that will, some kind of spectroscopy, some kind of technique that will probe something in picoseconds. Okay. Then what happens once it is at the lowest vibrational energy level, there will be fluorescence. Fluorescence is usually in the time scale of nanoseconds. Okay. So if you want to probe fluorescence, you need to do nanosecond time-resolved spectroscopy. Other thing that can happen is an intersystem crossing. It can cross over to a triplet state, and that can be at time scales of again picoseconds or femtoseconds. These can be competing processes. So again, you need something to probe these particular technique. Then again, there can be a vibrational relaxation, and from there will be either phosphorescence or there can be another intersystem crossing, and it will come back to the ground state. So basically, you can see fluorescence will be in the time scale of nanoseconds. Phosphorescence is more long-lived, so that will be in the time scale of hundreds of nanoseconds to microseconds, milliseconds, or even seconds. Okay. So question remains, how do you probe these? Okay, it can form other photo products also. That can also be probed. So this is just an example. I'll skip this slide because once it is in the excited state, how do you probe this? Basically, what happens when there is a vibrational relaxation? There can be a decrease in bandwidth and there will be a shift in frequency. So this is just one of the example how you probe excited states and how you understand what is happening in the excited state. Because what happens once you excite to the excited vibrational level in the excited electronic state, initially the population is well distributed over the complete vibrational ladder. So you will see that the width that you're getting is quite broad, initially at least. And then what happens, it will slowly relax along this particular vibrational ladder. It will come to the lowest vibrational energy level. So whatever fluorescence or whatever spectrum you're getting from there will be quite narrow and it will be shifted. So you can either probe that decrease in bandwidth, or you can probe the increase in frequency, and you'll get these kind of curves, and this can be fit to understand what kind of process is going on at what time scales. So these are the different instruments that HORIBA makes when it comes to fluorescence, ranging from steady state instruments for like Fluoromax, Fluorolog, Nanolog, and Aqualog and Duetta, to dedicated lifetime instruments like for TCSPC like Delta Pro and Delta Flex. Okay, I will just come to these applications. How do you study of biological processes by emission lifetime techniques? Now, while studying biological processes, there are two things, as I mentioned, fluorescence and phosphorescence. Fluorescence timescale is well suited to study biological processes. Okay, you have instruments for this, you can do this kind of study. But when it comes to studying phosphorescence, there are a few challenges. What are these challenges? Usually until now, most of the instruments, what was being done is, to study phosphorescence, which is a long lifetime process, you either use a source which is low repetition rate gas-filled lamps, like flash lamps, like xenon lamps, okay? But there are certain challenges associated with this. Otherwise, you use very large laser systems, like ultra-fast or picosecond laser systems, and that has its own problem. These are quite expensive, everybody cannot afford it. It needs high level of expertise to align these kind of lasers. So there are a lot of different challenges with it. But what if you have very small sources, which you just have to, it is kind of a plug and play. You just put it in the instrument and then do the experiment. So HORIBA has solutions for that. So these are the different sources. These are very small sources. You can just remove the source whenever you want, and you can just plug it in, and then you can start using the system. So these come with discrete wavelengths. If you want 416, you will have 416 nanometer. If you want to do an experiment at 350, you can have a 350 LED laser. So there are different delta diodes, nano LEDs, and spectra LEDs. Spectra LEDs are specifically for studying phosphorescence kind of timescales. Okay, these are the different delta diodes that are available at all these different wavelengths. Okay, wherein you can do, it's a fast repetition rate laser source, and you can study nanosecond and few microsecond processes in this. Next is the nano LED, which is again a plug and play. It is a standalone or works with a delta hub data station. So I understand you have recently purchased one of the data stations here. So all these different kind of wavelengths can be used to study, to excite the molecule and then study these different excited state processes. Then there are spectra LED. This is specifically meant for studying phosphorescence lifetime measurements. Okay, coming to two applications very quickly. How do you study the temperature-induced changes in proteins? One example is using HSA. Here I've taken the example of human serum albumin, which has a tryptophan amino acid. There are a lot of tryptophan amino acid residues, and we know that mostly the aromatic amino acids that are present, these aromatic amino acids show fluorescence, okay? And they are also capable of showing phosphorescence in different conditions. So usually when you record a time-resolved fluorescence spectrum, it looks something like this. You have the time axis, okay, and you have the intensity axis. Basically, there will be an initial rise, and then there will be a very fast decay. So this is the intensity axis. You might as well write the intensity axis here. So it is basically an intensity versus time curve. So you will have a decay of this sort. And here in what has been done is that it has been studied at different temperatures, starting from ambient temperature, starting from 20 degrees, then 25 degrees, and going all the way up to 60 degrees. Okay, you study it at very different temperatures, and the decay profile has been monitored at all these different temperatures. And to understand what are the lifetime, what you need to do is you have to fit the data, and you can fit the data using these exponential. We basically know this from our chemistry classes from maybe BSc, that any kind of kinetic process can be fit to an exponential. This is sum of exponentials which you can fit to. And alpha I here basically is the contribution towards each of the components that are present in this particular fit. If I here is equal to 1, it means it's a single exponential fit. If I is equal to 2, it means it's a bi-exponential fit. Likewise, you can have a tri-exponential or 4-exponential fit. And for each of these different components, you'll have an alpha, and that's the contribution. Okay. And by this, what has been done, time-resolved fluorescence study has been done, and time-resolved phosphorescence study has been done. And at different temperatures, you can see that starting from lower temperature, which is at 10 degrees Celsius, the lifetime is around 5 nanoseconds. Okay. As you increase the temperature, the lifetime decreases to around 2 nanoseconds. Fine. So this way you can study what is happening to a particular protein or particular biological molecule as the temperature is increased. Similarly with phosphorescence, there is an initial decrease that is seen until 40 degree, and then there is an increase in the time after 40 degrees Celsius. So this basically means how your protein is actually folding and misfolding as a function of temperature. Okay. So you can correlate this particular that particular process using the lifetime studies. Fine. This is another example. This is the last example of detecting conformational rotamers using TCS-PC. Here the HSA, the human serum albumin protein, was put in with the quercetin, which is basically a flavonoid. Flavonoid is seen in many different food products like tea and coffee and all these different products. So here in what interested the authors was to study what is the effect of these flavonoids on the protein. Okay. So to understand this, different concentrations of this flavonoid was used, and it was done using this particular instrument, which is called the 5000U lifetime instrument with a nano LED. And these are the TCS-PC results. So you can see here it was fit to a 3 component, and these are the lifetimes, different lifetimes. These are the contribution of each of those components. Okay, and that has been just graphically explained in this case. Okay, and with increase in the concentration of quercetin, this is how the component contribution changes. Fine. So this is basically how you can understand with time-resolved fluorescence and phosphorescence spectroscopy what is going on in a protein molecule, how it is folding or misfolding. If you add some kind of a flavonoid, what happens? If you subject the protein to a temperature, temperature-dependent conditions, then what happens? So you can understand these kind of different physiological phenomena. With that, I come to the summary. So time-resolved fluorescence is indeed a useful technique when it comes to studying different biological processes. And HORIBA has a complete portfolio of different kind of steady state and time-resolved instruments to measure fluorescence and phosphorescence. These are versatile instruments with the capability to measure lifetimes ranging from picoseconds to milliseconds. And these simple light sources are available like delta diodes, nano LEDs, and spectra LEDs for your routine research work. And thank you for your patient listening, and thank you for your time. If there are any questions, I'll be happy to answer them.
Thank you, Dr. Sudaksha. Very detailed talk about Raman spectroscopy and the time-resolved spectroscopy. Thank you very much. And the students, you can interact with him during the poster session detailedly. Without further delay, I'm going to next talk. Let us honor Dr. Sudaksha with a small momentum. This book only. Okay. Next, I'm going to invite Professor Jayaraman Govindasamy. Is from Department of Energy and Environment Unit, Indian Institute of Nanoscience and Technology. Professor Jayaraman is graduated from Indian Institute of Science, Bangalore, under the guidance of Professor Jayaraman. He has done 2 postdocs. One is in ETH Zurich with Professor Francis Dietrich, and University of Cambridge with Professor Jonathan Nitzke. After that, he joined in NIST, Mohali. Now he currently is a professor. With a few words, I invite Jayaraman to deliver his talk. Thank you.
Thank you. Thank you, Dr. Jayakumar, for the kind introduction and also for the invitation. I also would like to thank co-organizer Dr. Suresh and Professor Ramaraj sir for inviting me. So it's a really great privilege and pleasure to be at MKU. It has been, I have a very good feeling about this university, and I know a lot about this university. And so my talk, since the lack of time, I will a little bit rush up. And my talk will be a little bit different from so far, where I have seen hardcore organic synthesis and catalysis mechanism. though my part of talk also will have organic synthesis, I will also add up a flavor called nanotechnology. Okay, so I will use the organic synthesis and nanotechnology as a tools to make materials, and using that materials, how we can exploit and show applications such as energy, environment, and health. That's the overall mission of my work. So where I come from, before going to the work, let me acknowledge the people responsible for this work. So these are my graduate students, and those people highlighted with the green are contributed in this presentation. These are my collaborators. Since it's an interdisciplinary work, without this collaboration, it's not possible to realize this kind of work. And of course, not without adequate funding from DST, DBT, and SRF industry and INST. So where I come from, Institute of Nanoscience and Technology is situated in Punjab, Mohali. It's near to Delhi. This is our permanent campus. It's the actual campus. This campus institute deals with the chemistry, physics, biology, but all centered around nanoscience. So those students who like to do PhD in organic synthesis and nanotechnology would like to write to me. So I will be happy to accommodate. So the goal of my research is, can synergism of molecular chemistry and nanotechnology would lead us to develop functional nanomaterials for this energy and environment and health applications. So I have two different themes. One is towards luminescent push-pull chromophores, again on this application part. And second is the biomass waste to wealth using nanotechnology. So in which two approach again, one is functionalization of biopolymers for value addition, and second is biomass as a precursor for sustainable catalysis. So just to give you a brief introduction about the other topic, today I will not be discussing about this topic, that is push-pull chromophore systems, where we also work on synthetic methodology to develop this reaction and the products and the mechanism involved in that reactions. Also to bring new additional property, that is fluorescence property, in addition to chromophore property, by using nanotechnology approaches. And we also work on sensing and develop fluoride detection kit in drinking water using small test type paper kit, and blood cholesterol serum detection kit also there, which are patented. So, however, today I will be discussing about the second topic, that is biomass waste to wealth using nanotechnology. So here we adopted two approach. One is through value addition, in order to use that large abundant amount of cellulose material available from nature or generated from nature. And second, use that direct biomass as a material for both as a catalyst making as well as to convert the abundant cellulose into fine chemicals. So just to give a very brief introduction about bioenergy, of course, there is no introduction required for energy. We all need energy. And there are two types of energy. One is renewable energy, another non-renewable energy. Obviously, we need renewable energy for sustainable world. In the renewable energy, there are several types in which biomass energy is more promising because of the two reasons. One, we have abundant biomass, and that has to be used. When you use, you also prevent the pollution. The land and water pollution can be avoided. And there are so many different sources of biomass in which the agriculture plant-based biomass is huge. And according to EAI, 450 million tons of biomass per year being produced, and 32% of our primary energy being coming from this biomass energy. The world energy can be made from this particular energy itself. How I will tell in the next slide. So biomass is being produced directly from photons coming from sunlight, water, and CO2, which converted into lignocellulose in the wood. Lignocellulose is a very complex molecule having different kinds of cellulose, hemicellulose, cellulose, and lignin with the different ratios. And among these molecules, cellulose is simpler in terms of structure, so it can easily exploit unlike lignin. And cellulose, just take 1 kg of cellulose, it contains 16.5 megajoule of energy. And if you convert into 450 million tons, that's sufficient for world consumption energy per year. Of course, it's not possible to exactly take all the energy from that. That is the thing we want us to work on. So these are the ways people utilize biomass into materials, advanced material, functional food, chemical products. And there are two ways one can approach. One is the degradation of fine chemical. Another one is utilize them as value addition. So this approach is very good because you use all the energy which is nature produced. Whereas in the degradation energy, you lose some energy by breaking the molecules. So wherever it is not possible to utilize the material, then we have adopt the degradation method. So by this, we would like to achieve a sustainable circular bioeconomy. So in the inaugural session, Professor Chandrasekharan mentioned that the future of chemistry should be sustainable chemistry, right? And to address our energy needs. And exactly this work is working towards that mission. So as such, in one functionalization, another one is recycling directly as a precursor biomass. And those biomass waste we deal with cellulose, sporopollenin way, this genetic material made by the plants, and citric acid as a raw material. So by using this approach, we would like to address sustainable processes and through value addition for environmental friendly. So these are the various work we have published recently by the approach of functionalization through thiol functionalization or amine functionalization and corresponding nanomaterial synthesis in drug delivery and catalysis area. So what I mean by functionalization, you take a cellulose, just functionalize with the tosyl cellulose. Tosyl reacts specifically at the C6 position to give this tosyl cellulose. It's a easy living group. Just add the nucleophile ethylene diamine. It replaces tosyl with the generating aminocellulose. It has been published in this paper. So we can make this compound in gram scale with 75% yield. So why we are interested in aminocellulose? In order to get the more biocompatible polymer, because we want to use iron oxide nanoparticle. Iron oxide nanoparticle does show some slight toxicity. To reduce the toxicity of iron oxide nanoparticle, we coat with the highly biocompatible aminocellulose, and we develop, in addition to biocompatibility, it also provides functionalization. Using the amine functional group, we grow the hyperbranch polymer and then cover with the peg chain in order to get bioavailability. So this is a nano carrier we developed, and then we collaborate with our in-house biological expert, Dr. Regan Khan, and he came up with another approach that is called synthetic lethality approach. Synthetic lethality is nothing but in the cell, we have one enzyme, one gene product, BLM gene, SOD1 enzyme. This drug molecule known to inhibit SOD1 enzyme, compared to normal cell and cancer cell, in the cancer cell, the BLM gene is deficient. When the BLM gene is deficient, when the SOD1 is inhibited by this drug, the cell dies. Whereas the same normal cell, which is proficient with the BLM gene, even if the SOD1 is inhibited, it doesn't die. So that means we can selectively kill cancer cell. So by mixing this dialysis-1 drug with this nanocarrier, we could able to improve the selectivity of killing to 1.04 times, as well as the sensitivity 1.7 times over LCS1 drug alone. So we also demonstrate that the developed nanocarrier and the aminocellulose coated nanoparticles are more biocompatible than the only nanoparticle, even at the concentration of 500 microgram per ml. So after publishing this work, we originally intend to develop not only chemotherapy, also the hyperthermia. That's the reason why we use hyperionized nanoparticles. They are known to have superparamagnetic character. Upon shining a magnetic field, it generates heat, due to which you can kill the cancer cells. So in addition to chemotherapy, we also demonstrated magnetic hyperthermia, by which we could be able to kill cancer cell compared to control, only control. So in this case, we use another anti-cancer drug that is called niclosamide. So after demonstrating the drug delivery application for using aminocellulose, we are also interested in catalysis. And not only the objective of addressing biomass utilization, also as the chemist, we would like to address the gap exists in the literature. For example, in catalysis, click chemistry, everyone know. You take azide, alkyne, react, put a CC copper metal, it forms triazole ring. Even mechanical engineering people use that, or biochemistry people use that. But there's a 150 years old reaction called Glaser coupling, which is not that famous. The main reason is click chemistry brings orthogonality. You get two different functional group R and R1, and that is brings two different functions. Whereas in Glaser coupling, you get CC bond forming between alkynes to produce homo as well as hetero products. The homo product is not required because it brings only one functionality, whereas the cross-coupled product brings two functionality. If you can get that, that can be used for several applications. So people, what they do, they just change the metals and get some selectivity, and that is not a versatile one, and the maximum selectivity could be actually 60%. So at that time I was thinking, can I come up with an approach where I can use this cross-coupled product as a major selective product? So when you look at the structures, the products, you get they're inherently different in polarity. This 2OH group is highly polar, 1OH medium polar, no OH non-polar. So if they are differently polar, the starting material as a product, then we can selectively bind the catalyst backbone. If the backbone polymer has a hydrogen bonding donor acceptor moiety, they can have more number of hydrogen bonding interaction by which we can selectively control the formation of Glaser product. So this is the approach. Using this approach, we design the catalyst. We made a series of aminocellulose. In this catalyst, there is no nitrogen, here 2 nitrogen, here 4 nitrogen. So we tested I will not go into synthesis and characterization due to lack of time. I'll directly go to the catalyst testing activity. So this is the final slide in this work. So this is the trend catalyst with the 4 nitrogen. We tested with this model substrate reaction, and we got the cross-glacial product 3A, 82% yield, which is very high compared to the literature work. And then most importantly, this when you change this, when you add dodecanoic acid to the reaction mixture with the same catalyst, now the selectivity shifted towards non-polar homo product. That's because the dodecanoic acid binds with the tertiamine in the center, and then that area becomes hydrophobic in nature, that prefers non-polar product. And similarly, when you change to polar acid, that is benzene tricarboxylic acid, in this case, the product favored is this one with 76%. Whereas other two catalysts, the tertiamine, don't do that changes. So this proves that there is hydrogen bonding interaction happens and the backbone, which favors the cross-coupled product. That was proved by my colleague Dr. Hassan Ali, who has done DFT calculation and showed that cross-coupled product shows hydrogen bonding interaction with the backbone. And then using that same catalyst, that is amine functionalized, then we took this normal without amine functionalized cellulose copper oxide catalyst, which is very easy to make. And since that is very active in Glaser product, then you should also catalyze click reaction, because copper can catalyze click reaction. So we tested its activity for click chemistry, various substrates, and it worked very well. And most importantly, it works in water itself, 100% water at room temperature. It's a very greener condition. And we also demonstrate by changing the nanoparticle size, we could be able to get the higher activity. Because of the lower size, higher the surface area, you get higher activity. So this is the catalyst synthesis, where we get by changing the ratio of copper chloride to substrate, you can get the different sizes that we tested. And then as an application, we also demonstrated for its use in synthesis of rufinamide drug. Rufinamide drug is an anti-epileptic drug. It's very expensive. If those have epilepsy should try this drug for lifetime, and it costs them a lot. The reason it's expensive is the one particular step, that is click reaction involving where the another regioselectivity also comes as a problem, challenge. And also they use organic solvent like DMS5 boiling point solvent. So we thought, why not we test our catalyst, whether it can do the same chemistry in a 100% regioselectivity. So we tested this particular step. In water isopropanol as a solvent at room temperature, and gave only one isomer by NMR as well as by HPLC, and we could be able to, in the laboratory scale 10 gram, we can just get this compound. And we have filed patent for this work, and we currently Biocon also approached us to test this catalyst for their industrial quick reaction usage. They are currently using it. And then now later we'll switch from catalyst to again drug delivery. So here the drug delivery is again proof of concept, where the lack of information in literature I would like to address through nanotechnology. Here is another interesting concept. Since we are interested in drug delivery nanocarriers, by using system chemistry, it is well known, you take amphiphilic polymer, put it in water, it will self-assemble to form micelle, to forming hydrophobic core, hydrophilic shell. That is well known. And by which, if you make a polymer, amphiphilic polymer like brush type, again put it in water, it has self-assembled to produce core shell structures. But all those literature structure usually have balanced both equal number of hydrophilic and hydrophobic chains. Then in order to improve the drug loading in the hydrophobic core, we thought if you can increase the density of hydrophobic core compared to hydrophilic shell, then the drug loading can be more. So to achieve that, we thought we come up with the concept called narcissistic self-sorting versus self-sorting. What is narcissistic? The self-assembly of the same molecule on its own group is called narcissistic self-sorting. But instead of self-sorting on its own, if you can mix with another different hydrophobic polymer, this one, by these two mixture, that leads to social self-sorting nanocarrier. So here what we done, you took the hydrophobic chain, functionalized with the ionized nanoparticle covalently. This is the covalence hydrophobic chain functionalized ionized nanoparticle. By mixing with the amphiphilic polymer, it generates self-assembles to give core shell structure, wherein you have double the number of hydrophobic chain, whereas hydrophilic chain is less. So by which you get the higher drug loading. So we demonstrated this by synthesis, and I'm not going to synthesis and characterization part due to lack of time, sorry about that. So this is very interesting study. You take that amphiphilic polymer, put it in water, you get the CMC concentration. The concentration is 0.02 milligram, whereas social self-sorting nanocarrier initiation concentration is 0.01 milligram. So that means further to prove that social self-sorting is happening, the TEM gives you very clear indication that you get core ionized nanoparticle and the shell polymer, whereas in narcissistic self-sorting you don't see core shell structure. Further, we also demonstrated by using the iron oxide nanoparticle hydrostatic magnetic field, we can increase the drug release profile, also by increasing the temperature. And another interesting proof of concept work in drug polymers, drug carriers. We all know drug polymer means you take a drug attached with the non-polymerizing monomer, make a polymer, either at the end or at the branching or at the backbone, or as a hydrophobic encapsulation through. But all this type of polymeric biocides have non-drug component, which are not required. Suppose I needed to take a drug 100 milligram, I will take 100% drug molecule. But if you have to take 50 milligram non-drug, that is not required. Maybe the activity also it can reduce. So then its hypothesis proof of concept question, if I make a drug which is 100% drug without any non-drug component, if I hydrolyze, it releases the monomer. So then we screened which drug I can polymerize and get that polymer. But for that we chose ciprofloxacin as a commercially used drug, antibiotic drug. So this is the type of polymer we are proposing where there is no drug component. In order to compare the spacer effect, that if you have more spacer, activity will be less, we also made two different polymer with the spacers. I'll show you this is the synthesis. Take ciprofloxacin, which has amine and carboxylic acid, just heat it up, condensation reaction, you get long polymer. It is not reported. And then we take another polymer with the spacer, C2 and C10, and then we characterize them, and then we coat it with the sutures, and the sutures shows clearly that polymer without spacer shows highest activity than the C10 polymer and C2 in against E. coli, similarly for SRAs. So even you can compare with the ciprofloxacin alone, the non-drug spacer shows higher activity. That is because the efficient coating as indicated by the SEM study, where ciprofloxacin coated sutures are clearly not visible, not coated, whereas the C2, again you can see the sutures threads, whereas in C10 you can see but they are porous, whereas the C0 without any spacer shows smooth coating. That's because of smooth coating shows higher activity. This was published in cover letter. And finally, after the value addition approach, we also showed just 2 minutes. 2 minutes, am I here? Yeah. So just to show how we use it as a precursor, as a biomass, as a precursor for other applications. So first we have one in cellulose where we use pesticide release by comparing organic nanoparticles as well as hybrid nanoparticles, which whether hybrid nanoparticles can be better or organic nanoparticles can be better approach. And sporoplenine polymer as a template to produce nanomaterials, and citric acid as a starting material to produce carbon quantum dot, and then we use it for catalytic applications. So due to lack of time, I stop it here. Thank you very much. If you have any questions, I would like to answer.
Thank you, Professor Jayaraman. It's fantastic talk. It's very kind of, totally different from the morning those who are given. Questions, 1 or 2 questions from students. Quickly. Otherwise, we can discuss in the poster session. I'll move to next session. So,
okay.
Yeah, talk now, photo.
Thank you.
Sorry.
Thank you.
Next. So next talk is going to be bioinorganic. I invite Professor Shankar, M. Shankar from IIT Roorkee. He is our alumni. He did MSc from Madurai Kamaraj University. And a PhD from IIT Madras. Mainly, he is also the bronze medal. He received a bronze medal from CRSA last 2023 last year, and also he is a JSVS fellow. With these words, I invite Professor M. Shankar to deliver his talk.
Thank you, Dr. Shiva, for your kind introduction. At the outset, I would like to thank Dr. Shiva and Dr. Suresh and the mentor, Professor Ramaraj, for the kind opportunity to discuss about our recent work. As Dr. Shiva mentioned that I did MSc from School of Chemistry, Madurai Kamaraj University. It was a wonderful period for me. Like I finished my BSc and came here. Then this institute made my life actually. I'm really grateful to this institute. So after coming here, I cleared GATE, CSIR. Then I moved on to IIT Madras and postdoc and other things. So really, it's a wonderful place. That 2 years I enjoyed like anything. Still, I'm with my group. Like we had 20 members in my class. So we have a WhatsApp group. And we have many juniors, seniors. So it's a wonderful meeting and the kind hospitality. Like yesterday, I didn't even reach Delhi, but a taxi driver called from Madurai that I will be picking up from the airport actually. So it's really nice, actually nice to be here. So today I will be talking about synthesis of pi extended metallocorroles and their application in sensing, NLO and catalysis. So I have organized my presentation in the following manner. First I will be giving about a brief introduction about corroles and their applications, and then I will start about beta disubstituted corrole. That's our first molecule we made. And after that, like we utilize that phenylacetylene appended corrole to tetracyanobutyridiene, like Professor Jayaraman also explained about that. Then we have prepared aldehyde substituted corals, and then we made dicyanovinyl out of it, and also like we have made aldehyde with a mixed substitution, followed by like a dialdehyde formation of corals, and then we used that dialdehyde to make dicyanovinyl and dicyanobutyridienyl. So then finally what we have done is that we have prepared some molybdenum corals and used as a catalyst. So our approach is that lab on a molecule. So I will show you that we have only 1 or 2 macrocycles. By functionalizing them, we can have different materials, and then by inserting different metals, we can have different properties. So that's how we are planning that. So this is about tetraprol macrocycle. You know, tetraprol macrocycle is very important for life processes, particularly hemoglobin, myoglobin does oxygen transport and storage respectively. Cytochromes are involved in electron transfer reactions. So here this is the porphyrin ring present in heme. Because of this ring only we have a color. So red color comes because it absorbs the visible region. Then when we have this one of the carbon missing, that is actually here, that is called corine. It is present in vitamin B12. You know, vitamin B12 is very important for DNA synthesis and then the biomethylation and many other processes that. So here this is actually the core size is 4 angstrom. Here we have reduced core size around 3 angstrom. That, so that gives wonderful properties. Actually this is corine ring which is present in vitamin B12, but it is difficult to mimic this ring, so that we have a fully conjugated ring system called Carol. So this is the one I am going to talk about this ring. And when we can insert different metal ions, so you can see that there are 3 NH, so that it will accommodate +3 charge, but porphyrin it will accommodate +2 and then as well as +3. That, so we can have a different isomers of porphyrin. Is one of the beta prol is reduced, it is called chlorine, which is present in these plants. So that gives green color. Actually it absorbs in the red region, that complementary color is green. Then we have an unconfused, this is synthetically formed. So in case of Carol chemistry, it is developed by 3 chemists actually. That, so initially Geogras and Roberto Polese, they reported synthesis of Carol. That was not a high yield method, but in 2006 Greco, what he has done is that he has taken the pyrrole and aldehyde mixed in water methanol mixture, then he got 30 to 40% of yield of Carol macrocycle. So here what we do is that pyrrole we take 2 equivalent and 1 equivalent of aldehyde. So the beauty is that actually if you take 1:1 of pyrrole and aldehyde, you will get porphyrin, this macrocycle. So if this aldehyde is the limiting agent, when you take less aldehyde, it forms Carol. And pyrrole 1 ml is only about 18 rupees. So in 100 rupees you can make at least like 500 milligram of this Carol actually. That. So actually because of this synthetic method, the publication has grown up recently like too high, because everyone can afford that. So here this is actually in 1999 Carol size was bigger, actually it is about 17 angstrom, and then in 2018 it reduced it to 11 angstrom because of the CF3 group instead of pentafluorophenyl. But recently one of my students also contributed to this work, that they made a baby Carol. It is only just half of it, 8.5 angstrom width. So this is useful for biological application, because cell permeability, when you have a small molecule, it will permeate, it will diffuse easily. So that is the new development here. So here, as I mentioned here, Carols actually have 3 NH, so it will minimum it will accommodate +3 ion. Okay, it can accommodate +4, +5 also. So and core size is small, that's why it's also stabilizing high valent metal ions. You can see here up to 6 charge. I will also show you molybdenum 6+ using Carol. Actually also from Carol to porphyrin also can be prepared using methanol. And also our interesting feature is that we say copper 3 Carol, but copper 3 Carol it never exists. It actually exists as a copper 2 Carol and then radical anion. Radical anion, you can see that there are many proofs over there, that as well as actually you can tune the this reactivity of Carol by substituting. These positions are very reactive, that's why I will show you that some example.
that where the reaction takes place over there. That. So here these are the some examples on pi extended corolles that we made in 2017. This corole is wonderful that actually it can take up electron, it goes up to 4 minus. That means that because of the extended pi conjugation, it's acting as electron sink, and also it can be used as electron donor to the porphyrin. Also people have used this kind of corolles as a nanomaterial stabilizer. This carboxylic acid will go and stabilize the nanoparticles. So it has a medicinal application also. That. So apart from that, actually corolles can be used in dye-sensitized solar cell because it has a visible absorption, and they can also used as electrocatalyst for reduction of oxygen to H2O2 selectively. It can be used as a sensors, and also it can be used as a photodynamic therapeutic agent. In photodynamic therapy, where they use porphyrin or corolle as a light absorber and then sensitize oxygen. That normal oxygen is triplet oxygen. If you sensitize that into singlet oxygen, is highly reactive, it kills the cancer cell. So one can use it for anti-cancer therapy. So apart from all this application, actually for our lab, these are the molecule of our choice. This is very simple, as I mentioned earlier, you can make gram quantities very easily. Every day we are making this molecule in our lab, that for different purposes. That. And one more system we would like to explore is that simple this tritoluene corolle or trinitrophenyl. This is also useful. When you put metal, it is acting as a catalyst. So these are the two systems I have chosen to discuss today. Actually, we started with the simple molecule dibromo corolle, silver corolle. So here silver is in silver 3. Copper only is copper 2 cation anion radical, but here it is actually copper silver in +3 oxygen state. This stabilizes, corolle macrocycle stabilizes the +3 oxygen state. Otherwise silver 3 is generally not stable. That. So we have taken this, we have prepared selectively dibromo, that it is because these 3 and 17 positions are reactive. Then we did simple Suzuki coupling reaction. We appended methyl and phenyl groups, and then also that we did Heck coupling, we got methyl acrylate, and then using Stille coupling we got phenylethanol, this in decent yields. So after that, when you look at the absorption spectrum, this molecule absorbs from 400 to 500 region, very intense absorption, because you have a pi conjugation. Any pi conjugated molecule will absorb light intense, so in the order of 10 to the power 4 and 10 to the power 5. Then when we look at the phenylethanol as well as methyl acrylate, because of extended pi conjugation, you can see it's broadening. That this is called Soret band, it is getting broadened. These are called Q bands, quasi-allowed bands. These also get broadened. That. So and also huge redshift we observed because of the extended pi conjugation. So that's what's our indication that, so extended pi conjugated corolles, metal corolles will show redshifted absorption, and different applications will be there. So that's where our starting point. Then when we look at the electrochemistry, any pi conjugated system, when you try to oxidize, it will form cation radical. If you try to reduce, it will form anion radical. That is the nature. But benzene also can undergo that, but it's highly unstable. But here these molecules are pi conjugated. Because of that, these cation radical and anion radicals are stable. So when you oxidize the corolle, you see that it will form cation radical and dication. But in case of silver corolle, when you try to reduce, silver 3 is easily get reduced to silver 2. So silver 3 is getting reduced to silver 2. But in case of oxidation, corolle macrocycle is reacting. So it forms cation radical and dication. So this is where observed. Of course, we have seen substituent effect. When you have phenylethanol, it's easy to reduce, okay, and difficult to oxidize. So those electronic effects always will be there. That. Then whatever we made in the this diphenylethanol corolle in the first work, then we have utilized that. Then when you react with this tetracyanoethylene, you remember that Professor Jayaraman told, when you take this phenylacetylene and if you react with this, there will be a cycloaddition followed by retrocyclization. So that they call it as a CAR reaction. So that cycloaddition followed by retrocyclization will give you tetracyanobutadiene appended porphyrin. So this will conjugate with the porphyrin 5 system. then you will see extended pi conjugation can be made, and we got excellent yield. Then when you look at their absorption spectrum, compared to normal corol, it's get broadened because you have extended pi conjugation, you have a corol donor and the TCBD acceptor, so that brings interesting properties over there. Their molar restriction coefficient also increases. So when you look at the electrochemistry, very interesting electrochemistry you can see. Here phenylethanol, you are seeing 1 reduction and 2 oxidation. But when you look at the TCBD appended, you are getting 4 reductions. So when we closely look at why the 4 reduction, first actually TCBD is very electron deficient. That undergoes reduction. That TCBD forms anion radical, followed by corol forms. Then next is again TCBD dianion, then corol. So these are 4 reductions takes place. Oxidation from the corol ring, cation radical and dication. So you can see here copper shows 5 reduction. How is shows 5 reduction? First, because copper 2 radical. So that undergoes the reduction, followed by actually this TCBD and as well as a ring. So that's how they are reacting actually like that. So these are the so you will have here silver will undergo TCBD followed by macrocycle, TCBD macrocycle like that. In the case of oxidation, ring that. So when you look at their this DFT structures, you can see that actually that LUMO that having electron withdrawing groups, so that it contributes. HOMO is from the macrocycle, that means macrocycle is acting as a donor, your TCBD is acting as an acceptor. Then when we do this photochemical studies, that particularly tension absorption spectral studies, we could see that there is a charge separated state, that TCBD is getting reduced, corol is getting oxidized. So we have seen their lifetimes are in picosecond, and also we can see that they are having higher triplet levels. Triplet levels are really populated. When it will get triplet levels are populated, when you have extensive charge transfer. So this indicates that they are having extensive charge transfer in the excited state. That. So when you take this aldehyde corol, I told that we have prepared this aldehyde corol by simple. You take corol and then add 15 to 20 equivalents of Wills-Meyer reagent, you will get aldehyde corol. Then you can do this Navier condensation with the dicyanovinyl, and also you can prepare metal complex followed by Navier condensation. One can do it. So we can able to get the crystal structure. In the case of cobalt, cobalt is in +3 oxidation state. It needs a pi acceptor ligand. Always we had PPH3. That PPH3 is stabilizing cobalt in +3 oxidation state. So without also is stable, but in the sense that some side reaction takes place. So always we protect cobalt 3 with PPH3. That otherwise we need to add another pyridine or some nitrogenous bases to protect them. That. So when you look at the electrochemistry, here also interesting that you see the TPC to aldehyde 140 millivolt anodic shift. Then if you further with the malonitrile, you will have 110. So when you have copper centered, metal centered reduction followed by ring centered actually like it. So when you look at their spectral properties, this is that when you have this silver corol, then when we had different type of anions, it is okay, I will try to finish. So when you have different anions, that it is really reacts with the cyanide. So we found that cyanide is a strong nucleophile, a strong base, so it attacks the double bond, so it forms dicyanovinyl appended, and also color changes. Then when you look at that reduction limit, it is in the sub ppm level, so that is very sensitive to the cyanide ion for the deduction actually. We have characterized by NMR as well as UV-visible spectroscopy, that followed by okay. So here mixed substituted corol we have prepared aldehyde, then when we brominate it is getting heptabrominated, but when you take porphyrin it does not react that way. So corol is unique in that, so that we could able to prepare wide variety of this mixed substituted porphyrins actually. The structure also shows that they are non-planar and having very good dipole moment. It is good for nonlinear optical application. That, so we have seen their absorption spectral properties. This CV also according to the electronics, when you have 7 bromo groups, then it is anodically shifted about 200 millivolts. That So we have done nonlinear optical studies. So this molecule having nonlinear optical behavior. Why they are having nonlinear optical behavior? Because this molecule has aldehyde and bromo groups. So they have polarization in the molecule. So when you shine with the laser light, so it responds to that actually that. So it is having very good nonlinear optical coefficients. Due to lack of time, I could not discuss in much in detail. Then further, actually, we also prepared this dicyanovinyl as well as TCB, this acrolein appended porphyrin, then dicyanobutadiene. So this dicyanobutadiene is shown that you see that it's a panchromatic. So in the beginning, I showed that it is having only one shorted band and Q band, two Q bands. Now you can see that we can tune the entire visible region. You can, because of strong electron withdrawing group, this dicyanobutadiene, it is in conjugation with the porphyrin pi system. Because of extended pi conjugation, they are showing redshifted absorption. When you look at the redox properties, it is actually we have induced some 4 to 5 reductions because you have multiple electron withdrawing groups, and then now pi system is ready to accept electron. So as much as you pump, it is accepting. So we can be able to see up to 4 reductions. Then when you look at the crystal structure, they are planar in nature actually. That. Then we could see that cobalt actually, we want to know whether cobalt is in +3 or +2. Actually in simple Carol, it is existing as a cobalt 2, this radical. But here in case of our electron withdrawing Carol, it is existing as a cobalt 3. That means that our electron withdrawing group stabilizes cobalt 3. That. So actually we have utilized the catalytic activity of Carol. Actually particularly we have prepared molybdenum Carol. Actually, sorry. Yeah. So simply we inserted molybdenum, we utilized as a catalyst for this kind of bromination. It mimics the biological process called bromoperoxidase. So we have done a wide variety of catalytic activity for the this phenol derivatives. We could able to get very good yield because these catalysts are robust in nature, and we have done also epoxidation. These catalysts are showing very good efficiency for the epoxidation. Then we have also established the catalytic mechanism, particularly like when we take this molybdenum Carol, it is already in +5 oxygen state in presence of peroxycarbonate. This is additive. Then it forms molybdenum 6. We have also analyzed by this NMR as well as this mass spectrometry techniques, and then we have established this catalytic mechanism. So at the end, actually I could say that we can have a simple mesoid substituted Carol, and then we can able to tune that into a catalyst as well as actually as a panchromatic material, as well as a triplet exciting the materials TCBD for that high population of triplet state, as well as we can also tune as a NLO material actually. So I am really thankful to my students. So far 11 students have been graduated, 5 of them are faculty, one of them Dr. Mandeep, she is faculty in UK, and currently more than 10 students are working. I have presented only Inderpal's work, others are working on porphyrin systems and related. So, sorry, last slide is that this is my current group. Recently Professor D.K. Chand visited, during that time we have taken the photograph. And actually Uttarakhand is a unique place. Actually I invite you, please visit Uttarakhand as well as you can come for PhD program. We have Haridwar, Rishikesh, Badrinath, Kedarnath. One can explore as a touristic place as well as academically rewarding. Thank you so much for your kind attention.
Thank you, Professor Sridhar, for your quick time. Maybe we'll move to poster session quickly. Maybe you can interact with the professor very detailedly. I'll close this session. Thank you. Sorry, Sankar. I'm very sorry. Okay. Sorry, bro. No problem. Thank you.
Thank you. So I will be available. You can ask questions, whatever you need.
Yeah. Now. So now the poster session will be okay. Ninga. Time. Okay. Now the time is 3:48. Okay. So up to 4:48 we can spend. Huh? So those who are posted, you can go there. The second year MSc students, you can stay here itself. Okay, the DSK is going to conduct an exam for a campus interview. Okay, the rest of them you can go. Shankar. Small book.
Yeah.
ஆமா. ஆக்சுவலா லேட்டஸ்ட் போட்டோ எடுத்திருக்காங்க பழைய போட்டோ இது.
இல்ல சார் பழசு. Welcome back. Dear participants, we are in the final stage of the second day program of the very great event. We have with us Dr. Kannadasan S. He is an associate professor in Department of Chemistry, Vellore Institute of Technology, Vellore. Let me have the privilege of giving you the details of Professor Kannadasan. He had his education in Department of Organic Chemistry, University of Madras, where we together grew as postgraduate in the year 1995 from the University of Madras. And after that, he did his PhD in organic chemistry in the same department and same university. After that, he moved as a postdoctoral fellow in National Chiao Sung University, Taiwan for 1 year. And after that, one more PDF in the National Tsinghua University, Taiwan for 1 more year. With this short introductory note, I welcome Dr. Kannadasan to present his lecture on the topic displayed there. Thank you.
Thank you. So, good evening, everyone. So, I am Kannadasan from VIT. So, I'm going to talk about, directly I'm going to the subject. I'm going to talk about synthesis of potential bioactive N-heterocyclic molecules by using 7-aza-indole and 7-aza-2-indolone as a versatile synthan. So, you know that actually heterocyclic compounds are having more attraction these days, actually in various applications in synthetic organic chemistry as well as electronics, optics, pharmaceuticals. So we have been working long time with the indole. So more recently we have started working on 7-aza-indole, which is a hetero analog of indole in the 7th position. The nitrogen is there. This is a 7-aza-indole. So when we started this work, actually we want to explore the reactivity of the 7-aza-indole. Surprisingly, it has shown a property which is very slower in a reaction rate compared to indole, and also it has given us a lot of fruitful results as well as challenges. So you know that it's aza-indole moiety and its derivatives exhibit various biological activities. There are a lot of precedents in literature. So we have chosen this moiety to explore because we have seen that actually more recently many reports are there showing 7-aza-indole derivatives are very interesting molecules both in pharmacology and biology. So the overall theme of my presentation today is, so I'm going to divide into 3 parts. The part A is I'm going to discuss about synthesis of 5,5-diaryl 3,3' base 7-aza-indolyl methanone, and one part synthesis of 3-formyl and 3-iodo 7-aza-indoles. The part B, synthesis of 3 spirocycloalkenes fused 7-aza-2-indolones via ring closing metathesis. Part C, synthetic transformation of 7-aza-2-indolones and 2-indolones into the spiro heterocycles via 3+2-AMY cycloaddition, as well as mixed Michael addition followed by spirocyclization. So to achieve the first target, so we have taken 7-aza
indole is a starting material, the sequential transformation into that target molecule. This is the diaryl, so bis-indolone, aza-indolone. So starting from 7-aza-indole via N-methylation and followed by methylation to get that bis-indole. These are 7-aza-indoles, and then followed by oxidation of this methylene carbon by CAN, and then followed by the derivatization of, because once we have achieved the target compound, we need to do derivatization. So we have done Suzuki coupling, we have achieved various bis-aryl compound. The second one is, so we have discussed accidentally in the lab. So the same synthesized bis-7-aza-indolone compound, we have cleaved this CSP3 and CSP2 bond, and we have received that, we have got that aldehyde corresponding 3 aldehyde, indole 3 aldehyde and iodo indoles, which these two compounds are very interesting because traditional methods are available in the literature, but it's a little drawbacks are there in those methods. Here very easily we can achieve these targets. Once we achieve the target, so we have done some synthetic transformations into the various compounds. So I'm going to use cetic ammonium nitrate. So why it is used? Because it is the 1-electron oxidant, as well as it is having low toxic and easy to handle and easy to work up. So it is soluble in various organic solvents. So we have selected this as an oxidizing agent, and in the first part of work we are going to use the CAN. So also the literature methods are well known to synthesize this bis-indole derivatives, also the bis-indolone derivative also, but it requires a lot of using a lot of toxic reagents. So in our method we are not using such kind of toxic reagents also. So to optimize the conditions, we have taken that N-methyl 7-aza-indolone and then treated with the formaldehyde, and we have tried with various conditions, temperatures, reagents, everything we have changed, and we have achieved that target in good yield, 85% yield. So using formaldehyde and Monte-Carlo catalyst and microwave conditions. So these are the various starting materials screened, and the scope of the reaction is given here. Various materials have been synthesized, and all the materials synthesized are characterized by spectroscopy techniques and also single crystal XRD. Then we want to oxidize this methylenic double bond, so using a CAN, and we have optimized with various oxidizing agents, and also the solvents are changed, and we have achieved that the excellent yield using CAN as an oxidizing agent. So we have achieved this target. Again, this molecule is characterized by spectroscopy techniques as well as single crystal XRD. So the mechanism might be like this oxidation of the methylene group using cerium ammonium nitrate. We can get that the benzylic radical cation, and followed by further oxidation, and followed by the addition of the nitro oxygen into that cation intermediate, and then finally it can eliminate the molecule of hydride, and then we can able to get this oxygen. This oxidized product. So these are the various synthetic transformation we have tried in this product. So we have done a synthetic reaction with various boronic acid. So we can able to find out the fascinating, so synthesize the fascinating compounds. And so coming to the second part of the work, so we want to get that the aldehyde and iodine. Actually, initially we thought of reacting this molecule with iodine and CAN to check that what is the reactivity. So accidentally we have observed this. There is a cleavage, the sp3CH2 oxidation and sp2CH iodination happened simultaneously within one part. So we can able to separate the product in good yields, and as well as we have confirmed these products by various spectroscopy techniques as well as single crystal XRD. And further, we have derivatized this molecule into that into aryl biphenyl derivatives. But method known for synthesis of 7-aza-indole 3-carbonyl is well known in the literature. Indoles, in aza-indoles also known it is, but it requires a lot of efforts to get that, because the small scale it's okay. When you are going for the higher scale, it's also very difficult. So we found that in the literature, it takes a lot of time, as well as many corrosive reagents. See, for example, POCl3, and then it required a lot of high temperature. In the harsh conditions, we have found that. So we want to find out that the fruit, see, very simple and efficient technology. Always, this iodo compound also, synthesis of this iodo compound also, it requires a lot of efforts. Also, some of the reactions, see that the quenching is taking a lot of time, longer time duration also. Instantaneously, it is not forming. So when we treat this compound, this methylated compound, with the iodine and a can, so we have found that initially the aldehyde, the iodo compound, as well as the traces of this oxidized compound also isolated. Then we have modified the conditions to get that excellent yield in the aldehyde and iodo compounds. So we have got it. So these are the various starting materials screened in this reaction. Also, so these are the various synthesized aldehydes and iodo compounds from this starting materials. So some of the products were characterized by single crystal X-ray diffraction studies. The mechanism would be like initially what I told you, like exactly. Initially that benzylic, the benzylic carbon is getting oxidized into that benzylic radical cation, and then followed by the addition of the nitro O minus into this cation intermediate B, and then follow. Also, the reaction mixture is having that iodine in situ. So synergistically, this iodine also reacting with this intermediate and eliminating one molecule of HI, and then form the intermediate D, which is written the other way. Also eliminate that in pathway. If you eliminate it, we can able to get this oxidized product, which is in traces, right? Also the other way, so it can form the indole 3 radicals and the iodo radical, which can further form the corresponding iodo compound as well as the aldehyde compound. So also we want to get the synthetically transformed product. We have treated with the N-propargylated derivative, a click reaction. We can able to get that triazole product, as well as so we can able to reduce this aldehyde. Also, we can able to get that alcohol, the primary alcohol, followed by that trimethyl orthoformate. We can able to get that ether. Also, so we can make that we can synthesize the imine in situ, and then followed by the reduction, we can able to get this secondary amine product. The iodo compound is further treated with the boronic acid, so we can able to get that corresponding product. All the products have been characterized by all the spectroscopy technique, as well as so some of the products are conformed by the exotic. Coming to the part B, so because I am talking about only the 7-aza indoles as a useful synthan. So this is the second work which we have used as a 7-aza indolones, 7-aza 2 indolones as a starting material. We can able to prepare the 3-spiro cycloalkene derivatives, fused 7-aza indolones derivative. So via ring closing metathesis. Because these spiro compounds, the indole 3 spiro compounds are very essential in biology, and these compounds have shown a lot of biological properties, so which make us to work on this type of molecule spiro compounds. So before going to, for the sake of MSc students who are sitting in the back, just for this slide is for them only. What is ring closing metathesis? So the ring closing metathesis, initially this terminal alkenes or can cyclists together form the cycloalkene. Also the next one is a double bond in a ring can be opened, which is called as a ring open metathesis, and then rearranges the connectivity is called acyclic diene metathesis. Also sometimes the cross metathesis also can able to form. So we have used Grubbs second generation catalyst as well as Grubbs first generation catalyst for the RCM reactions. Previously, we have worked with RCM reactions, and we have published a few works more recently.
So we have synthesized various pyrroles, pyridines, azabines, bipyrroles using RCM methodology, starting from orthoamino amides. So here this can be alkylated, and then we have done the ring closing metathesis, and also this amide NH2 also can be alkylated. We can able to achieve the diazinone derivative. Also, we have reported more recently the ring closing enine metathesis for the synthesis of 1,3-dienic compounds. So based on this experience, we want to do a ring closure metathesis of 7-aza 2-indolone to synthesize the 3-spiro compounds. So for this, we have taken 7-aza 2-indolone as a starting material. We have alkylated in this position. We can able to get that corresponding in products. Once this product is ready, so we have planned for ring closing metathesis using Graf's catalyst. So to achieve the corresponding spirocycloalkenes, so 5-membered, 7-membered. So that is the idea. So initially we have taken 7-aza indolone, and then we have alkylated. We have got that dialkylated product as well as the trialkylated product, and then we have optimized to get that required compound. Once this compound is ready, so these are the various starting materials screened for these reactions. So we have prepared all these intermediates. Also, all are characterized by the spectroscopic techniques. Some of them are characterized by XRD also. So once we get that trialkyl derivative, so we have done that ring closing metathesis using Graf's 2 catalyst. So we have optimized the conditions, and we can able to get that the product in 84% yield only in the case of this 9. So the scope of the RCM reaction is given. We can able to get that various cyclist spirocycle product. Also, noteworthily, we have surprisingly we have got this bis spirocyclic and 18-member ring. So it's a wonderful result. So we have identified and we have characterized this molecule by a single crystal X study. So unusually, when you are using indole, so you can able to see that cross metathesis product. So this type of product which could not be seen in the other intermediates. So these are the various spirocyclic products which are characterized by the single crystal X. This is the 18-member ring. You can see that how beautifully it is happening. So we need to prove that why this is happening. So we have done some catalytic concentration studies also, even solvents also various amount we have tried and how this is happening. So we have taken this and then we have studied with the Graf catalyst. So we are taking solvents also in various amounts, and then so we have achieved that compound 11C, the 18-member ring only in this case of the first case. And then the second case, so we can able to get that another product also, which is nothing but the trimer. It's like a trimer, but we are so we were going to work on this molecule also to cyclize this. So we'll see that, but it's very. So we have to work on this molecule also in the future, and also the cross metathesis product. Only in the case of indole, I told you, indole 2-indolone, we have seen these molecules. So once we got that the 3-spirocyclic product, so we tried to transform this molecule synthetically into the useful molecules. We have tried a Synthesis reaction as well as we have used iodine and CAN to get that iodo-substituted that ether product. So coming to that, the last part, part C, synthetic transformation of 7-aza 2-indolones and 2-indolones into spiroheterocycles via 3+2 azomethane ylide cycloaddition and a mixed Michael addition followed by the spirocyclization. So in this, what we have tried, actually we have taken that 2-aza indolone and then we have made this alkylidine, 3-alkylidine. This alkylidine is right, which undergoes cycloaddition reaction, whether it is cyclic AMY or acyclic azomethane ylide. So we can able to get that corresponding cycloaddition product, which is a spirocyclic product only once again. So they are, spirocyclic products are very important actually in the biology. So we want to try this type of reaction. We have achieved it successfully. And also, all these molecules are characterized. So this way only we have tried this reaction. So this compound is synthesized from corresponding two indolones, and then we have made that alkylidine, and this alkylidine, which is reacting with the acyclic AMY as well as cyclic AMY, which can form the corresponding cycloaddition product. So these are the scope of the reaction, various 3,3' dispyropyridine, bis 7 other 2 oxo-indole derivatives that we have synthesized. All these molecules are characterized. Well, this is the other one. So some of the molecules are characterized by the single crystal X-ray diffraction studies. So the mechanism should be like this, right? The AMI formed simultaneously, it may add into that alkylidine, so which is forming, this is acyclic as well as a cyclic AMI can form the cycloaddition product. Also, once we have done that cycloaddition product, at the same time, we want to try that Michael addition reaction, what is happening. So it is known, actually, some of the indole molecules are known, that Michael addition, triple Michael addition. So we have tried this with this azaindole. So we got that one, so we got that triple Michael addition product. Further, it is undergoing cyclization. We can able to get that 8-member ring. Also, we have tried that one part mixed Michael addition, because the various diversity of this molecule makes a lot of interest in this type of molecules to synthesize. So we have taken that 2-azaindolone, and then the MEK potassium carbonate is a base. So using methanol as a solvent at room temperature, we can able to get this product initially, and then we have stabilized that reaction, and then we can able to synthesize the various triple Michael-addition product. Some of the molecule, one of the molecule is characterized by single crystal X-ray diffraction studies. And then initially we want to try that mixed Michael addition by taking various receptors, so here the condition is the same at room temperature. We tried, we can able to see all these 4 types of molecules, but actually expected molecules are these 2 molecules, which is not happening. Then later we tried, and we have changed the temperature, reaction condition, addition of the Michael receptors, so everything we have changed, and then we can able to get that optimized condition for the synthesis of the mixed Michael addition products. So one of the molecules is also characterized this molecule by single crystal X-ray diffraction studies. So these are the various synthesized Michael, mixed Michael adducts, and then synthetic transformation of the synthesized Michael adducts. So we have tried the intramolecular Alder reaction to get that compound 8-membered spirocyclic compound, and then also there is a conjugate addition product we have tried. So using methanol and KOH, we have got that product in good yield, and also all the products are characterized. The mechanism should be like this. Here the cyclization mechanism is just ketoenol tautomerism is happening. Then once that enol is formed, then it's getting cyclized to get that 8-member ring. So overall, I think am I having time? Right. So overall summary of this work. So we have taken the 7-azaindole as a starting material to explore the molecule. That is our target actually. We have done few works, and some of them are published. So the first work is methylation followed by the oxidation, and then the second work is the cleavage, sp3-CH2 oxidation and sp3-CH iodination as done within one part. Then third one is the ring closing metathesis product. So we achieved that 18-member ring as well as the spirocyclic, various cycloalkene spirocyclic compound might be achieved. And then the cyclic and acyclic AMI 3+2 cycloaddition product with that corresponding alkylidine, and then triple Michael addition product reaction was carried out with this the indolone, 2-indolone, and then mixed Michael addition reaction is also done. So My acknowledgement, first my sincere thanks to the organizers of CRSA LCC, Dr. Jayakumar, Dr. Kalyani, Dr. Suresh, also other organizers to give, because of this opportunity is not given, it's not possible. So I must say my sincere thanks on behalf of my group also. And then my sincere thanks to our Chancellor, the founder of VIT, Dr. G. Vishwanathan. Without his vision, the institute may not be there, and I may not be there right now, because right now I'm working there. He has given a lot of facilities and chances to grow. Then Dr. P. Shanmugam is my mentor. Actually, he is my senior. So when I was in a difficult time, he was the person, is the person taken me, and then because I have came from industry to academic. At the age of 40, I entered to academic. You know that really difficult to get the funding and other things to do the research. Basic academic research is not an easy case. It's very challenging that time. So is the person taking care of me, and then now also all the work. So my students may go to his lab to do some of the experiments, and we can exchange the students also. So we have a collaboration with the CLRI, CSR CLRI. I must thank him at this time. This is my small group. Maybe I'm bigger, but the group is very small in numbers, right? So without these students, it's not at all possible. These 3 girls and these 3 boys. So they are the responsible for this work. Thank you.
Thank you so much, sir. Now the space is open for just 2 questions from the participants. Sir?
Wonderful work. I'm amazed to see that. Really enjoy it. I just have one question where you showed the methylation reaction on indole. It's Savanaza indole. Same reaction also possible only in indole?
Which one?
Savanaza indole, methylation.
Methylation.
Also possible only indole without aza, Savanaza?
It's possible.
I mean, is it reported already?
It's reported. Indole it is reported.
Okay. But in Savanaza, it's not reported.
Yeah. That's why we have taken specifically Savanaza indole.
Yeah.
There's not much work explored in this compound.
Yeah.
So we have
And drug activity study will be very interesting.
Yeah, we have to
see.
do that.
Yes.
So we are doing only synthesis part.
Yes.
So due to the lack of time, we are not done. We are going to do that in the near future.
That will be interesting.
Yeah.
Thank you.
Is that all? I sincerely hope that his speech might have induced a spark in your research career. Thank you so much, sir. I have the responsibility in handing over a small memento from the team of organizers. Thank you so much.
Thank you.
Next on the series, we have Professor V. Ganeshan, Professor, Department of Chemistry, Institute of Science, Banaras Hindu University, Varanasi. Please permit, sir, because I have a lot to read about his accolades and achievements. I have in. Okay. So he is Professor Ganeshan. Professor V. Ganeshan had his post-graduation in Bharathidasan University, Tiruchirappalli, and later on obtained his doctoral degree in MKU, Madurai. Dr. Ganeshan was a postdoctoral fellow in the University of Houston, USA, for a brief period, and later on in the University de Lorraine, Nancy, France. He is a recipient of the prestigious award of the Most Productive Researcher Award conferred by the Banaras Hindu University. With this short introduction, let me have our speaker on stage, please.
போய்க்கலாம் சொல்லுங்க இது நெக்ஸ்ட் இது ப்ரீவியஸ் இது ஓகே வெரி குட் good evening to all first of all I thank the madam for the nice introduction and then I thank the organizers for this dance especially Suresh and Jayakumar. Okay, and I should say for the student sake I should say that I am also alumni of this department. I did PhD From 1995 to 2000, I spent more than 5 years in Professor Ramaraj sir lab. And of course, these days are very, it is a memorable one actually. So I enjoyed this campus and yeah, roaming around, it was very good. And sure you are also enjoying. Right, now when we go to the subject, today I am going to talk something about the selectivity issues, what we are observing or what we modified in the oxygen reduction reaction, electrochemical oxygen reduction reaction. First of all, why we have to reduce the oxygen? So it is for the two. First of all, what is oxygen reduction and what is the process? So oxygen reduction goes in two pathways. One is 4-electron process, which directly goes to the water as the product. Another one is the 2-electron process, which goes to the hydrogen peroxide as the product. So if it is a 4-electron selective reaction, one can utilize this in the fuel cells. If it is a 2-electron reduction, one can utilize it in the for the synthesis of hydrogen peroxide. Okay, so fuel cells also high in demand, I can tell you, and synthesis of this green hydrogen peroxide is also important. So, but it is possible only when the catalyst is selective for either of the reaction. If it is selective, highly selective for 4-electron, okay, best, we can utilize in the fuel cells. If it is highly selective for the 2-electron, we can utilize to synthesize green hydrogen peroxide. So we have to tune the selectivity. How one can tune? This is the one. So this is the, let us say that most of the time for the catalyst, we use catalyst support to spread uniformly the catalyst so that all the catalyst will be active. So this is the catalyst support. And then here we are having the catalyst. That is, in our case, we have taken, in this time, we have taken MN4, that is metal complex with the N4 system, macrocycle, mostly porphyrin, thalossanine type ligands. So this is a metal, metal is the active center. The first intermediate, when oxygen reacts with this catalyst, the very first one is the formation of metal oxygen, means MOOH bond, this one. This is the most accepted mechanism also, and it is isolated also, and it is the well-known intermediate. So in all cases, basic, neutral, in acidic medium, all cases, this is the intermediate. Maybe in basic H will not be there. Other than that, it is the intermediate. So if this MO bond is stronger, then one can break the OO bond, so that 4-electron process will happen. So it is selective for the 4-electron reaction. And then suppose this MO bond is stronger, so it cannot be broken, so that, sorry, if it is weaker, then this OO bond will cleave, so that OOH will comes out and hydrogen peroxide will form. This is the thing. Now, what are the factors going to affect this selectivity? How one can make the MO bond stronger or MO bond weaker? It should be highly weak or highly strong. Then only one can achieve the selectivity. These things I am going to tell you by selecting one or at least whatever. Okay, before that I will tell you something about the fuel cells. So by selecting 3 systems, I am going to explain. Whatever results we got it, based on that I am going to summarize what are the possibilities to improve the selectivity either towards 4-electron or towards the 2-electron. So first of all, what is fuel cell? Fuel cell is very similar to, of course, energy storage device you can say, but it is having just one difference we can say, that you have to pass continuously fuel into it, not like a battery, you have to pass continuously fuel. And another one is that it is not due to the combustion of the fuel, it is due to the chemical reaction. So just fuel and oxygen gives you electricity and heat. Okay, this is the fuel cell, and the emission is only water. So there is it's a good chance, means no CO2 and all. And the fuel cell is having these components. I can read out, but it's okay. So we are mainly concentrating on the cathode catalyst. So there is a catalyst, oxygen reduction reaction will occur, and we are developing material for the cathode catalyst, this one. So I am going to concentrate on this one only. So now, yes, if you consider the fuel cells, one can classify, for the just kind of introduction, those who are not knowing what is fuel cell, for the students I am saying, So one can classify it is based on the H plus membrane. You can say that membrane fuel cell based on the proton conducting electrolyte membrane, or it may be oxide ion conducting membrane, solid. So an oxide ion conducting electrolyte, one can say, one can classify in a different way, but still further you can say, see, proton exchange membrane, alkaline fuel cells, phosphoric acid fuel cell, molten carbonate fuel cells, solid oxide fuel cells, all are possible. But you can see in all cases, oxygen is used as the fuel, and at the cathode, oxygen is reduced. And of course, at different temperature it will be there, that is fine. So again, this is the same reactions. So in cathode, oxygen reduction occurs in all kind of fuel cells. Okay. If it is a 2-electron selective reaction, I told you it can be used to synthesize the green hydrogen peroxide. So what is green hydrogen? One can understand electrochemical synthesis, so it is green. So actually currently hydrogen peroxide demand is very high, and even a prediction says that several $1 billion are involved in this business, means hydrogen peroxide production and transport, especially in the COVID time. There was, everyone is using hydrogen peroxide, at least some percentage of hydrogen peroxide was there in all sensitizers. So it is going to be high. Another point is that hydrogen peroxide is corrosive in nature, so transport issues also there. Suppose any person devises a method to synthesize hydrogen peroxide in situ, means wherever you go and in situ you synthesize, means at the place itself, so that one can avoid the transport issues. So this 2-electron process can be used for the green hydrogen peroxide synthesis. Currently used method is this one, that is a palladium-catalyzed, some anthraquinone devices involved. So it is not environmentally burning, and it is having purification issues also. Of course, in our electrochemical synthesis also, there is purification issues. Still, other than that, the advantage is also there. So in any case, your catalyst should be selective for anyone. If it is not selective, we are trying to be selective towards either 4-electron or 2-electron. That is the point I am going to say. So for that purpose, how I am going to say is that, yeah, these 3 systems I am going to discuss. I think at least one system I can discuss very well, and then if time permits, I will do it. Otherwise, I will go to the comparison where I can compare all the systems. Okay. And yeah, this is the strategy of my talk. So first of all, I will say the synthesis, and then with examples of these 3, and whether it is going to 4-electron or 2-electron, then I will compare and conclude. That's all. So going further, why we have selected the carbon materials as the support. So carbon materials, you can see high surface area, tunable conductivity. Yeah, several factors are there. So one can use it for this one. Mainly, you can uniformly disperse the catalyst on the carbon support, so that all the catalyst will be, almost all the catalyst will be active. So metal atom efficiency will also be high. Okay. So this is graphene. Of course, many of you know it. If you round the graphene in a roll, you can say it is a single walled, and then multi-walled, several carbons, nanotubes one inside the other. So this is our first system I have taken. So in this system, catalyst support is multi-walled carbon nanotubes and 3 different catalysts. So these are the 3 different catalysts we have taken, and it is by simple equilibration method, it is adsorbed on the carbon nanotubes. So first we have taken the carbon nanotube mixed with the corresponding perfluorines in DMF, and then sonication, and then stirring for overnight. The aim is that maximum amount of that catalyst should be immobilized on that one. At the same time, aggregation effect will also be there. Anyhow, it is not at all absorbed very much, so that maximum amount is fine for us. So in all this, after this synthesis, we made the characterization. This is UV visible, of course, neat solution, and it is having some Sorad B band and all as expected. And then this is the XRD characterization, which includes that this is the only MWCNT, that is curve D, and then this is E is only that porphyrin, and then this is the composite D, E. One has to compare for the A, D and E. So if we see that, it shows the sum of the features, SUM, sum of the features of both, that is porphyrin and also carbon nanotubes. So it simply says that integrated. So the remaining 3 porphyrins composites are B and C. And we studied the TGA also. Just I can tell from this one, it is stable up to at least 400 or 450 degrees Celsius. So because certain fuel cells operates in little high temperature, so it is necessary. Okay. After that, further characterization were there. So before 5 minutes, just inform me. Okay. Yeah. So this is the SEM characterization, and this is the TEM characterization. From the SEM, this is MWCNT only, and this is the 3 different composites. Okay. MWCNT, one can see very clearly that MWCNTs are there, and then in the composites, you can see kind of nodes or some dark spots or some crystals are also there. In addition to that, if we see little bit more clearly, let us say, so this is the carbon same scale, 50-50. So comparing A dash and D dash, okay. So this carbon nanotube is there, but here one can see the kind of layer is adsorbed on the carbon nanotubes, and even that length is means breadth is increased. If you see it, one can understand that. So that porphyrin is uniformly adsorbed on the carbon nanotubes, and in some places aggregations are there. Okay, this is the characterization. Further we characterized probably, and then we did the electrochemical studies. So this is the oxygen reduction reaction study. This is the E, curve E for the platinum carbon. That is the standard one or benchmark catalyst or commercially correctly used catalyst. So one has to compare with this platinum so that our material is how much efficiency, like that one can discuss. So this is for acidic medium, and our material, this material shows better results among the 3 composites, and this one is that material adsorbed composite. So when we compare A and E in the acidic condition, definitely platinum carbon is working well, and our composite is not very near, but this is still better than several other similar catalysts. But when we come to neutral and then basic, this is basic. Again, E is the platinum carbon and A is the our material, composite material. Almost exactly same. Onset potential also, current density also same. So one can think of replacing that platinum carbon in fuel cells by using this material. So for that purpose, of course, we proved that it is a 4 electron reduction reaction, and for the selectivity I will come later. So it is based on the RDE studies, and then for that purpose to say that it can be replaced, it can be used to replace that platinum carbon, we compared with several other reported matter. At that time it is published in 2017, so we compared before that, and our material shows either better results or comparable results. So it was okay to say. And then it should be stable also. Platinum carbon, like platinum carbon, it should be stable also. So we checked the stability by running 3000 CVs or applying the ORR region reduction potential for 1 hour or 2 hours or several hours. Then we checked this. Mainly we checked XRD and morphology. Then it shows stability, so it is fine. Now I have taken the another one. That is in this case we have RGO, reduced graphene oxide. So graphene, graphite, graphene oxide, single layer, reduced graphene oxide, then composite, then this one that is coated on the electrode surface. Very simple equilibration technique only. And then after we characterized it, of course, IR, UV and XRD, we characterized it, and then XPS to characterize nickel is in nickel 2+ system, and then okay, thank you. So at least I should skip it a little bit quickly. I could say it quickly. Yes. So these are the SEM characterization and TEM characterization that nickel thiocyanine is integrated on the reduced graphene oxide surface uniformly again. So especially from this nickel, one can understand. Okay. So what about its selectivity? it is the CV characterization, and then it is the ORR study, and it shows 4 electron reduction reaction. Okay. So in the previous case, when we have porphyrin, it shows porphyrin, substituted porphyrin, it shows 4 electron to slightly less selectivity. I am going to compare it. So in this case, nickel thalocyanine on reduced graphene. So catalyst support is reduced graphene oxide, catalyst is nickel thalocyanine. So it shows 4 electron, fine. So then we made another 2 supports. In this case, these are the 2 catalysts. And catalyst supports are mesoporous carbon nitride and mesoporous carbon. So all are playing a role in the selectivity. That's why we have taken different carbon materials and different catalysts. Yes, so it was synthesized using this procedure, templated procedure. We have MCM41 silica material, and we put carbon precursors inside and nitrogen precursors inside the channels, and then carbonize it, and then remove the template by HF or high concentration of alkali. So that is MCN. Very similarly, we prepared mesoporous carbon also, and we studied it. So in this case, tetramethoxyphenyl porphyrin substituted composites shows highly selective 2 electron reduction reaction. Of course, these are the characterizations. Yeah, I am coming to here. Yes, it shows oxygen reduction, efficient oxygen reduction reaction, but at the same time, it is highly selective for the 2 electron. Here we got the question why some of them are 4 and some of them are 2. See, in this case, at exactly 2 electron, almost 2 electron in both acidic and basic medium, cobalt tetramethoxyphenyl porphyrin on MCN. But at the same time, when we change the catalyst support, the selectivity is lost. It going towards the 4 electron. And when we change the that catalyst also, it goes to the selectivity is lost. So, this is the Faraday yield. Of course, that is the produced hydrogen peroxide is analyzed, and then we calculated these are the synthetic yield. Now I come to the comparison. So see, this one we discussed, these 3 we discussed. They show more or less 4 electron reduction reaction. The first one is highly selective. That 0.2 is not exactly 0.2, it is some error in the number of calculation. So we understand that there should be a hydrogen bonding with the MOOH intermediate, there should be a hydrogen bond. Then there is a possibility that the OO bond can be broken, so that 4 electron reaction can happen. Similarly, this hydrogen bond may be formed by different other things. So we have dihydroxytetraphenyl porphyrin. So these are all our work only. We compared because exactly similar system we cannot see it from the literature. So this is the comparison. So here it shows 4 electron reduction, but in basic condition it doesn't show. Probably this also dihydroxytetraphenyl porphyrin, the same tetraphenyl porphyrin, but it is dihydroxy. So in that probably the H is not there, so there is no possibility of hydrogen bond formation. So when we go further and further, yes, in this case also it shows 4 electron in iron thalocyanine, it shows 4 electron in the basic medium, but unusual. This reason we not at all known until now. So there is a problem, but coming further. So this is our first time what we discussed. Okay, again I am saying if MO bond is strong, then OO bond will cleave, so that water will be the product. One can use it in the fuel cells. If it is MO bond is weak, then MO bond will cleave, and then hydrogen peroxide will be forming. That is useful. So far based on these studies, of course I am not able to discuss all the parameters, and we should consider further parameter also. You see, this is our system and this is the substitution. If you are having electron deficient substitutions on the porphyrin, and if there is a possibility of favorable hydrogen bonding formation with the intermediate, then it may go to the 4 electron process. And of course, this conductivity of the support also slightly affects. For 2 electron, one should go for the reverse case. You should not have electron rich, you should have electron rich groups and then non-favorable hydrogen bonding. So this is my conclusion. Of course, already I discussed it, and we have a unique situation in our BHU itself. So all of you invite for the BHU, that is Banaras Indian University, whenever possible. That is the same thing. Varanasi and Kashi are the same. So you are invited, and then I have to acknowledge funding agency, and then this work is mainly abuse. Of course, all the 3 persons were done, and I used to disturb all of them. Sangar, Ratanraj, of course, John is no more, so cannot call. Whenever I get any question in any spectroscopy or anything, immediately I call. Before even search Google, I call them. So I acknowledge them also. So thank you for your kind attention. I'll be happy to give some.
Thank you so much, sir. Thank you very much for giving us a vivid picture on electroreduction of oxygen. Thank you so much, sir. Any questions from the audience?
Yes.
Interaction between the molecules and the graphene.
That is graphene, that pi-pi interaction probably.
Yeah. But pi-pi interaction, how much it is stable? Because you
It
are
is,
applying the power. How much is stable its interaction? Just a pi-pi interaction is sufficient.
Yeah. Pi bonds of porphyrin and pi clouds of graphene having interaction. Because of that, very small adsorption only there, and that is enough for this catalytic studies.
Okay, what is the ratio between the graphene material and your porphyrin? How could you control
we didn't actually calculate exactly, but the ratio in the beginning itself, it was 1 millimole to 50 milligram.
Of the porphyrin?
Of the graphene.
Graphene.
Okay. Graphene or graphene oxide, 50 milligram, 1 milligram of your catalyst.
So you take the pure graphene or RGO, reduced graphene oxide?
Reduced graphene oxide.
Reduced graphene oxide. So if it is a reduced graphene oxide, means it's a lot of the oxygen moieties. Few oxygen moieties there. So the oxygen moieties may disturb the interaction between the porphyrin and this graphene.
Yeah, there must be some interaction that also will affect. Yeah.
Okay, because if such type of the interaction, some disturbance is there, so the reproducibility, the material, not the results, may be difficult. How could you overcome that one if you go for the applications?
Yeah, reduced graphene oxide, it is okay, same reproducible results. Actually, this is produced from, if you are producing and keeping the conditions, everything same, you can get the similar reduced graphene oxide.
That's why I'm asking, because whether the reduced graphene is synthesis in your lab or your commercial.
Yeah, it is synthesized in lab, na? Kamar's method from graphite.
Yeah, I understand Kamar's method, everything. But however, how could you confirm the same reproducibility if each and every time?
If you want to compare, of course, you can take IR, that is EO bond.
IR, it may be confirmed the functional group only, but the presence of the ratios on carbon, that's it.
No, they were based on the property, electrochemical property, it is similar. So we have to say it is same. Similarly, you may have defects also.
Defects, okay.
That's very important. When you have defects, this interaction becomes more stronger, some weaker. Therefore, that is also because it is basically a physics option kind of interaction.
Thank you.
Take one more question.
So I have just some clarifications. You know that adsorption and the pi-pi interaction, there is also the heterogeneity also helps, hydrophobicity. That also helps to keep the
hydrophobicity
molecule.
also will help.
That is one thing. Another thing, there will be a concentration effect, the amount of adsorbed. So that I suppose you might have checked, right?
Yes, sir, we checked it.
That's very, very important.
Our aim is to adsorb maximum amount. So we do it for
But you optimized that the time of adsorption,
Yes,
then the result.
Yes, sir.
Okay, the final, another okay, yeah, these are the
optimized the timing, concentration.
One point I want to mention, either the phthalocyanine or porphyrin, when it is adsorbed as a heterogeneous film, there is a possibility that it might show semiconducting effect. So
Yes,
that has to be checked.
Yeah, it is semiconducting effect. Both are.
In addition to sir's explanation, actually to Dr. Suresh, that when we have reduced graphene oxide, that we have oxygen functionality. In the porphyrin, we had hydroxy groups actually. So they are having hydrogen bonding. In fact, that helps for the electrocatalytic activity. That's why we found that actually hydroxy is showing better catalytic activity. One thing is that hydrogen bonding interaction. Second thing is that hydroxy induces the EC reduction of cobalt 3 to cobalt 2, as well as EC oxidation of cobalt, the redox of cobalt. So that helps actually.
The presence of
the presence of OH. so that we could able to see that good redox behavior in presence of that.
That group forms the hydrogen bond with the intermediate. OH helps to form.
Just one observation finally. Since you have experience in the photoelectrochemistry also you have been doing, right?
Yes, sir.
So why don't you check this electrode surface for photocatalytic or photoelectrocatalytic reduction of oxygen? It
I
will
do,
work.
sir.
It will work.
Definitely, sir.
Yeah,
I'll do, sir.
sure. Both photocatalytic alcohols you try.
Yes,
Electron supplying like a sacrificial electron donors. Then photoelectrochemical also. Both will certainly work.
Okay, sir.
I too have a doubt, sir. Sir, did you encounter any generation of ozone if water is used as an electrolyte?
When we use water,
Water,
we
yeah.
are reducing
Oh,
actually.
yeah. Okay.
Oxygen, ozone cannot form.
Cannot be formed.
Yeah.
Okay. Thank you so much, sir. So we request you to accept the small gift from the organizers.
Thank you.
Thank you, sir. Next in the series, we have Professor A. Pandikumar from SECRI. From SECRI Karaikudi. He has obtained his PhD in Madurai Kamaraj University in the year 2014, and he has been a PDF at University of Malaysia during the year 2014 until May 2016. His research interest lies in the area of design and fabrication of functional materials, photoelectrochemical water splitting and decoupled electrolyzer, interfacial engineering in dye-synthesized solar cells and perovskite solar cells, and the list goes on line by line. He is a recipient of many research awards. He is also an active member in many professional and academic bodies. With this brief note, let me invite Professor A. Pandikumar on the stage, please.
Thank you, madam, for your introduction. First of all, I would like to thank the organizer for providing opportunity to share some of the research in this forum. So I have chosen the topic is photoelectrochemical water splitting, lab scale and beyond. So recently our Indian government announced a national hydrogen mission. So this objective of this national hydrogen mission to India to become a global hub for green hydrogen production and export. So this photoelectrochemical water splitting is one of the methods for producing the green hydrogen. So why we want to concentrate on the hydrogen and hydrogen economy? Why? Because the hydrogen is alternative to the fossil fuel, so it has a high energy density, so which can be used for the transportation purpose. You can easily produce a hydrogen by splitting the water into oxygen and hydrogen, as well as you can easily store in different form like gaseous form, liquid form, or you can store it as a solid form in combination with the metal hydrides. Okay, so based on the production method of the hydrogen, it have the different color code. Normally the hydrogen can be produced from the hydrocarbon. The hydrocarbon is used as a starting material. While producing the hydrogen, it liberates the carbon dioxide into the environment. So this kind of hydrogen production is called as a gray hydrogen. Suppose the emitted carbon dioxide is captured and converted into value-added product, then this kind of hydrogen production method is called blue hydrogen. Suppose if you are having a water, then it will be subject to electrolysis process, then that electricity is produced from the renewable source. So that process is, we call it as a green hydrogen. Okay, so you just to look at this is the kind of electric vehicle. So I don't want to explain about this vehicle in detail because Professor Ganesan already talked about the fuel cell and working principle. So I just briefly explain about this working principle of this fuel cell vehicle. Sorry. Okay. So this fuel cell vehicle contains a fuel cell stack, and there is a hydrogen storage tank, then there is a battery, then specially designed motor, and there is a specific membrane that will allow only oxygen from the environment to the fuel cell stack. Once you ignite the vehicle, the hydrogen, what is the play பண்ண சொல்லுங்க சார். Okay. So this fuel cell having that the one side the hydrogen will approach to the fuel cell, the other side the oxygen will approach the fuel cell. Once the hydrogen and oxygen is combined together, it will produce the electricity. As a byproduct is a water. So in that way it produces the electricity. The produced electricity is used to drive that fuel cell vehicle. So based on that the fuel cell vehicle is work. So in India for the first time our CSIR institute, our like CSIR Sikri in collaboration with the CSIR NCL Pune, together they have developed a fuel cell stack and the technology is transferred to the Sentinel lab for that further validation, and this is the India's first zero emission fuel cell bus that is developed with the collaboration of our institute. Okay. So now the technology for the fuel cell is available in India, but when we are going to achieve that the net zero carbon emission as per the Paris Agreement in the year of 2050, so once the fuel cell vehicle is enter into the market, we have to look at that the hydrogen energy demand. So at the just we crossed 2020, so at the moment the hydrogen demand is the 70 million metric ton per annum. Then if you move to the next decade, it's like a 98 million metric ton. So you just look at this, there is a few color codes are appear newly here. That means this hydrogen open the application in various sector other than the agricultural industrial feedstock. Okay, so in the 2050 we are going to achieve the net zero carbon emission. So that time if you look at this, the hydrogen demand will be 539 million metric ton. Especially in the transportation sector, 154 million metric ton of hydrogen is required. But what is the technology available for the hydrogen production? So in our CSIR system, if any technology we will measure using that the TRL scale. So if you look at this, the hydrogen is available in the market that have that high TRL level. So that is produced from the blue hydrogen and gray hydrogen, as already pointed out. So it is produced by steam methane reforming with carbon capture and storage, or else autothermal reforming. When we talk about the green hydrogen production method, the alkaline electrolysis method is just a matured state. Okay, still it has to go for long way. But what is the demerit of this alkaline electrolysis method, you know? So where they are using the expensive electrodes, anode and cathode. For example, ruthenium and iridiums are very expensive. They are used for oxygen evolution catalyst, and platinum they are using for the hydrogen evolution catalyst. But when we talk about the solar to hydrogen production, it is still in academic level, that is below TRL level 3. So when we make use of this solar for producing the hydrogen, so that is the promising way for the making the green hydrogen when as compared with the alkaline electrolysis. So as already mentioned that in the 2021 our Indian government announced the National Hydrogen Mission. So the objective of this National Hydrogen Mission is India to become a global hub for green hydrogen production and export. So why the government want to invest this much huge money for the National Hydrogen Mission? So these are the deliverables for the National Hydrogen Mission. So from that deliverable, you can increase that the green hydrogen production capability up to 5 million metric ton. Then as per the requirement, you can increase that the renewable energy, because since this electrolysis process that will involve that usage of the renewable energy. So, but this electrolysis process requires the limited amount of electricity. The rest of the electricity is connected through the grid. Then you can increase that the renewable energy power up to 125 gigawatt. Other than this, these are the allied benefits of this National Hydrogen Mission. So in this case, how we can produce that hydrogen from the sunlight? You just take a semiconductor, put it into the water, expose to the sunlight, finally you will get a hydrogen and oxygen. But this simple reaction involves two different steps. The first one is hydrogen evolution step, that involves 2 proton and 2 electron. The second one is oxygen evolution step, so that involves 4 proton and 4 electron. So overall, you want to split the water, you need 1.23 volt of energy. But involvement of this 4 proton and 4 electron, it is a rate determining step. If you make this step is more facile, then you can come out with the efficient hydrogen production rate. So how it is works? So the semiconductor has that valence band and the conduction band. Once it is exposed to the sunlight, the valence band electrons promoted to conduction band, and it creates the holes. The holes oxidize the water and produce oxygen and proton. This proton is moved to here and it accepts the electron. Finally, it come out with the hydrogen. Okay, so as already known that the photoelectrochemical water splitting, we are mainly using the semiconductor as the photoelectrode. So based on the usage of the type of semiconductor, for example, if you have a N-type semiconductor, you can use it as a photoanode. That there you will get a water oxidation process. As a complementary reaction, you will get a hydrogen. Suppose you are having a P-type semiconductor, it will used for the water reduction. As a complementary reaction, you will get a oxygen evolution reaction. Suppose you are having P-type and N-type, you can make a tandem cell. Simultaneously, you can produce hydrogen and oxygen. Okay, so this is the first report, is reported by Honda and Fujishima. Together they have developed a photoelectrochemical cell for the hydrogen and oxygen production through the photoelectrochemical method. So this is the criteria we are applying for the developing of visible light active photoelectrodes. So first criteria, we are applying the suitable optical property. Since the solar radiation having that 46% visible light is the predominant, so that we are choosing the material which will absorb in the visible region. Then the second condition, the band gap position. So the band position must be above and below the hydrogen and oxygen evolution potential. The third criteria, once it absorbs the light, it readily undergoes charge separation. Finally, the promoted electron is moving towards the current collector. That is the other criteria. Finally, the stability is the crucial role. So accordingly, we are choosing the catalyst. So our interest is bismuth-based material. So why? Because the bismuth-based material, if you look at that, the valence band is below to that oxygen evolution potential. Since as already mentioned that the oxygen evolution reaction is the rate determining step. So if you make that the oxygen evolution reaction is more facile, then indirectly you can improve that the hydrogen production rate. Okay. So if you take that the pristine material, it suffers from the charge recombination process. So these are some of the alternative strategies we are adopted in our lab. So I just skip this introduction slides. Okay, so I will just give one example. So we have developed the bismuth vanadate and copper oxide based PN heterojunction for photoelectrochemical water splitting. So in this case, we have done that the preparation of bismuth nanoparticle through the electrode deposition, followed by we have added that the vanadium based precursor over this subject to the calcination process. Finally, we come out with the bismuth vanadate electrode. Then again, the copper oxide is deposited over the bismuth vanadate through the electrode deposition method. Finally, we created the heterojunction material. So this is the XRD pattern. It clearly reveals that the prepared materials are highly crystalline in nature and it forms that heterojunction, but the copper oxide will not disturb the crystalline property of the bismuth vanadate. Then this is the FESM image of the prepared bismuth vanadate electrode, and this is the heterojunction electrode. The elemental mapping, it's clearly represent the corresponding elements present in the photoelectrode. Then the heterojunction formation is confirmed through the XPS analysis because here you just look at this. This is the pristine material. After depositing the copper oxide over, the binding energy values are shifted somewhat. So this is the clear evidence for the clear interaction between the bismuth vanadate and the copper oxide. Then the optical properties also studied. Then finally, we have done the photoelectrochemical water splitting. This is in the absence of light. Once it is exposed to the sunlight, the photocurrent density is immediately increased. Then we want to check that whether the system is reproducible or not. For that, we are chopping the illumination frequently. So the rise and fall in the photocurrents are stable. So that reveals that the fabricated materials is highly stable. Then the durability also we have checked. Sorry. இல்லை வரல சார். ஓகே, இல்லை தெரியல, அது அப்படியே ஜம்ப் ஆகிட்டு இருக்கு. அங்கே ப்ராப்ளம் இருக்குங்க. Okay. Then we have recorded the Nyquist plot through the impedance that shows that the smaller semicircle, it shows that the less charge transfer resistant and high electron transport property that it is revealed from the Nyquist plot. Sir, திரும்ப same problem. இதுலையா? Okay, okay. Then further the performance is improved by adding the sacrificial agent with the electrolyte. So we have added the sodium sulfate, then we got that the enhanced photocurrent as well as we have choped the illumination. So this time two-fold enhancement we have observed. Then the Morsky plot we have recorded, and the XPS UPS spectra also, valence band spectra we have recorded to prove the mechanism. Based on the value, we have plotted that the band position for the bismuth vanadate and the copper oxide. This is the before contact of bismuth vanadate and the copper oxide. Once it comes to the contact, so it will undergo Fermi level equilibration. On the interface, it forms that internal electric field. So accordingly, here we are able to form that the PN heterojunction between the bismuth vanadate and the copper oxide. Then the copper oxide is easily oxidize the water and produce oxygen. As a complementary reaction, we are able to get that hydrogen indirectly. Okay, the stability also we have checked up to the 60 hours. It shows that the excellent stable, and before and after also we have done the morphological studies. There is no much change in the morphological. So we have worked with 2 different methods. So normally we prepare the nanomaterial as a powder, then coated over the electrode. Then the second attempt is like a direct fabrication of thin film electrodes over this. Then it's concluded that direct fabrication of electrode, it will be more facile and makes that higher activity as compared with the powder processed method. Then we used this methodology for making the prototype device. Okay, so at the lab scale, we start with 1 centimeter square, then we move to up to 25 centimeter square. So this is that bismuth electrode via electrode deposition. This is after forming that bismuth vanadate electrode after the thermal and chemical treatment method. Then this is what we fabricated the prototype photoelectrochemical cell in our lab. So we used a quartz window for the illumination, and Nafion membrane is used for the result of this particular electrodes. Whether that 25 centimeter square electrode is behave like a 1 centimeter square electrode. So we have converted the current into current density. So after converted into current density, you can see that there is a huge difference in the current. That means the loss, while we are increasing the area, we are able to get more loss in the performance. So then we decide that we want to change the fabrication pattern. So that's why rather than going for the plain deposition, we are going for the line pattern and the grid pattern. So this time we have investigated the performance. So this time we are able to control the loss, energy loss up to 27% of that the grid pattern, the prototype cell. Finally, we add that the sacrificial agent, then the reduced the loss. So we are able to get 0.61 milliampere per centimeter square in the full cell. Then we avoid the loss less than 20%. Yeah, I'm going to finish. Okay. Okay, then the durability in the full cell also checked with by applying the different voltage versus the platinum. Since we are using that platinum as a counter electrode, this is the working electrode. So here we are able to achieve high protection rate with 1 volt versus the platinum. So we are able to get 12 milliampere per current for the 25 centimeter square the prototype device. So this is the summary of the presentation. So while we are going with the grid pattern with the 25 centimeter square in the presence of sacrificial agent, we are achieve the high hydrogen production rates, as well as we are able to protect the efficiency loss less than the 20%. So this is my current group member. So these are some of the group alumni. So finally, the acknowledgement for the funding agencies and our institute director and my head of the department and my students. Thank you.
Thank you so much, sir.
Thank you for your attention.
Thank you so much, sir. We welcome 1 or 2 questions from the audience. Thank you, sir. As there are no more questions, we welcome
Thank you,
Professor Pandikumar to receive this small gift from us. Thank you, sir. Next on the line, we have very, very young scientist, come researcher, Dr. Suchitra. Last is me. Okay, anyhow, sir. Sorry. We have Dr. V. Subramaniam on the next as a speaker. Yeah. We have Dr. Subramaniam from IIT. Thank you.
Very good evening to all of you. And I would like to thank the organizer for having me here. And it's a very intense program. I think it's packed local organizing committee meeting. It is, if you look at this, the local chapter committee, right, it's more lectures than the regular CRSI meeting. I think hope somebody in the audience who have attended many CRSI will agree with me, right? So previously I was associated with CLRI, now I am with IIT Madras, right? This is the current status. I thought that I will expose this audience to density functional theory, right? I won't bore you with more mathematics here, but I will show some equations. And in the end, some applications. Maybe I may have a lot of slides, but I will restrict only to density functional theory and one application and leave you here. What is density functional theory? Now, we are all familiar with the density, right? Mass by volume. But what is electron density? Right? Many are doing X-ray diffraction. You collect the structure, you collect the details, right? Structural details are obtained from X-ray, right? The information that you gain from the X-ray diffraction is called the electron density, right? I will tell you what is that. And in density functional theory, many of the students, in fact, when I organized the first Gaussian meeting in this country, right, about 150 students have attended. So this is one of the largest event in the overall Gaussian workshop, all right, even today, all right, this number has not exceeded in anywhere else. This tells you that most of the students are using Gaussian packages, all right. But what is inside, how they are using those numbers to understand some of the chemistry is really a question. I thought that because most of us use density functional theory, I want to tell you what is density functional and what kind of functional can be used here. But I may not go into that detail, but at least I thought that I will tell you that there is something called Jacob's ladder. So this is the Hartree-Fock world that we do in our quantum chemistry, right? In our regular quantum chemistry, those of you read Levine or any other Indian version of Levine or any other book, there will be a chapter on Hartree-Fock. So that is the first fundamental assumption that you come across. But from here, we want to go to chemical accuracy. What is chemical accuracy here? Meaning that you want to get any number, any prediction within 1 kilocalorie per mole. We want to have a number within 1 kilocalorie per mole, right? That is the chemical accuracy. That is a heaven. We are here somewhere in the world, but in order to achieve this, right, in this raising ladder, you will have several approximations. You are all familiar with LDA. It's called LDA, meaning that you may see in most of the papers in computational details something called LDA. What is LDA? LDA means that it is local density approximation. Then you will see that, sir, I used GGA. What is GGA? GGA is, now if you see that it is NR, NR means it is rho of R, it is density. And here you will see that it is GGA, gradient corrected method. GGA means you are taking the derivative. Delta means it is a derivative, delta of the density. Then you have a meta GGA. What is meta GGA? You will have a del square, second order derivative of the density. That is the meaning of the correction to the kinetic energy, right? Then you have a hybrid method. This is, you include the part of Hartree-Fock correction to this density functional theory. It is called hybrid, and so on. There are several levels of approximation to get a good number which can be used for understanding the chemistry. So what is, why we have this problem? The problem is we have a Hamiltonian. Hamiltonian is, you will have the nuclear kinetic energy plus electron kinetic energy plus nuclear repulsion, nuclear electron attraction, and electron repulsion. And the kinetic energies are simply, it is p square by 2m. P square by 2m is nothing but del square. Del square gives you information about the kinetic energy. That is why the del square terms are included in the calculations. Basically, we start with a Slater determinant. In a Slater determinant, I thought I will explain you, because this is told in the class, explained in the class, but we miss certain points. Now, if you look at this, if you look at the row, each entry means it is electron, number of electron. Each column represents the orbital of the system, all right? So, this is orbital 1, this is orbital 2, this is nth orbital. This is electron 1, this is electron 2, this is electron n. Therefore, n electron determinant is the fundamental assumption of the wave function. You start any calculation with this. In fact, these are all printed in any standard packages, maybe Gaussian packages, maybe ARCA, or whatever the package that you use, this wave function are printed here. But what is density? Our aim is to show what is density. All right. One of the important problem here is what is called electron correlation. What is electron correlation? Why it is important? Now, when you form this kind of a wave function, right, the electron in, I have one electron here and another electron here. This A electron and B electron, they are seeing each other, all right. When they come closer, then they repel. It is a simple Coulombic repulsion. We are all familiar with that. You have two charges, that of electron in space, their repulsion is called the electron correlation. So, what is our fundamental assumption? We all use Hartree-Fock approximation, and E0 is the total energy, exact energy of the molecule. This difference is called the electron correlation. So, Loudon in 1959 called this effective difference Fock, or this is the error which arises because of the determinantal starting point, determinantal wave function that we start here. This is also called dynamical correlation. Why dynamics? Because you are instantaneously in the space looks for how do they move each other. It's called a dynamical correlation. There is something called static correlation. The static correlation is you are considering a determinantal wave function as a starting point for all the calculation. Therefore, it is called static correlation. Therefore, two kinds of correlation you will see in the most of the papers. One is static, another is dynamical correlation. So, we want to, this is a many body problem. So, in the standard conventional ab initio calculations, we use Hartree-Fock and post-Hartree-Fock methods to understand that. There is another branch of science started from Kohn-Sham theory, which we are started from Fermi theory. Fermi-Thomas model is the density functional theory. It was developed by Kohn-Sham in the later years. That is another way of looking at the many body interactions in system. Now, why this is important? Even if you look at this, a simple hydrogen molecule, textbook example, we all study this, right? It is there in Leuven, it is there in most of the quantum chemistry book. And this is the potential energy surface drawn for the hydrogen molecule. And you will see that this curve is a restricted Hartree-Fock curve. And if you see this is unrestricted Hartree-Fock curve. and you will see the third one is the exact curve. When this is get extended, that is that bond length between H and H increases, there is a tremendous increase in the error due to the approximation that we make use of it. This error, you call it as the electron correlation error. That too, when you stretch it, correlation error, which is almost equivalent to 0.04 in Hartree, right? This 0.04 Hartree means 1 Hartree is 627.5095 kilocalorie per mole, right? It's a large number in terms of chemical units. When you stretch the same H2 molecule, the error becomes 0.25 and it is 627.5095. If you multiply, again the error becomes much larger. Therefore, this we have to take care of this error some means so that you get a meaningful number from the calculations. That's why we go to post-HF methods or density functional theory methods. Why density functional theory is this. The nature of wave function, I already said. The nature of wave function, that what we have is a Hartree-Fock level, and this leads to some kinetic energy increase and strong nuclear electron interaction because the calculated bond distance is shorter, right? When you have a shorter bond distance, the interaction becomes much stronger, kinetic energy increase and nuclear attraction is increasing. Therefore, there is an error because of the nuclear because in the molecule because of the representation that you use in terms of the Hartree-Fock. What is electron density? Therefore, this when we started this kind of a work, density functional theory, people thought that this will be a real alternative for the Hartree-Fock kind of a treatment. What is density functional theory? I thought that I will explain you what is density. And it's not a very great thing at all, but because we study this quantity. All right. So we know what is wave function, and we know what is square of wave function. The square of wave function we call it as a normalization. All right. The integral of the square of the wave function you call it as a normalization. What is density basically? What we do here is we integrate over all the space, over all the spatial coordinates, except the point at which you want to locate what is the density in the space. Therefore, I will integrate over all the space here. This S1 is a spin coordinate, and you will have a x2, xn, all these are the spatial coordinates. I integrate over all the space, right? And except also spin coordinate. There is integral theorem tells you that integral of alpha alpha equal to integral of beta beta is 1. They are normalized, and alpha beta is 0. Up spin, down spin integral is 0. All right. Alpha and beta are the spin functions. All right. Therefore, the electron density is this. It is the probability of finding any of the n electrons within a given volume element R1, but with any arbitrary spin. It can be alpha spin or it can be a beta spin, while remaining n minus electron can have arbitrary positions and arbitrary spin. This is called the electron density. Basically, it is nothing but the probability. This is also called in literature, it is called electron density. In the literature, it is called charge density, all right? But we know what is probability. See, we know from the quantum mechanics, postulates of quantum mechanics, state of the system is represented by psi, and the next one is a linear combination, then third one is integral psi star psi equal to 1. Therefore, this is that. But you eliminate one position and look at the density in the remaining position. That's what it is. Therefore, it is called electron density. What are the properties of electron density? When R goes to infinity, electron density becomes 0. The integral of electron density over the space at which it is calculated as the number of electrons. This itself is a very, very important point. This is called the n representability in the density functional theory. Unlike the wave function, this quantity, electron density, is experimentally observable and can be measured by the X-ray diffraction. If you look at this quantity, it is n variable actually. We'll have a x1, x2, xn. But if you look at this, if you look at the electron density, all right, electron density is a 3 variable, rho, x, y, z. It is only the 3 Cartesian coordinates. Right? It gives you the information about the structure of the system in the X-ray diffraction. Therefore, this can be experimentally calculated or experimentally understood. the number wise it may not match, but the profile is the same. So, electron density has certain properties. I may not go into the details, but it is the electron density map for H2O molecule, and you will see that a maxima on a oxygen atom, there is a slightly less maxima on the hydrogen atoms. This is the other view of the electron density in the case of the water molecule. All right. So, this is a kind of a picture that you will get for all molecules using this. But we are interested in worrying about what is called a pair density. Why pair density? Because I said that I am interested in mapping electron-electron interactions, instantaneous electron repulsion between two electrons of alpha spin or alpha beta spin, which is very, very important. Therefore, what I have is a pair density. So, I have identified one electron, let us say x1 I have identified. So, I will have n minus remaining electrons. Therefore, how many pairs can be formed? By choosing this x1, I will form n minus 1 pairs. Therefore, this number, the normalization constant will be n into n minus 1 of this integral. It is an n-dimensional integral here, n minus 2 dimensional integral, because I am keeping 2 points constant, I am not integrating over that space, only n minus 2 times I am integrating. This is called the pair density. This gives me information about how two electrons are moving in space with respect to each other. In turn, it gives me some idea about what is called electron correlation. Right? So, this electron pair density is a very, very important quantity. It is symmetric in the coordinates and normalized to the total number of non-distinct pairs, unique pairs of n into n minus 1. So, this is the pair density. The pair density can be written like this. This is the independent electron density of that one, independent of that x2. Right, since it is an independent particle model, this density can be combinedly represented like this, product of the density. Meaning that if I know the density at one position, the density at the other position will be reduced by a factor n minus 1 divided by n. So, this the pair density can be represented like this. And let me not move more into this, but to tell you what is very, very important one, we have something called the correlation. Now, do you understand? If you put the pair density, we have seen that this is the product of the electron density at the position 1, product of electron density at position 2, and there is no correlation. How do you include the correlation? I introduce a correlation between these two electrons in terms of a function. Here, it is f of x1, x2. This function is called the correlation factor. When this correlation factor is 0, there is no correlation. But if there is a correlation between these two, this quantity definitely creates, because it is not the simple product, there is some function which is there. This function is very, very important, but this way of representation or definition is wrong, because this is not the pair density. We have seen that it is integrating to n into n minus 1. But when you integrate this quantity, rho to quantity, using this particular definition, it is not integrating to n minus 1, n into n minus 1, but it is going to n square. It should give n into n minus 1 by definition, but it is not giving. Therefore, there is an error. This is an unphysical interaction that is taking place in the definition. This is called the self-interaction correction or self-interaction energy. There are several ways of doing this. How to avoid this? To avoid this, we define what is called a conditional probability. What is this? I know the probability of occurrence or the electron density at position 1, and I want to calculate, I know the probability of occurrence of combined probability of occurrence of x1 and x2 or electron density of x1 and x2. This quantity is called the conditional probability, and when you put all these into that, this will give you the integral of this will give me because this integral will give me n and this integral will give me n minus 1. Therefore, it gives a number n minus 1. Therefore, overall this quantity gives a correct integral value. This quantity hxc rho 2 divided by rho x1 minus rho x2 is called the exchange correlation term. So, most of the all these are built into what is called a universal functional. People still trying to get a good functional which can give a uniform picture of all the chemical phenomena. That's what our idea is, to have one function which gives all the phenomena of chemistry. But it is not happening in density functional theory. We have to take several functions to get some number. Right? So let me move here. This is some idea that I gave, a flavor of electron density. I will take only one example of electron density methods in chemistry and how to understand that. Only one example. With that, let me close. Right. What I am going to show here is how do you apply this to a variety of, you can apply density functional theory to various applications. One such application here is, we used this for aromaticity spectroscopy relationship in conjugated polymers, all right? Or we can use this for designing any photochemical sensitizer, or we can develop new molecules for TIDF applications and so on. There are several applications we can use this technique to understand variety of phenomena here, all right? So this is, this slide has been shown by many people yesterday. I don't want to repeat this here, all right? But this organic molecules are very, very important. People have been talking about in this particular forum several times about the importance of organic molecules and its variety of applications. Previous speakers have mentioned about that. Ganeshan has mentioned about that. Pondicherry has mentioned about that. These molecules are very, very important, all right? But we looked at certain concepts like aromaticity. Aromaticity is mentioned in most of the organic chemistry, but it is quantified in various ways, but there is no unique quantification for the aromaticity. There are several ways. I said one of the ways is the nucleus independent chemical shift. All right, that is a technique that we use here to get the quantification of the aromaticity. All right, so we want to develop the relationship between how the spectroscopy properties of the molecules are influenced by the aromatic nature. Is it possible to have some relation between these two, so that if I can predict aromaticity of some combinations of molecule, can I have some understanding of the spectroscopy property here? All right, we are all familiar with Huckel aromaticity, Mobius aromaticity, spherical aromaticity in the case of fullerenes and so on. There are several things are said in the chemistry literature and organic chemistry. Control this aromaticity, there is a control over the HOMO-LUMO. HOMO-LUMO energy is high. The gap is crucial for designing the optoelectronic materials, especially based on the organic molecules or even in inorganic systems. It is one of the central concepts in organic chemistry. All right, it plays an important role in photochemical systems. But how do you understand their importance? It is a concept. The quantification is a questionable one. But when you select a molecule with aromatic character and combine them, then you have a conjugation. The conjugation is a structural consequence. This is benzene, biphenyl, thiophene, bithiophene. These kinds of small molecules are used in variety of combinations in organic molecules. The aromaticity is a concept I mentioned. Conjugation is a structural consequence, but these are observed in spectroscopy. What is the relation connecting aromaticity, conjugation, and spectroscopy features? That's the question that we asked in our group, right? So for quantification of the aromaticity, we have used a technique called the NICS, nucleus independent chemical shift. All right, this is illustrated with the help of a benzene here. This is a benzene, all right, C6H6. All right, what we do here is we keep some hypothetical atom here, red atom, at various distances from the center of this. All right, so 0, 0.5, 1, 2, 2.5, and so on. Therefore, you calculate the chemical shift at various distances from the center of the benzene. This quantity is called the nucleus independent chemical shift. This can be calculated for any molecule. What we put here as a ghost atom does not have any electronic effect, right? We see only the effect of the ring current on this particular atom, right? It is a ghost, no electronic effect here, right? So this approach is called, this is due to the Schleyer, one of the famous organic chemists in the world, right? He is no more, but he is the one defined this index. There are several index for the aromaticity. So what we did is we developed several molecules, all right, of this nature, right. We calculated the aromatic index at the centers of these molecules. It's only using this technique. This is the Gaussian package that we use, all right. We calculate the Nix value. Not only that, we also calculate the reorganization energy, hole as well as electron reorganization energy we calculate. And what we do is we have done all these calculation, and these are the homo-lumo gaps of the molecules, all right, and the values have been calculated, all right, various conformations have been searched, all right, and what we plotted here is we calculated the total Nix value as a function of the 1 by n. That tells as the number increases, it is a polymer, right? As the number increases, how this Nix value varies, all right? And then what we have done is how this 1 by n correlates with the homo-lumo gap of the polymers that we have constructed, all right? Not only with that, we also looked at the relationship between what is the first transition energy between homo-lumo transition, all right, that energy as a function of the 1 over Nix, how the aromaticity influences. Therefore, you can see that there are certain cases there is a linear relationship between 1 over Nix with the first transition lambda max, all right. In some cases, we are not observing. We have to look at why it is not happening. It is it depends on the what kind of a linker, what kind of a bridge that you use between the two donor and acceptor units. When you have a benzene unit, this kind of a relation. When you have a thiophene, you get a more reasonable correlation that you get. Not only with that, we also correlated the outcome with the reorganization hole as well as the electron reorganization energy. In some of the cases, you can see that there is a good correlation. In some cases, it is a scatter plot. Therefore, in a nutshell, what we have done here is we have used the density functional theory to understand certain important aspects. Is basically the how aromaticity influences the structure, and from structure how the spectroscopy property is altered. Right. With this, I stop here. I thank the organizer for the wonderful opportunity to talk to you all on this day. Thank you.
Thank you, Professor, for a wonderful talk. Maybe quickly, maybe 1 or 2 questions. Any students? Anyone? If no more questions, first, we will congratulate and
thank you,
Professor. Maybe you can interact. You will be here tomorrow also. Yeah,
thank you.
Anytime you can talk. Yeah. Sir.
Yeah. Thank you.
Thank you, sir. Thank you very much.
Thank you.
The next speaker, Dr. Susitra Madhavan from IISc Bhopal. The last speaker of this session. She has done a PhD from Dr. Shanmugam's. Right now, he is in CLRI. She is a chip scientist. She has done several postdoctoral fellowship. Like, she was a research associate from IIT Madras, and then postdoctoral researcher in Kangwa University, Japan. Then she moved to postdoctoral for IIT Robar, India, under Professor mentor with Avid Goswami. Currently, she is a DST women scientist in IISc Bhopal. With these few words, I invite Susitra Madhavan to deliver it. Okay, thank you.
Hello. Thank you, sir, for your kind words of introduction. And yeah, so I can see everyone is getting saturated with a lot of chemistry in the evening, maybe organic, inorganic, material chemistry, and so on. Yeah, please hold on. Some interesting chemistry is to be discussed. So few, at least 20 minutes. Okay, so I'm thankful to Professor Ravi Kumar. He gave an ample introduction to CH activation and Catalani approach. So I will not be spending time on describing about the CH activation and all. But I am here to present my first independent work it is strictly on distal CH functionalization based on 2 methodologies, and it especially in ferrocene. So the title is Distal CH Functionalization of Ferrocene, the synthesis of ferrocene 1,3 derivatives. So I will not be giving an introduction to CH activation, so everybody will be knowing about the CH activation as a very important synthetic tool for synthetic organic chemists to functionalize an organic molecule because of its ubiquitous nature in organic molecules. So it is advantageous and bit challenging also, because there are a number of CH bonds along with the heteroatoms and functional groups. So selectively functionalizing one CH bond is a challenging task, right? So there are these are the challenges, mainly the lack of reactivity due to the large kinetic energy barrier, and the lack of selectivity due to the ubiquitous nature of the CH bonds. So transition metal chemistry came up as an intriguing potential solution, one of the solutions for this dealing with this problem. So strategies for transition metal catalyzed distal CH functionalization before going to the details. So transition metal catalysis with the help of a dialectic group, I was able to functionalize selectively a CH bond. So usually this is by proximal CH activations, that is via thermodynamically stable 5 or 6 membered metallo cycle. So that is selectively activating this ortho CH bond in case of arenes. Okay. So, and what about the other CH bonds like 2 carbon away or 3 carbon away from the functional group? So it is a challenging task, right? So here comes the distal CH activations. So here the CH bond that is 1 carbon away from the functional group and activating selectively this bond is quite challenging, because these distal CH activations are on the meta or in a para position in case of the arenes, it's usually via 12 to 14 membered cyclophane like a transition state, that is highly energy demanding transition states. So, but people have worked on this one, especially in case of arenes, using many methodologies like template. Template is nothing but a carefully engineered directing group that carefully it is engineered so that that metal can specifically go to this meta CH bond and activate this one. Or in the para, if it is carefully architecturally arranged, that it can go to the para position also. There is a very fantastic review by Maiti and co-workers in Science, so you anyone interested can go through that one. So another one interesting is the distal CH functionalization via transient mediator. So as Professor Jayakumar has explained this one, but this is a little bit different. I am interested about the distal CH functionalization. So by Professor Martha Catalani, she introduced this reaction using a transient mediator. Here the norbornene with palladium. So this palladium norbornene cooperative catalysis is used for the ipso and ortho functionalization of aryl halides. So after that, from Professor Jingwan Yu of MIT and Professor Dong, they independently used this, cleverly used this methodology for along with the CH activation for both ortho and meta CH functionalization reactions. So this is now is called a Catalani approach or Catalani type reactions, which it involves a CH activation, then it is a Catalani type reaction. So another one is using a traceless directing group like acid. It can be already it is present in the substrate as a carboxylic acid, or in situ you can introduce this by using the carbon dioxide. Many groups utilize this methodology. Anyway, this weakly coordinating carboxylic acid will do CH activation, direct the metal to the next CH bond, and in the same reaction pot it will get cleaved. So this will is a triceless directing group. So finally you will end up on the meta CH activations, 1,3 derivatives you will be getting, right? So another one is CH functionalization via non-covalent interaction approach. Mainly Phipps and other group, mainly Phipps used this methodology for ion pairing interactions. For example, the directing group has like a secondary amine, that quaternary ammonium salt has been produced, and the ligand is having an anionic side. Then the ion pairing interaction will help to deliver the metal to the meta positions selectively. They have achieved and published many papers. But the problem is that this is mainly in case of arenes and only borylation using B2Pindu mainly. So borylation of the arenes using this type of bulky bipyridine ligands. Later Chattopadhyaya and Kanai groups used this type of approach using these hydrogen bonding interactions to deliver this metal and getting activated the meta CH bonds. So this is the mainly the strategies for distal CH activations. So this is the main review. You can go through that one. Different. It's a comprehensive review. So coming to the ferrocene chemistry. So the ferrocenes, after since its discovery, we know that it's an accidental discovery of this ferrocene. Since its discovery, this funny organic organometallic molecule have attained enormous attention because of its applications, mainly as a ligand or a catalyst. Very recently, this is acting as an organocatalyst. It's both a redox tunable and a normal organocatalyst, and a number of ligand and catalyst has been designed by using this organometallic scaffold. For example, this xylifos is a 1,2 ferrocene derivative. This is used for the enantioselective selective synthesis of metal-laclode. That is one of the largest asymmetric synthesis that has been used for the metal-laclode, that is a herbicide, for the imine hydrogenation using the xylifos ligand industrially. So that is one of the large-scale industrial productions using asymmetric catalyst, iridium xylifos ligand. And this is a 1,2 ligand, right? So DPPF, everybody knows that one. This diphenyl phosphinoferrocene that has a remarkable place in cross-cutting reactions, and there are many. And this is an interesting pincer ligand, the 1, 2, 3 substituted ferrocenes. And then coming to the drug and biomolecules and material properties of ferrocene scaffold. So particularly this ferrocene, we have seen this 1,1 dash ferrocenes, 1,2, these are 1,2 ferrocenes, and this is a 1,3 ferrocene. And this is a blood glucose level monitor that is also commercially used. This is a 1,3 ferrocenyl molecule. And this one is a ferrocenyl trisulfur triiodothreonine, and this is a 1,2 derivative. Ferroquine is an antimalarial drug, I think is phase 2 clinical trials in combination therapy. And these are the thermotropic liquid crystal having important material properties. These are 1,3 ligands. So this 1,1-1,2 ligand synthesis is mainly explored because mainly the ferrocene monoderivative, we know this at textbook chemistry by Friedel-Crafts reactions. We can introduce any functional group in the ferrocene by virtue of its electronegative nature. So after the monoderivative, the introducing one of the, substituting one of the ring positions, it's usually done by lithiation. And chiral induction is done by using lithiation reactions or using a chiral base or a chiral auxiliary. So 1,3 derivatives are a very unique class of ferrocene derivatives in this class, which is actually there is no catalytic method to prepare these 1,3 derivatives still now. So these derivatives already prepared by 1, 2, 3 substituted ferrocene derivatives. I will go to the details of this one. For example, Hafner and group in 1984, they prepared this small amount of 1,3 derivative by the fulvenes, suitably substituted fulvene, then metal complexation. That is one method. Then Weissensteiner, they used produced 1, 2, then 3, 1, 2, 3 substituted, then consecutively 2 lithiation procedure. Produce the 1, 2, 3 substituted and knocking out the second one. So it will end up into the 1,3 derivative. So it needs a lot of harmful reagents, mainly organometallic reagents, and consecutively 2 steps we have to use, and yield also not so good. So coming to the other one, Airbud co-workers used this halogen dance. That's a disproportionation type reaction. So iodination, then another radiation will lead this iodine to the 3rd position. Then it will end up in a mixture of products like 1, 2 and 1, 3 products. And these ferrocene chemistry who is dealing with, they know that separation of these different products is a tedious task. So these are not very reliable. So they can prepare in a small amount, but there is no general method for the synthesis of 1,3 derivatives still now. And this one is also use S-butyl lithium, then 1, 2, 1, 1 dash also derivatives also simultaneously formed. And if iridium by B2 that bulky system we are using, then in case that also 1, 1 dash is also the side products. So what I am telling that there is no general catalytic method for the synthesis of 1,3 derivatives still now. So this our hypothesis is that any commercially available ferrocene derivative, the functionalization of the 3rd position of this ferrocene moiety, that is so as I discussed, this is an unknown chemical space that is not used by any method. So can we functionalize the 3rd position? So it's a little bit making a little bit interesting, like a cartoon. So I prepared like this. I'm sorry, there is a problem with the Windows and Mac. So yeah, so I'm sorry for that one. So the palladium came to the picture, palladium, and then I template. These 2 gentlemen may be helpful to send this aryl or alkenyl moiety to the 3rd positions. They promised like that. So it ended up like this. So we can do like that, or yeah. Oh no, I just started early. Okay, so okay. And then norbornene as a transient mediator, it also can help to form the 1,3 derivatives. That is our hypothesis. What happened? Is it interesting? Yeah, is it interesting? We have some interesting results too. So this is the first result. A long journey to the unknown chemical space, that is synthesis of ferrocene 1,3 derivatives by distal CH activation via a template approach. So So we have achieved this goal by using a long engineered pyridine template. So there are palladium and using a amino acid, N-protected amino acid ligand. So recently we published, and it is accepted as a hot paper. So there are some challenges when we started this work. Mainly the stability of this thermodynamically disfavored ferrocene appended microcycle transition states. Still now it is not known, so we are very worried about that one, the stability, how far it is possible. So another one is the controlling the regioselectivity. There is a proximal ortho and meta is in valid in case of benzene systems only, or case C2 and C3 will be better for ferrocene. So C2 and C3 versus C3, and multiple CH activation. So usually in the benzene system, this template approach will lead to mono and di derivatives. So this multi-CH activation is another problem, and the compatibility of ferrocene attached template under the reaction condition obviously, and finally the easy installation and removal of this template. This template is developed by Jigwan Yu, and we have done some simple modification into this template, and we used this one. Okay. So this is a representative scope of the example of this alkynated ferrocenes with initially we tried with benzyl acrylate, and then we extended this methodology to form a number of substrates giving a very good yield, and fortunately we are able to get the single crystal structure of this one. So confirming that the first 1,3 derivative has we make sure that it is the 1,3. Okay. And then see, these are the some important natural product of the olefins. So you know that previous slides I have showed that 1,3 derivative have some material properties also, and these type of we believe that this methodology can used as a biomarker for making some natural products or biomolecules by the labeling with the ferrocene moiety. And this is the tuning and removal of the template. First the tuning, we first you made a template without a methyl group. So we came to know that the methyl group, the position of this methyl group is very crucial to make this 12-membered macrocyclophane to deliver the palladium very precisely to the meta-CH bond. So without that methyl group, no reaction, and then the fluorine is not there, then it also there is no reaction because it may somehow affecting the lone pair donating nature of this nitrogen, and obviously this nitrogen is not there, the reaction is not working. So we want to check whether the ortho CH functionalization is working with because of this amide moiety. Right, this is an amide. So there is a possibility of ortho CH functionalization. So very impressively, we didn't get any trace of the ortho functionalized product or a di functionalized product. This very selectively the meta product we got, and yeah, this is the selective removal of the directing group. So this acid reflex gave the quantitatively they give the template back, and then in situ protection give this amine 1,3 derivative. So, and then coming to the Catalani approach, we did this again the ferrocenyl amine that is commercially available. So with iodobenzene and norbornene as an 50 mole percentage. We are able to deliver this aryl moiety to the third position using the first time the ferrocenyl norbornene pallada cycle intermediate. So this is we put it in a chem archive. So I will tell about the story a little bit later. Out of time? Oh, sorry sir, 5 minutes more please. Okay. So this is a representative scope C3 arylation. Okay, so it's and then coming to the late stage diversification and etc. This is a synthetic mutation of the directing group. This dimethylamine moiety has been transformed to a methyl or a nitrile, and here comes the interesting part, the enantiomer selective synthesis of the C3 arylated ferrocenes. So in this case, we used an acetyl phenylalanine that is a norbornene that's commercially available. The acid we just esterify it and used for this enantiomer selective synthesis, and we got a pretty good enantiomer selectivity with moderate to good yield. And here the ferrocene 1,3 derivatives with both central and planar chirality is having a central chirality and planar chirality. So we have achieved synthesized a number of derivatives impressively with a very high diastereomeric ratio. And this is the possible mechanistic pathway as yeah. So and this palladium will first coordinate to the ortho position, and then migratory insertion of this norbornene, and this norbornene will relay hand over this palladium to the meta position, so that's ending to the distress EH activations, and this one we characterize by ESI-HRMS, and then oxidative addition to the palladium center aryl iodide, norbornene extrusions, and then proto-demethylation will give the product, and a kinetic isotopic study, a detailed kinetic isotopic study also we have done, and so meanwhile, Jingwanyu and co-workers has published this same results in Nature Chemistry. They used this valine and this norbornene. In our case, this is the ligand and the norbornene. So, but fortunately, we have put it in the chem archive earlier to this one. So, now what we are doing, some synthesizing some impressive ligands. And the correlative activity is screening. So, we hope for the best. And then one more result we are having, the weak chelation assisted distress CH functionalization by Catalani approach. There is a ferrocene carboxylic acid that is also very cheap, commercially available. So, this time we won't do the mistake. We initially started the enantiomer selective version with norbornene and ligand, and we achieved the 1,3 derivative synthesis and enantiomer selective synthesis of planar chiral ferrocene derivatives and post-synthetic modification, and the novel chiral ferrocene ligand synthesis is under progress. And in conclusions, as in the hypothesis, this readily available monosubstituted ferrocene, with the help of palladium and norbornene via AFNP intermediate, we are able to synthesize the 1,3 derivatives in an enantiomer selective manner, and using a designed template, we are able to via a 12-membered macrocyclophane like transition state, we are able to produce the 1,3 derivatives. One is in the arylation and another one is alkynylations. So, and post synthetic transformations and catalytic application is under progress. So, the most important slide, this is the acknowledgement. I am thankful to Professor Manmohan Kapoor for his undoubted support and guidance. Without that one, it's not possible. So, this is the current group members on the organic chemistry synthesis lab at IISc, and the entire work now has been done by Princey Gupta. He was an MS student with me. Princey and Prakash were the MS students with me, and Princey decided to stay with us for a PhD, and Prakash moved to Amsterdam with Tim Noel for PhD. And Shivam came, and Prakash with the Catalani approach with the amines, he started, and Shivam started with the acid work, and then now Princey is combining and me combining and getting the publications so far. So, and DST for Women Scientist Fellowship, without that one it is not possible. And the IISc, Director IISc, SIF facility for giving all the infrastructure facility, and thank you very much for your kind introductions. Patience listening, and coordinators for giving such an opportunity to present my work. Thank you so much.
Yeah, very good presentation, and very good, excellent work also. Congrats. Any questions from students?
Good evening, ma'am. Ma'am, if some limitations or challenges associated with the direct acceleration approaches for synthesizing discharge CH functionalization ferrocene compound.
You mean what are the challenges?
Yes, ma'am, challenges and limitations.
Limitations and challenges. The first thing is the template designing, and then introducing this template to the ferrocene backbone. That is very difficult. Actually, this is more than 3.5 years work, and we were struggling in then optimization and then characterize. The most important is the characterization. We don't know whenever we are getting the crystal that before that we are not sure that we prepare the 1,3 ferrocene derivative for the first time. So I still remember when we got the crystal, we were so surprised and happy that we prepared a 1,3 derivative. So these are the challenges, and in Catalani approaches it is a bit challenging because it's 50 more, it's like a cooperative catalysis, right? So and then the chiral ligand, the chiral norbornene derivative, and the condition, optimizing the conditions is a bit difficult task, but we can do that.
Ma'am, condition in the sense temperature, pressure related with
Yeah,
that.
in some cases we have to add silver salt as an additive, and yeah.
Okay, thank you, ma'am.
Yeah, thank you.
Yeah. Then congrats, Susitra, and small momentum.
Thank you very much.
Thank you.
So session is over. Thank you, students. Tomorrow we'll meet, okay? Tomorrow, not like today, okay? You should come in a time. Okay, good night.